Our third day, and I'm sure you're happy. We only have 2 days left, today and tomorrow is a short day, so, I'm sure everybody's ecstatic, especially the organizers. I would like to make a couple of official announcements first, and then we will move towards our lecture. So, official announcements. One thing we need to do is we need to figure out the group photo. We need a nice group photo for such a nice meeting. At 12:30, after the last student talk, we will all assemble at the entry of the hotel, there where you come in, and there will be a photographer sitting a little high, and then will kind of like photograph us on a low, sort of like a slanting view. It's nice. This has been done before at this hotel, and we'll get plenty of good pictures. Ali, was there any other announcement that I forgot? No, guess that was about it. Okay, then I would say let us start with our official first presidential lecture delivered by Edward Boyden. So why did we call it presidential lecture? In the failed attempt to squeeze some money out of the president of the University of Göttingen, it didn't work. I think it's the 5th president in 5 years or something, and one came more broke than the other. So, that's just life. So, but it's still a presidential lecture, and it's really a huge honor to have Ed here. Ed, who, we are here because of him, right? If he did not invent expansion microscopy 10 years ago, none of us would be here. We would all be doing productive work somewhere instead of sitting here listening to science. So, speaking of that, sometimes when you're an announcer, you say, and this guy needs no introduction. Then you proceed to actually do the introduction. I'm not going to do that today. He does need no introduction. I mean, if you need an introduction at Boyden, you're at the wrong conference. I'm sorry. So, and to prove that, and to prove it to Ed as well, I'll make a pop quiz. Like in your days of school, it's a pop quiz, unannounced quiz. Now I'm going to ask you things. So let's start with the easy stuff, and then we'll go towards more tricky questions later. It won't take that long. I mean, we still have the lecture to go. Where did Ed study and get his PhD degree? Shout. If you know it, shout it. Yeah? MIT, excellent from the students in the front row who trust themselves more, I guess, than others. Where did Ed go for his postdoc afterwards? Stanford. I heard it from the back. Yes, thank you. What did he invent at Stanford that we here don't care too much about, but there's probably a Nobel Prize in it? What did he invent at Stanford? Optogenetics, that's right. Where did he go after after Stanford? The answer is in the question. Where did he go after Stanford and became a professor? I just said it, MIT. What did he invent at MIT, and it was, really useful, and we actually like it? Expansion, right. Now, so, last 3 questions. Where was Ed yesterday? You paid attention. He was here. Thank you. Excellent. Last question. How long is his official CV with lists of achievements that is posted on the University of, MIT website? How long is it? It's a PDF file, so it's like, it really has a page count. How many pages does it have? Let's see who. 115 for the man in black. Exactly. Thanks a lot. And those awards really contain the best you would see, and including the Brain Prize and many, many others. So you see, Ed, you did not need any introduction. Thank you so much. We are honored to have you here, and we are looking forward to your lecture.
Great. Well, thank you so much for the kind introduction. Yeah, it's really great to be here and to see so many wonderful and fascinating discoveries and inventions, and many thanks to Silvio and Ali and the rest of the team for organizing. Not only are they great scientists, but also really builders of community, and we need both in this complex world. So, so it's really fun to get to share with you today some thoughts on expansion microscopy. I kind of redid the whole talk because I couldn't introduce all the basics in the standard way since it's already the 3rd day of the conference. But I thought it'd be fun to both talk about some sort of principles and early thoughts that maybe I've never publicly talked about before, and then also share some really new stuff that we're doing, including a really great collaboration with Silvio and Ali that's yielding some really interesting findings. So of course, in Expansion, the core original idea was to try to physically magnify biological specimens. And we had a lot of ideation in the early days of the lab about how we could do it. I dug up some old notes where he talked about even taking an object and slicing it into very thin slices and then spacing them apart. There's even a handwritten note I found where we thought about exploding the brain, and then like it would fling the pieces against the wall, and then we could reconstruct the pattern. That we didn't try that. But then this note here, I don't know if this is visible. This is from July 2007 with Brian Chow, the first postdoc who joined our group. And we were, at the time we were calling it massively internally correlated measurements. Like how would you observe lots and lots of things inside a specimen? But already we were thinking about like cell swelling and trying to see if we could pull apart objects for better labeling. At the time, we actually were focused more on decrowding for better labeling because, of course, a year before, you know, the visionary work of super resolution was starting to go big. Palm and Storm had come out the year before. Stead was finding more applications. So we kind of were thinking that nanoscale imaging was a very established thing. So we were focused more on how to decrowd things for better labeling. You know, we thought about, interesting, we wrote Hans-Uli Doth's name in the corner for some reason. He ended up writing the news and views about the Expansion paper 8 years later. I don't know what we were thinking, but we put his name in the corner for some reason. A pioneer of brain clearing techniques. We thought of elastomers. We could fill it with something stretchable and then try to stretch it out. We thought about trying to express something transgenically so that it would bind things and allow us to pull things apart. I had the habit at the time of taking constant notes during meetings, and then I would photograph the notes and then give the original to the postdoc or the student. So it was very helpful for me to like revisit thoughts. We thought whether you could just stretch the membrane and concluded it can't just be the membrane. You have to put the stretchable or swallowable substance everywhere in the tissue. And then finally, in the lower corner, we realized, hey, if we're going to stretch something, the rigidity of the tissue would matter. So what's left really has to have no structure. So it's kind of interesting that even in the early days, we were kind of trying to think about like all the different principles that we would need if we wanted to expand something. But at the time, again, super resolution kind of went big the year before. And so Brian did think about swallowable hydrogels a bit at the time. But we thought, well, it's not a high priority project because nanoscale imaging is a very well-established arena. And so I like to think that we had a bit of the knowledge about expansion at the time, but not the wisdom. And a lot of the downstream knowledge and wisdom really only arose when two then grad students, Fei Chen and Paul Tilburg, joined the group. And we tried to do nano imaging. Fei spent a big chunk of his first year in the lab building a Storm super resolution microscope, and at the time, we didn't know much microscopy in the group, so this was all very independent work by Fei and Paul. Paul spent his first year of the group trying to do electron microscopy, and we had an idea to try to build little barcodes that we could then sprinkle around in a living cell so that when you did EM, you could identify the proteins. And you can find his master's thesis online, which has some of our attempts to do this, but this is pre-AlphaFold, the modern era of generative AI protein design, and so we didn't have much success. And so after a year of this struggle, we realized, wow, nanoimaging is much harder than we thought. And so we started wondering, like, well, what could we do? What is something that would be possible that would allow nanoimaging to be done by anybody, even people like us, where we were struggling to do highly technical things. And so we started brainstorming and thought, well, why don't we give this a try, the expansion? And Fei and Paul very independently created all sorts of wonderful ideas and thought up lots of the debugging. And within probably less than a year since starting the project, we were getting expansions that looked reasonable. And so the first paper, of course, that we published was using a form of the chemistry that's not super practical. And we've already heard about many improvements on it over the last couple of days. But it still allowed us to show a discovery, I suppose, which is that we could take a piece of the brain, this is mouse brain tissue, and then form this sodium polyacrylate mesh that's cross-linked. That's really important because that helps you with nanoscale preservation information. And then when we soften the specimen and added water, we could swell it. And so Daniel Rand, a person who joined the group as a photographer, actually, he had been an artist beforehand, made this beautiful little time-lapse video with some of the other students in the group, taking a piece of the brain and forming the sodium polyacrylate hydrogel earlier. And then it's about half an hour, sped up to half a minute, but then we add the water right there, and then you can see it swelling. So we made this video in part because in the beginning, a lot of people just didn't really believe it. And so this allowed us to show in seminars and so forth that this was real. Although I guess in this era of generative AI video, maybe this is less compelling than 10 years ago. Okay, so when we started buckling down to business, I guess around mid-2012 was when we really were realizing that we needed to think of ways that we could do nano imaging that are much more scalable. Like if you wanted to image a whole mammalian brain, and we were struggling with basic nano imaging, but if you wanted to image a whole brain, how would that be possible? And we were very inspired by this paper from Toyoichi Tanaka and colleagues in 1980. He was an MIT physicist who unfortunately passed away relatively young of a heart attack playing tennis. In 1995, when I got to MIT for my undergrad, I wanted to do undergrad research, and he was actually the first professor I emailed. Never heard back, unfortunately. But when we started reading lots of papers about swallowable gels, this paper sort of jumped out, and I think Faye and Paul found it independently as well. But the basic idea is that if you take an ionic hydrogel, so a highly charged polymer backbone with an aqueous environment, but a reasonably high salt concentration, and then you add water to it, then you get this beautiful phase transition-like physics. And we need this little animation just to sort of dramatize it. We bring in water, the polymer chains are the white lines, and osmotic force brings in the water. And then the polymer backbones are extremely highly charged. The ones that I guess a lot of us use have carboxyl groups all over the place, so negative charge. And as the water comes in, you get decreased charge screening. And then the polymer backbones will have a lot of exposed negative charge and get repulsion. And the beautiful phase transition-like physics that Tanaka and colleagues worked out, we thought, wow, maybe this is well behaved enough that maybe you could actually do this in an information preserving way. Now you have to get the polymer inside, of course, and when we were kind of going over all the literature at a later point, I don't think we had actually found this paper right away. We found it at a later point. It was curious to find that in 1981, there's this paper by Christian Dreyer and Peter Hausen where they actually did hydrogel embedding with polyacrylamide, sort of the uncharged cousin of polyacrylate, if you will. And they showed that they could take preserved embryos and tadpoles and do a preservation with this polyacrylamide hydrogel and show that this facilitated staining and imaging, and the hydrogel preserved the integrity of the specimen. So it's kind of fun to think that if somebody had put two and two together, maybe this could have happened half a century ago. So the core idea then that we hoped for, and of course a lot of the effort since then has been on trying to prove it to be the case, and if it isn't proven, then to improve the technology, is basically dramatized by this little cartoon. So can we take a cell like this golden brain cell on the left and make it look like something on the right? So in the ideal case, two molecules that we're touching are now some minimum and hopefully reliable distance apart, and two molecules that are some other distance apart will be scaled up by a linear factor. That would be the ideal. Expansion protocols, we've heard about many of them already, but almost all of them have more or less the same four kinds of steps. So of course we have some kind of anchoring strategy. So in this cartoon, proteins are in brown, and we have covalent anchors that will bind to, for example, amines off of an NHS ester group, or more recently, many groups have built very clever anchoring moieties, including many invented by people in this room. A couple of years ago, we had a paper led by Yi Kui and Gao Jiang where we worked on epoxide anchors, for example, trying to harness more of the anchoring possibilities across different reactive groups. Then, of course, there's the polymer formation, and the original polymer strategy we used was free radical assembly of sodium acrylate monomers with bisacrylamide crosslinkers, it's sort of striking how similar the recipe was to the 1981 paper I mentioned, except of course they use uncharged monomers. And of course, a lot of creativity by many people around this room has been on improving the polymers, changing the monomer compositions or the style of polymerization, and achieving expansion factors that can vary in size. Our original protocol only expanded things about 4-fold, but then many pioneering efforts, many by people in this room, have been able to extend it to 10-fold and even more. The third step is softening. In the original protocol, we used proteinases to chop proteins into bits, but more recently, many people have come up with clever softening strategies involving detergents and heat, the ability to use other enzymes to soften bacterial cell walls, the ability to soften complex tissues with tough structures ranging from the cell walls of yeast to bone and cartilage. And then finally, we add water and we get a swelling of the polymer. And so I kind of titled my talk, this democratized platform, because I think one of the things that's been so striking is how the community has been able to create many, many inventions and discoveries of how to do expansion that we did not envision. And so it's been kind of fun to be part of a community that's so collaborative and so creative in its efforts. If we built a hardware microscopy platform, there's no way that everybody would be able to modify it on their own, but it's sort of an interesting possibility where if somebody is really great at a biological question, their knowledge is enough in many cases to invent a method of expansion that's appropriate for it in the case that the existing tools are not adequate. And so it's been a lot of fun just to sort of see the surprises that people have come up with working on their own, and then this conference, which really is the first global conference of this kind, it's been just a real joy to see what everybody's been doing. So in the early days, of course, one of the questions was how can we help people understand the technology, and in part to understand its shortcomings. And so we tried to be very upfront about the shortcomings from day one. In fact, the first panel in the original expansion paper is about the distortion that expansion introduces. We always thought that by being hyper-transparent, no pun intended, about all of the shortcomings, that not only would it build trust in the technology, but people would also understand where they could contribute and innovate as well, although it happened to a far more explosive degree than we had even imagined. So in the beginning, we spent a lot of time trying to define standards and metrics, and of course, many people around this room have come up with improved ways of doing this, which has really been wonderful. But the initial paper, we kind of outlined two standards, which would be how much distortion are we introducing? Because of course you will get some when you have any process, and hydrogel swelling is a process that does have some distortion. And so after a lot of thinking about this, Faye and Paul and I decided, well, let's just try to do pre-expansion imaging with a classical super-resolution technique. At this time, I think MIT had a SIM super-resolution microscope, so we used that, although later we were able to collaborate with others on other techniques. And then just to take pre-expansion images with the classical super-resolution technique, here shown in purple, and then the post-expansion image would be taken on a regular old diffraction-limited microscope, and here it's shown in green. And then we calculated a vector field of distortion, which would allow us to measure the amount of distortion. And so it wasn't zero, of course, but it was small enough, a few percent over, let's say, a microscope field of view on a typical 40x, 63x lens, that we thought people can start to answer biological questions with it. The other thing that we wanted to figure out how to do was how to measure resolution. And resolution, of course, is a very formal definition of the distance between two things that can be resolved. And the way it's often formulated, if you think about like the diffraction fringes off of a point, is meant to be very lens-centric. So we had to think a little bit about what are other measures of resolution. Now, of course, there's many ways of measuring resolution that are quite elegant. But in the beginning, we thought, let's just look for natural scale bars. Microtubules. More recently, of course, many people around this room have been developing wonderful ways of measuring distortion and resolution by expanding objects of known size and shape, and it's been really wonderful to see much better ways of building little natural scale bars or artificial scale bars being invented by the community. But the core idea of let's find something that we know, and then we can see how much either error we've introduced or whether 2 points that we want to distinguish, we no longer can distinguish. And so, the polymer itself, has a mesh size or spacing that's quite small. Maybe the polymer threads are only 1 to 2 nanometers apart. But of course, at the beginning, we were limited by expansion factor. And a 4.5-fold expansion would take a 300 nanometer resolution lens divided by 4.5. You get about 70 nanometers or so. But of course, many people in this wonderful community have developed better, higher expansion factor protocols that go beyond that. In the beginning, we often were trying to compare what we saw to super resolution as the gold standard and the, a stunning achievement in its own right, of course, with many amazing pioneers represented here, of course, but also taking advantage of different physical and chemical tricks. And so the first thing that we tried to do was simply to look at synaptic stains with antibodies against proteins like Bassoon and Homer. And so left is pre-expansion, right is post. And then we would look at the distance between these protein centroids and compared, in this case, for example, to work done by Catherine Dulac and Zhaowei Zhuang using storm super resolution, developed in this case by Zhaowei's group, and found that we could get the same distance, but this is now measured on a confocal microscope. One thing that's been a lot of fun, and I think we've heard a bit about this already from several talks, are the side effects of expansion. And one of the things that was really striking when we first did it was how transparent things became. So this is one of our early pictures that we took of a piece of the mouse brain, and then after we expand it 4.5 times in each direction, and it's very transparent. And so it's been really exciting to see how people in the community have either developed applications of this or optimizations of it, including, for example, light sheet imaging, where you can image from one side by shining light from an orthogonal side, and then you can go at blazingly fast speeds. So, so those are sort of the early principles that we tried to work out in terms of like steps of expansion and ways of validating it. As I prepared this lecture, I was kind of digging around, and it was fun to see some of the reactions that we got at the beginning. We had a bunch of grants that got rejected on it, and then a foundation called Open Philanthropy in March 2016, so actually a year after our first paper came out, bit the bullet and gave us a gift. They're very transparent in their grant giving, so you can read this, I guess, 30-page report about it online if you want to. This is all public information. Like, success is realistic if highly uncertain, unconventional. They drove how it's highly uncertain a second time, just in case we failed. They have some cover, and very speculative and too speculative. So, but they wrote us a check, so I'm not complaining. But yeah, it's sort of an amusing read just to see how. Even after we had published it, there was still, I think, a lot of concern. We started engaging with people in the super resolution community, and that's been a lot of fun. People in this community are so wonderful, and many people were simultaneously skeptical and encouraging, which I think is probably the healthiest attitude. This is some work that Eric Betzig led with close collaboration from us, and they published a paper in 2019, but the backstory is kind of a fun depiction. This is from a little Howard Hughes press release that came along with this paper, where we did expansion together with Eric's group and then scanned the samples on a lattice light sheet microscope, originally designed by their group. Eric didn't expect the experiment to work. Two scientists, Ray Gao and Shosano, postdocs from our group, wanted to try it out. The light sheet from their group on the expansion, I thought they were full of it, Betzig remembers. The idea does sound a bit crude, Ray admits. I was going to show them, he laughs. And then, and it was great to get like very constructive criticism, right? And it allowed us to think about our work and to work with people who knew, far more about microscopy than we did. And that's been, I think, a real wonderful hallmark of this community, is just how eager people are to share, but to be constructive in a way that is maybe rare in many fields, and it's been such a wonderful joy to be part of this community where you can see technologies and results and ideas flow in ways towards maximum impact and democratization. These are some images from that paper that was led by Eric and us. So we were expanding and then doing lattice light sheet microscopy, taking advantage of the transparency of the sample. These are using fluorescent antibodies to decorate different proteins, looking at mitochondria and lysosomal proteins, myelination, work that was triply led by Ray Gao, Shou Asano, and Goku Apariula. we collected a lot of interesting data sets at the time. The paper came out in 2019, but of course we were doing the work over the years leading up to it. And it was really just a lot of fun to be working side by side with somebody who could really coach us on what kinds of things they were looking for, and to be in a, they were doing the work at Geneva, and to immerse in sort of microscopy culture and to understand what kinds of things were important. And so even though we were continuing to work on fundamental chemistry back at MIT, the feedback from the super resolution community was very, very powerful. This is one of my favorite data sets from that paper. 3D rendering done by a visiting scientist from Toshiba Memory, of all places, Yosuke Bando. This is a fruit fly brain where all the dopamine neurons are expressing a fluorescent protein. The color codes are done by hand, just by human annotation, but it's the whole fly brain. A subset of labels not shown here are synapses being antibody labeled, but it was a lot of fun just to sort of see the intrinsic multi-scale nature of what one could do with expansion coupled to sort of an expansion appropriate microscopy platform. So back at MIT, we were trying to figure out ways to extend the toolbox and, in hindsight, really trying to outline principles of how to go about it. We had a quick follow-on paper to try to get to work in a more user-friendly way, and Josh Vaughn and Kwang-Hun Chung had independent papers around the same time to show how to use all off-the-shelf chemicals to do expansion. We tried also to explore different kinds of stain, in this case expressing fluorescent proteins, which we can sometimes also boost with antibody staining. This is usually the so-called rainbow technique developed by Jeff Littman and colleagues, so combinatorial expression of fluorophores. And then without expansion, you get beautiful pictures, but the finest processes are very difficult to see. And the upper right is the same field of view as the upper middle, but after expansion, and then you can see those fine processes just fine. And so many groups are using some kind of combinatorial labeling plus expansion to try to delineate brain circuits. E11 Bio, which is led by a former student from our group, Andrew Payne, and you'll hear, I guess, about some of these things that they're doing, is trying to collaborate with our group, try to take barcoding to its extreme, and in order to make it easier to trace brain circuits. So I mentioned at the beginning the need to anchor different molecules to expand them away from each other, and this has been a very ripe arena for creativity by many people here. You heard about Yongshin and the Magnify protocol and other protocols where by cleverly thinking about how to couple molecules to the polymer, you can get different kinds of outcomes. Our first hint of this was when Ozwasi had joined our team, and working with Faye and others, we built a two-part mixture which would then allow nucleic acids to be anchored. So we use an alkylating reagent that will bind to guanines, and then a second component that contains a vinyl group that can participate in polymerization. And we found that we could anchor and expand away from each other nucleic acids, in this case RNA. And these are cell cultures where we have pre versus post-expansion images, and we're doing post-expansion fluorescent in situ hybridization. And it was very pleasing to see that we could also preserve RNA integrity throughout the process. RNA being very fragile. At the time, it wasn't obvious that you could get, go through a free radical polymerization process with many steps and have decent RNA integrity, but it was very pleasing to see that it worked okay. And then we also could do this in brain tissues, and then as I alluded to, GB Chang, when he was a postdoc in the group, and he's here as well, leading his own group with amazing work, we came up with this idea to iteratively expand. So a lot of creativity has been developed, much by people around this room, to expand bigger by changing the polymer recipe, but we were concerned that if you expand too big, of course, that maybe you eventually run out of steam, right? You can't expand arbitrarily large, and so we came up with this idea to take a specimen and polymerize it, expand, form a second polymer in the space opened up by the first expansion, and expand it again. And so if you could do a 4-fold expansion, 4 times 4 gives you almost 20-fold expansion, and we were able to, for example, see microtubule sidewalls and other things that are difficult to see on a conventional microscope if you only have a 4-fold expansion. Later, Deblina Sarkar, Jinyang Kang, and Ozwasi developed a sort of a different version of this that keeps all the proteins anchored to the first polymer, and then when we form the second polymer, we can expand the first polymer anchored proteins away a second time. So it uses off-the-shelf chemicals. We also use this to explore another topic, which I think has come up beautifully in several of the presentations, which is this idea of decrowding, right? So proteins and other biomolecules, the way molecules work, of course, is largely by touching each other, and then you get bonds formed or cut or changes in shape. But that also means that many things you stain for might be inaccessible. So in this cartoon, the red and green antibodies can decorate proteins on the outside of a complex. They can't get inside. There are creative ways to solve that. We've heard a bit about nanobodies, for example. But if you want to use conventional antibodies, it's still a challenge in many cases. So by pulling the molecules apart, we thought we could try to stain molecules inside these complexes. And so working with Tom Blanpede at the University of Maryland and Li Wei Sai at MIT, we started looking at crowded protein arenas within cells. And of course, synapses are chock full of proteins, very densely packed. And so we started this sort of a crowded slide, but we characterized different proteins like a calcium channel subunit, RIM1 to PSC95, where the pre-expansion staining with at least some antibodies was not as good as we hoped for. That's the yellow channel, and there's not much yellow to be seen. The pink channel is with post-expansion staining, and we saw increases at staining at synapses because we have other reference channels, but it was very refreshing to see that we could add them in a specific way. Not all proteins are crowded. Here's our old friends Homer and Bassoon, and you can see the yellow and the pink are more similar. Here's Shank3, another synaptic scaffolding protein as well. And so working with Tom, we were investigating some of these ideas that were first discovered with super resolution microscopy and other earlier techniques like nanocolumns, where pre- and post-synaptic proteins are aligned. And so now that we can see this calcium channel subunit, the CAV2.1 subunit a bit better, we were able to, for the first time to my knowledge anyway, to characterize this in intact brain tissue and to see the alignment of the CAV2.1 in the presynapse with the postsynaptic PSC95. Another theme we've heard a lot about at the conference is multiplexing. And so one thing that Jinyoung Kang and a then grad student in the lab, Margaret Schroeder, did was to show that by expanding and decrowding, we also could facilitate multiplexing. We had to come up with alignment software to align the images we get after we stain, image, and wash over many cycles. And it's been fun hearing how different people have been pioneering approaches to alignment and stitching and other image processing strategies at this conference. But we're very excited to sort of do, in this case, this is 20 antibodies over 4 rounds with approximately 20 nanometer resolution because we're iterating. And you can see here different stains that we could then overlay and look for geometries of expansion-related data. It's been also a lot of fun to work with fantastic chemists as well. We started a collaboration with Laura Kiesling in MIT chemistry. And together we came up with some ideas in a project led by Shiwei Wang and Tei Shin, working across the two groups about how to build from this wonderful DNA-based polymer that Sylvia's lab developed. So we can change the oxygen control, we can change a few aspects of the recipe, but not any entirely novel concepts there. But we were able to get 20-fold expansion in a single step and use that to basically shorten the labor that's required from a iterative experiment into a shorter duration. Paulo Valdez, a neurosurgeon working with us, started to investigate whether if we could decrowd proteins, this could have translational impacts. He works with brain cancer patients. He's a neurosurgeon who removes brain tumors. And so collaborating with Nino Kyoka, his co-mentor, we looked at aggressive tumor cells, which have high levels of vimentin, here stained for in cyan, and GFAP, stained for in pink. So these are actual human specimens from biopsies. And these first couple rows are pre-expansion staining. And then this row here, where my mouse is pointing, is the same specimen, same antibodies, but post-expansion. And Pablo took another step, by the way, which was kind of clever. He doesn't care about the resolution necessarily. He just wants better staining. So in this fourth row, you had water to expand. He actually added salt to shrink it back down. So it's what you might call a 1x expansion, for lack of a better term. So you get a lot better staining, as you can see. But if you don't need the extra resolution, I mean, all other things being equal, a larger object will be slower to scan than a smaller object, right? And if you're in the operating room, you might want to go as fast as possible. So it was kind of nice to know that even if you don't need the resolution, the staining improvement, even on top of antigen retrieval and everything, we did that in the control case here, could be useful. And then Pablo convened several scientists to count these putative aggressive tumor cells, the Vimentin GFAP double positives, and you get about 6 times more of these putative aggressive tumor cells with post-expansion staining at 1x magnification than if you do pre-expansion staining. So is this a new cell type? Is it a different cell type? We don't know. This is a technology paper. But it's intriguing to think that the improvement in staining might be a useful kind of thing to consider, even if resolution is not of interest. Yongshin Zhao, who's also here, presenting his wonderful group's work, when he was a postdoc in the lab, co-led the way on trying to develop a general pathology optimized form of expansion, speaking of pathology, and develop some pre-treatment steps and optimizations so that you could look at all sorts of human tissues, even aggressively preserved FFPE tissues, prostate and lung and breast and pancreas. Left is normal and right is cancer-containing specimens. The color code is in the upper right. And this work, I think, really illustrated how you could take the expansion steps and then fit them into other workflows, right? You could have it as a final step after different pre-processings. And in the years since, much creativity has been spent in developing all sorts of clever pre-processings. Or you could feed the expansion into other things downstream. You could then use as a preparatory step for other kinds of processing. Shahar Alon, who you've heard from already, had used expansion as a way to prepare for in situ sequencing, and work that was co-led along with some other folks, Dan Goodwin, Anu Sinha, Oswasi and Fei Chen, close collaboration with Aviv Regev and George Church. And so the idea of expansion is kind of like a step that can be used to precede a existing strategy or to follow an existing strategy. I think has been a really powerful and amazing emergence from the community at large. We focused a lot on teaching. Pre-COVID, we actually helped host several in-person workshops, including at Geneva and at the Jean-Paul Lamothe Institute and at other places. During summer 2020, this of course would not be possible, so we posted online some photographic step-by-step tutorials and tried to keep the teaching alive during that time. And it's been really exciting to see the continued spread of and innovation in different technological arenas. In the last part of the talk, I want to talk about some new things that we're doing that I'm excited about. And some of this is very early and preliminary, but I wanted to share it with you because it's exciting. And then I want to close by talking a bit about how we hope to apply expansion in our own group. Up till now, we mostly have been focused on tool development, but increasingly, the tools are so good. Let's start trying to use them for some scientific questions of our own as well. So I'll tell you 2 last short stories about new directions. I wanted to start by just acknowledging this wonderful work led by Ali and Silvio, which I think really opened up a new frontier in thinking about how expansion both works fundamentally, but also might have applications that are very non-intuitive, I think, from the way we originally were thinking about it in the very beginning. And of course, they did this amazing study where they could take a protein, anchor it to the polymer, chop into pieces, and pull the pieces apart. This is in contrast to pulling molecules apart, stain it, and then do surf super resolution. And these pictures, when they first showed it to me, were just totally stunning. You can see the shapes of proteins. This is one of my favorite side-by-side comparisons from their beautiful paper, where they showed the ground truth, if you will, in gray, like a crystal structure, or increasingly AlphaFold predictions are considered to be gold standards as well. And then these false color images are single images of expanded protein fragments. And so it's just really a beautiful privilege to get to see some of these images when they first came out. And then they went on to do this amazing, and I'm sure I'm not acknowledging all the people on their paper who contributed different aspects of this, but these beautiful cryo-EM style averagings, where here's like the GABA receptor shown in crystal structure form. Here's the AlphaFold predictions. You get those dangling tails that I guess don't crystallize. And then this is their cryo-EM style averaging of their what they call one microscopy. And so it's just a really beautiful discovery, you could argue, about what polymers can do, as well as a beautiful application. And so we've continued this collaboration. I want to talk a bit about work that is being led by a graduate student in our group, Helena Hu, who's a biological engineering PhD student at MIT. And so she's been trying to see if we could take the iterative expansion concept, and by tuning the hydrogel and also thinking creatively about the steps that are involved, whether you could expand something so big that you could maybe get these kinds of pioneering images, but with a completely ordinary microscope. And so this is, I guess these are the first time I'm showing these slides publicly, so they're a little bit early stage, but I thought it'd be fun to let you all have a sneak peek at it. But the core idea is to take iterative expansion and try to do way many rounds, so 4 rounds here. And this to me also is sort of a surprising polymer discovery, that you could stretch something that many times, because she's taking a specimen and polymerizing it, expanding it, polymerizing again, expanding it, and repeating this 4 times. And if you do this, you can get very large expansion factors, 1,000-fold expansions. These are some of the first images showed me just of the gel being expandable that much, which just showed this on a plate. It was very striking just to see this macroscopic object. And so collaborating with Silvio and Ali's groups, now we're trying to figure out how can we validate this expansion procedure. And so in this very close collaboration with them, we've been taking a cue from some of the amazing insights that they derived from the first study. And so here is M-cling, this beautiful chain of lysines. You also can have a fluorophore on the end, and it's of course can be used for classical purposes like staining membranes, but also we could use it just as a ruler, going back to the idea of a scale bar. And so we started trying to do anchoring, controlled fragmentation, and then 4 times iteration, working with Silvio and Ali and in their groups for a significant part of the project as well. And you can start to see what look to the eye like chains of individual fluorescently labeled amino acids. So of course these are examples. Right now our groups are all engaged in working on quantitative validation of these. And so these are just some images that I've tried to compare the shapes of these under different expansions to some models of what they would look like. And so in black you can see here some modeling, and then the green peaks are the fluoresceins. So it's at 1000x, so it's really beautiful to see some quantitation that points in the direction of measuring the distance between these. And then just this morning, Helena sent me some new figures that are so new that I just assembled these in the minutes before coming here. But she's been trying to come up with some automated ways of tracing down the chain of lysines. So M-cling has 7 lysines, and then you have a fluorophore on the end, and then the other ones you stain. Lysines having an amine, you can stain them. And she's trying to trace them in 3D. So this is a handful of data. And so this is very early point in the analysis for 1000x expansion. And we're trying to come up with some bootstrapping statistics so we can compare, the nearest neighbor distances to how big the distances would be if you shuffle the position of all the fluorophores in the region. And so it's early days. I think we still need to do a little bit more statistical analysis to really nail it. But the red line here is the mean observed distance between lysines. I'm sorry, this is the distance between the fluorophore at the end of the M-cling and the first lysine. This is the lysine-lysine spacing. The red dotted line is the observed spacing, and then this distribution is what you see if you shuffle the positions of the lysines randomly in space, sort of as like a control condition, just to make sure that we're not, fooling ourselves into thinking that we're seeing something. And it's exciting to see that the observed spacings are much closer than the shuffled spacings. So more work to be done, but very intriguing to think that this kind of expansion protocol can exploit the the beautiful properties of the gel that Silvio and Ali's groups have foundationally found. Another thing that we're thinking a lot about is protein sequencing. Can you sequence proteins inside a specimen? The ability to see the shape of the protein, as I mentioned earlier, is of course amazing. But if you want to look at detailed sequences of amino acids, can we do that? And it's quite possible that with some clever chemistry, different approaches could be used, but in work led by Camille Mitchell, with a great collaborative synergy with Sarah Tavana and Joanne Peng, we've been trying to figure out if we could get the steps of protein sequencing to work inside the expansion gel. So the beginning of this procedure, as schematized here, would be a bit like what you've heard about before. Take a protein, anchor it to the polymer, chop into pieces, pull the pieces apart from each other, as both in the one paper that Ali and Silvio led, and now in what Helene is doing. But now, once you have these fragments, could you use binders of amino acids, say on the N-terminus for sake of argument, and then use Edmond degradation to chop off one amino acid at a time? So Edmond degradation, of course, goes back well over half a century as a classical way of doing protein sequencing, still one of the gold standard ways of doing it, just because protein sequencing is so hard. Many people are trying to invent successors like nanopore methods and so forth, but to my knowledge, none of those have really gone mainstream quite yet. So we thought, what if we try to get Edmond degradation to work inside the gel, and then we could use N-terminal binders, maybe generative AI would help us do that, and then recognize the N-terminal amino acids one by one. Now, Edmond degradation is a fairly harsh procedure. You use trifluoroacetic acid at a reasonably high temperature. There's a lot of stuff going on. And so when we set out to do this, and here, of course, is expansion gel, which I guess doesn't need too much explanation in this audience, but would all just be compatible, right? And even some of the reactive groups in the expansion gel look a little bit, biological, right? So would we just mess everything up? So in this wonderful work, the first step was to figure out, well, are there any solvents that are compatible with Edmond degradation that don't totally mess up the hydrogel? And so we looked at pyridine and acetonitrile and DMSO and just screened through lots of solvents that could be used for the Edmond conjugation. I guess I should describe Edmond degradation in a few sentences because it may not be familiar to everybody, but you conjugate an Edmond reagent onto the N-terminus, which adds this group here. And then when you add trifluoroacetic acid, you get a series of reactions that basically cause the N-terminal amino acid to fall off. And then classically, you would then use some method to recognize it, maybe mass spec or whatever. But what we want to do is to have the protein anchored to the polymer and then recognize the amino acid that is in the polymer. We don't want to look at the bulk washout, right? Because you would then lose the position information. So that just gives you a bit of background. So yeah, you need a solvent that is compatible with the Edmond reagent, which is hydrophobic. So we explored which solvents would allow you to not just collapse the gel into a little solid block, which would then present any downstream chemistry. And so some of these solvents like formamide actually did not cause gel collapse. Others like pyridine or acetonitrile just totally collapsed the gel until it was like a chunk of plastic, basically. So that was very useful to know that some of these solvents actually would be gel compatible. Then the next step would be to know, could we actually do the Edmond reagent, PITC, conjugation onto the N-terminus? And so it's a crowded slide, but just focus on where my mouse is pointing. Basically, we did mass spec to gel conjugated peptides. We used trypsin then to chop off the peptide so we could mass spec it. And here you can see a positive signal for the PITC conjugated peptide or Edmond conjugated peptide. So we were able to get the first step to work in the gel with pretty good yield. And then the next question is, can you actually sequence? Can you do the trifluoroacetic acid step, which of course is maybe the harshest step in the procedure? And so to make a long story short here, we took some peptides and immobilized them in a gel. We also put a click group so we could add a fluorophore, and that way we could monitor the process of Edmond degradation throughout the process of different sequential chemistry steps. Whoops. And so here you can see a gel glowing because we can click on a fluorophore and see the peptide. If you add PITC, you can still click on a fluorophore and see the peptide. TFA by itself doesn't do a whole lot, but it's nice to know that it didn't ruin the gel. That was not completely obvious when we started it. But this is sort of the take-home. If we added PITC and TFA, the gel goes dark. We lose that N-terminal amino acid. And this is sort of cross-sections of the gel taken on a confocal microscope here. These are all the conditions where we didn't have everything. And then if we add the PITC and TFA, we get about a 70% yield. So it's not perfect yet. But it was nice to know, to go from 0 to 1 here, that we can get the basic steps operational in the gel. This green thing below here is like a control, chopping off all the peptide with trypsin. So we thought there's more chemistry to be improved, but some people are building N-terminal amino acid binders. They're not very good yet. The affinity is not optimal. They're like micromolar affinity. Obviously, we'd like to be much higher if we want to get really good images. But we thought, let's do some bulk fluorescence imaging. With micromolar affinity, probably single molecule imaging would be futile. But we thought, let's do some bulk imaging and just see if we can recognize the N-terminus with a binder in the gel and see if we can do one step of Edmond degradation. So we use this CLIP-S2 STV1 variant that's previously published by another group. Many groups are building these wonderful binders. This is all work from other groups to make them. And we've just been getting them from different companies or groups out there. Then you can add Alexa-48 to an antibody that binds an HA tag fused to it, work that we collaborate with Nick Polizzi's group on. And so if you have phenylalanine on the end of a peptide, then this phenylalanine-tuned binder will recognize it. You can see the gel glows when we add the binder. And if we have alanine, tryptophan, or tyrosine on the N-terminus, then you don't get much binding at all, which is consistent with what the previous literature showed. So that just shows we can recognize an N-terminus. And so we thought for a capstone demo, let's just try to put it all together. So let's have a peptide with phenylalanine on the end and see if we can recognize it and then show that the fluorescence goes away when we do one step of Edmond degradation. And then let's also put phenylalanine in position 2. It should be invisible when we start the process, but it should be unmasked when we do Edmond degradation. And so, with the acknowledged caveat of the 70% yield, it looks like we could get a basic demonstration of this. So you just focus where my mouse is pointing. If phenylalanine is on the end, the binder glows. Add PITC, now you block the binder, right? So that's why it goes dark here. TFA by itself doesn't do much. But if you put PITC on the end and then TFA, you chop it off, the phenylalanine is gone, the binder is nowhere to target, and that's what you see here in the gel cross-sections. So it glows when the phenylalanine is there and not obscured by the PITC, the Edmond reagent, and then it's gone when you chop it off. Okay, this is sort of our holding our breath to see if it all would work if we run it in the other order. You could also imagine that the Edmond, the trifluoroacetic acid or other treatments could just mess everything up. So we wanted to see if we could unmask a phenylalanine and still bind it. So again, if the phenylalanine is in position 2, the binder can't recognize it because it's obscured by the actual N-terminal amino acid. Adding PITC doesn't help. TFA by itself doesn't help. But if you add PITC and TFA, suddenly it goes bright. Now we've unmasked the phenylalanine and it can bind. And that's what this cross-sections show. So we're very excited about this because I think if we could start to analyze proteins in different ways, and maybe just analogous to how MERFISH and ways of tiling RNAs with probes and in situ sequencing and going base by base are very complementary and helpful in the RNA world. Maybe there are also complementary pictures in the protein world for fingerprinting and shape recognition and sequencing to answer different kinds of questions. And it's exciting to think about how all of these could benefit from the expansion context. I have a bit more time because this is an hour-long talk, so I wanted to talk about just a few other little things, although this is the end of the expansion part. I mentioned Daniel Oran, a photographer who joined our group, who did that, helped lead that beautiful time-lapse video. And we started working on a project to basically run expansion microscopy in reverse. So start with a swollen hydrogel, photo pattern interesting fluorophores inside of it at different points by using, for example, a 2-photon laser, anchor interesting things like nanoparticles at those points, and then dehydrate it to shrink it down. Could you do cheap nanofabrication without needing, zillion-dollar nanofabrication plants? And actually, I think Daniel's earlier training in photography actually maybe was very helpful because this is a little bit, metaphorically speaking, about how you develop a picture, right? You have your picture, and then you put it into a series of chemicals, and if it's a silver or other chemistry, you then basically nucleate interesting things onto the picture target, right? So it's sort of like a photography idea. That's a metaphor that Dan came up with. And so Dan and Sam Rodriguez are the co-first authors of this. Anyway, we can shrink things by about 10-fold. Not too surprising in that aspect, but and maybe it's not too surprising for this audience, but a lot of people that we talked to back then were a bit skeptical. You do preserve nanoscale information as you shrink. So here we are writing just fluorescein with a 2-photon laser line, and then we're depositing, in this case, metal nanoparticles and shrinking it down, and then we can do electron microscopy to look at the line width. And so a 500 nanometer line width after 10-fold shrinkage is 50 nanometers. And each point here is an individual line within sample, and then each yellow bar is a different sample. So it's kind of nice to see that we could reliably write lines of metal, and you can even make conductive wires. And then for fun, we tried to show that you could make something that you couldn't make with other techniques. In this case, we decided to try to make a non-continuous, a non-self-supporting structure out of metal. This thing has no function, just showing off, I guess. But we are very intrigued now about whether you could build interesting, optical metamaterials, optical computing. We're starting to learn more about what applications this could have that are useful, and be able to specify, for example, the refractive index of different points in space in a programmable way could be very useful if you want to do optical computing or other kinds of studies. So I think I'd like to start closing now, because we're almost at the hour. So far, we've been primarily a tool development group, but we started also thinking about using the tools in-house. And I trained as a physicist, and so the idea of solving something very small, very completely has a lot of appeal to me. And the history of biology, I think, also points that direction. The first genome to be sequenced was not the human genome, it was a small bacteriophage. And so what's the equivalent in the nervous system? Well, many pioneering scientists, and we've heard some data in previous talks, are related to model organisms that are quite small, like the larval zebrafish or the worm skelegans. And so when Limor Freifeld was a postdoc in our group, she worked out the expansion of the larval zebrafish brain. This is kind of our slow entry into thinking about small brain neuroscience. J.U., when he was a student in the group, worked out expansion of C. elegans, which is really quite tough to do. The cuticle, the outer coating of the worm, is a real beast to confront. And so a lot of, to be honest, we're still working on improved versions of this in order to get really great expansion of the worm. And so my dream is, can we combine this with earlier tools that I don't have time to talk about today, but the first project that our group did was optogenetic control of neural activity, which is a fairly mature toolbox. So we have light-activated proteins from the natural world. We express them in neurons genetically, and by shining light on them, you could turn, electrically speaking, neurons on or off. And then more recently, we've been working on ways to image neural activity as well. This is some work in a preprint from Ziguan Wang in our group to build a really fast light sheet microscope and to try to image voltage across an entire zebrafish brain. So these traces might look like electrical traces, but they're actually voltage traces. And by using positron 2 from Geneva as the genetically encoded voltage indicator, but by building a very fast microscope, we are now trying to scan the voltage of neurons across an entire brain. So where this would head, ideally, and this is just an idea, but we have a growing cast of characters that we're working with to try to think about it. What if we could image all the neural activity and perturb it using the voltage imaging and the optogenetics, as shown in the upper left, and then we build molecular maps using expansion, as shown in the lower left? Can we start building models of the nervous system? And in particular, network models that just use activity and maybe a bit of connectivity to try to understand whether we can predict the future of a nervous system. But importantly, with the molecules, how do we connect underlying mechanisms to those molecules? And so where expansion, I'm very excited about the potential, is in this purple part here. Can we use the molecular maps and just do bottom-up models from the building blocks of life to physiology? Because molecular mapping can scale really well, right? Physiological imaging is really hard. But I think, as we've heard during this conference, molecular mapping could probably scale arbitrarily, right? Whereas imaging all the voltage of a brain, we can do that in a fish, but can we do it in a mouse? Probably not anytime soon, right? And then also making the glue. If we can learn to predict physiology from molecules, this green area in the middle, then maybe the kinds of work that many people around this room are doing could be used in this era of machine learning to build predictive models of physiology based upon molecular recipes alone. So I'm very excited about that possibility. I hope I've acknowledged everybody along the way who's led these projects. I think I did, and I apologize if I didn't. But I do want to acknowledge a really long list of other people in the group and alumni who've helped with these other projects over the years. And then an even longer list of collaborators we work with all over the world. These are just the PIs of groups that we've worked with on developing or applying tools. And so it's been just a lot of fun to be part of this community and other neuroengineering communities. Our website, synthneuro.org, has I try to keep it up to date with protocols and papers. And in addition to being the PI of the group, I'm sort of the chief tech support person. So as many of you have done, you can always email me at Boyden@mit.edu if you have any any things you want to talk about. And with that, I can take questions.
So, thank you so much. It was, I guess, a bit of an exhaustive and exhausting presentation for the audience, but take a deep breath, take it out, ask questions. Sinem.
Hi, Ed. So I was wondering, with all the different expansion methods and folds out there, of course, most of the time the targets are polymers, like proteins, RNAs, DNAs. And do you have a sense of at what point, you know, when they stretch and unfold, and at what point they would break, actually, like the polymer would break?
Yeah, good question. Well, this idea of controlled fragmentation, like what Ali and Silvio's beautiful paper. There was a paper from a group at Harvard, Jason Buenrostro's group, where they took the genome and used TN5 transposase to try to chop it into pieces and expand it, and they tried to do in situ sequencing, but of DNA. They published this a couple of months back, I guess. So I love this idea of controlled fragmentation, and it's fun to see different groups trying this out in different contexts. Will work for all molecule types. I mean, there are molecule types like carbohydrates and so forth where maybe it's more subtle how to do it. But I love the idea of controlled fragmentation followed by some kind of fingerprinting or sequencing as maybe a way to think about this going forward. Although it is very early days, admittedly, but a very tantalizing set of ideas.
But you think there would be like a certain factor of expansion where different like polymers would break, essentially, like or fragmented?
Are you saying that would the polymers work?
Like for RNA polymer, DNA polymer, protein polymer. So like with different expansion factors to fragment them differently based on their elasticity and other factors?
Yeah, good question. It might also depend on the scientific question at hand as well. Like obviously fragment something into two small pieces, you could have loss if you don't have anchors on those pieces. And there's other caveats that are probably pretty obvious as well. Like, can you recognize such a small piece? I think probably that's something that maybe as a field we have to think a bit more about because, is there a sweet spot for how big a fragment should be? The smaller, the more interesting the expansion process might be. But of course, the smaller it becomes, you get more loss and other issues. But maybe new anchors could be invented that would, bind really small fragments of things.
Oh, thanks.
Yeah.
Question in the back.
Thank you very much for this talk. Very inspired by the protein sequencing part of your talk, because I've been thinking about this for a while, and maybe I have one suggestion about how to make it work without in a tissue, which is like if the chemistry exists to target the functional side chains of amino acids, and you have different fluorophores for the different functional groups for these amino acids. When you expand using distance and the color, maybe that's a way to also sequence proteins in a tissue.
Yeah, so actually, I almost thought of putting that slide in, but we are actually actively engaged in this collaborative process with this project that Helena and Ali and Silvio and I are. So that is something that we are actively exploring. Yeah, cool. It's a fascinating idea.
Yeah, love this.
It's going to work soon. Where soon remains undefined. Okay. Do we have other questions? Do we have other urgent questions? Seems not to be the case, which is maybe people are a little shy, but Ed is going to be here also today and tomorrow. Okay, perfect. So he's going to be around, and otherwise we have to move to the next lecture, and I am happy to welcome our next speaker, Paul Guichard. But let's thank, you're right, let's thank the previous speaker once more. Yes, so now as we are preparing the laptop and everything, I'm happy to welcome our next speaker, Paul Guichard from the University of Geneva in Switzerland. Paul did his initial work and got his doctoral degree in France at the Institut Curie. Then he moved as a postdoc to EPFL in Zurich. EPFL in Lausanne. L is not from Zurich. So most of the time. So at EPFL, and then fairly recently he obtained his own laboratory, first as an assistant professor, now as a full professor with tenure since about 4 years. And he has been highly decorated. He has a huge following especially in the direction of using cryofixation and other methods in relation to expansion microscopy. And I'm happy to see that the community has rewarded this really important direction. He obtained the ERC starting grant, a very selective grant in the European environment. Then immediately afterwards, the consolidator grant. So, this is really work that has really made some waves. So thank you for coming, and I look forward to your lecture.
Thank you very much. And thank you, Ali and Silvio, for the organization of that meeting. It's really a pleasure to interact with everyone doing expansion microscopy. So I think we saw already, the last 2 days, and from all work we are all doing, expansion is, no, it's disconnected. Here we go, it's there. That, yeah, so we are all, doing, imaging, and we had like this beautiful demonstration that we can now reveal, thanks to expansion microscopy, new features that were not accessible before. But even we think that seeing is believing, we are all microscopists. Of course, we should think, can we really believe what we see? So everything is beautiful, but we are not talking about artifacts. And I think this is something we should address. I'm not going to address today of potential artifacts of different protocols. I'm going to talk about the fixation issue. So I come from the electron microscopy field, and this fixation issue has been there for a very long time. And this is already from this textbook from Edouard Kallenberger, so it was the scientific father of Jacques Dubochet, where already was talking about this issue of fixation. So in electron microscopy, way before expansion, of course, we could see the molecular architecture. And they could realize by experiment, using different type of fixation or solvent, that the cell was basically precipitating. So depending if you have fiber or soluble protein, you end up with a structural disorganization. And one of the idea behind, still not very clear how it works, but basically when you have chemical fixative, you start to interact with this hydration shell, where basically protein will lose their nice conformation and start to interact together. Of course, you have those chemicals that also do intermolecular crosslinks. So this needs to be preserved very well. And I guess all of this you know very well. One of a nice example that has been published recently is these phase-separated organelles, where if you add 4% of PFA, very rapidly you start to form non-native structure. So you see that it's full of very nice dots in the nucleus here, but those dots are artifacts, and this is typical artifacts of fixation. So you can expand that, you are going to have like beautiful images, but that's not the reality. And this is where I think we have to be careful when we do now super resolution at that level. And that's not only an issue for expansion, but for all super resolution methods. So as I said, I come from the EM field and even the cryo-EM field. And of course, as I said, this is something we know. EM chemical fixation is great. We could reveal a lot of things. But to go further in terms of native architecture, the best of the best is the cryo-EM and cryotomography. And this is thanks to the invention of Jacques Dubochet that developed cryofixation. So for those that don't know, cryofixation is a method where you can freeze so quickly that then you can form vitreous ice. Because if you put, so it's a way to fix, right? If you want to fix by just freezing, if you are too slow, you are going to form a crystal ice. And those crystal ice will pierce membrane, will destroy the biological sample. Even the snap freezing in the liquid nitrogen is not fast enough. What Jacques Dubochet developed is a way to freeze so quickly in liquid ethane or ethane propane, so quickly that then the water molecule of water can't reorganize. And then with this, you form these vitreous shapes. So this is what you see here in the back of that image. And this is the crystal ice. And thanks to this discovery already more than 40 years ago, now we have access to the native architecture of the cells, not saying that chemical fixation, what we saw before, were wrong, but some molecular details were not accessible, and now we have access thanks to these approaches. Of course, this was developed 40 years ago. We are not doing the cryo-ET as we do today. Before, people were also using this cryofixation for regular EM, so replacing the regular chemical fixation by a cryofixation and then putting in resin to do regular EM. And for that, they developed the freeze substitution process. So it's a process where basically you can freeze in the vitreous condition, then you go in acetone at 100%, so solvent will create precipitation, but not at very, very low temperature. So what they do is that you put in acetone at minus 180 degrees, and slowly you go up to 80 degrees, so acetone at the beginning is solid, will become liquid, and then start to replace the water. And there is a long process where you go slowly, 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 and then you can reach 4 degrees, you can then put back in PBS, and basically the entire cell is kind of fixed, but without chemicals. And with that, after you can put in resin, EPON most of the time for electron microscopy, you do sectioning, and then you can image. And this is also a beautiful, I think, example of what happened when you look at the brain in chemical fixation. I have to say also in electron microscopy, fixation is very, very strong compared to immunofluorescence. This is a 2% glutaldehyde and 2% paraformaldehyde. You see that compared to a fresh section of the brain, there is some structure that collapse. And basically what happens is that you are losing also a lot of interspace between cells. So the water is lost, basically, and you see that the cell architecture is not as well preserved as the freeze substitution. And the same, so this has been quantified by the group of Graham-Nutt at EPFL, where 50% of the brain, for instance, is filled with this extracellular space. So that's why, again, chemical fixation is not totally wrong, but cryofixation could help us to go a bit further in this molecular characterization. And so that's why we decided to try if we can develop cryo, the expansion microscopy. So the idea was to combine cryofixation, this cryofixation that was invented 40 years ago, with expansion. And this was the work of Marine Laporte, that is here in the audience. So the idea was, can we just try this freeze substitution with expansion microscopy? And so this is what we've done. So here, what you see is what we call the plunger. It's a manual plunger where you just take the regular cover slip with your cells on it. And then this is the invention of Jacques Dubochet. If you plunge very quickly in this ethane propane mix, then you are going to form vitreous ice. Then we go for this freeze substitution. There is a very fancy system from Leica, but you can do that under the hood with some dry ice. Overnight, you go to PBS, and then when you are there, you can put your expansion protocol. In our case, we use our protocol UXM, but this most likely can be used with any expansion microscopy approach and can even be used for super resolution. You don't have to go, you can do immunofluorescence just after freeze substitution, for instance. So now if we compare the different type of fixation in UXM, so if so what we wanted to look at is our membranous organelles that are quite fragile, and here is the ER, endoplasmic reticulum. And you can see that methanol and PFA are quite bad. The ER is fully fragmented. And PFA-GA is one of the best fixatives, so we got nice result by expansion microscopy. But GA is very bad for expansion. So GA, glutaldehyde, is a cross-linker, super strong cross-linker between proteins. So cell looks good. Most of the time it looks very good. But I believe that there's a very strong interaction between proteins, and this expansion between proteins will not happen very well with glutaldehyde. So something that we always try to avoid when we do expansion. And now when we combine with cryofixation, so here there's no chemicals, and freeze substitution, now we preserve very well the ER. We don't see any fragmentation. And it's not only for the ER, but we can combine with any organelles or cytoskeleton part of the cell. If we look, for instance, here at the ER combined with the microtubules in blue, you see all feature that has been described before by live supervision microscopy. So you see, for instance, the ER that is pulled by the microtubule. You see ER, interacting with microtubules. So all of this is preserved. Another example of how cryofixation, or how good is cryofixation for fragile structure. When you have a cell in mitosis, you all know microtubule interact with DNA, but often we don't see the microtubule that we call the astral microtubules. Those are extremely sensitive to fixation. When we use cryofixation, we do keep every astral microtubule. We see them even interacting with the cortex. Similarly, for other organelles, such as mitochondria are super well preserved. You can see here in NHSS there, or here stained with TOM20. But something that was also quite interesting when we use cryofixation is not only that we preserve well mitochondria, the same than ER, we have much less fragmentation, but what was surprising that we got higher labeling efficiency. And this is also very classical for cell biologists. When we have our favorite antibody, sometimes we know that our antibody is good with methanol fixation or glycerol fixation, and we are always fighting with that. With cryo, there is no chemical. So we observe, at least in some of our studying we did, that the labeling was higher. And TOM20 worked for every fixation, but TOM20 here at the surface of the mitochondria was way higher in the cryofixation. And probably because we preserve the epitope, or the epitope are more accessible because there is no chemicals around. So cryofixation works, it's very easy to do it on cells when we do this plunging. Classical method we use in cryo-EM. But you can also do it in tissue. We don't have example here of tissue, but when you want to do a tissue, you have to do high pressure freezing. So this freezing process to do vitreous ice is very difficult in very thick tissue. You need high pressure. So for that, you can combine with high pressure freezing. We did it on yeast because yeast are difficult to freeze with plunging, so we use high pressure freezing. And as you can appreciate, the cell architecture is very well preserved, and even the vacuoles that usually are affected by chemical fixation are very nicely stained. What else? We were also surprised that when you use cryofixation, we can now also preserve the membrane. We don't really know, or we don't understand how it works, but if we just do M-cling, we just reveal the full membrane. So it seems that everything is preserved after expansion. We use UXM, so we are post-expansion labeling. We have 95 degrees denaturation. But when we use M-cling, as you can appreciate, membrane seems to be preserved. And the same here, if we zoom, membrane of the mitochondria are visible with M-cling, and we even retain probably the nucleoids here that you can see on those mitochondria. This is an inverted image of it, of the M-cling. You can see that probably we see also the cristae inside, and it's just a 4X expansion. So the cell architecture is very, very well preserved, and probably also the membranes, which, again, I'm not sure, don't understand how it works. Another example of membrane, we work in the lab with a different model system, work with human cells and chlamydomonas, a green algae. And so we try M-cling because Sabrina Absalon tried that in Plasmodium and worked very well. And we try in those small cells because they have those thylakoids, it's a green algae, so there is full of thylakoids. And when we try with body pie, we got a very, very nice staining here as well. So again, cryofixation, we have it a bit with chemical fixation. There is a bit of membrane, but when we cryofix, it seems that everything is preserved, and you can see a cryotomogram from our colleague Ben Engel here that shows similar structure of the thylakoid. So the architecture seems to be well preserved. So cryofixation is, for us, again, it does not replace entirely chemical fixation, but I think it's really a new way to ask some biological question. And one of them we addressed was the T-cell. Because when we developed the cryofixation, we started a collaboration with the MD in the lab, Benita Wolf, that joined our lab because they want to understand better how T-cell attack the cancer cell. They work on CAR T-cells, so the idea is to then improve the immunotherapy. And the architecture, the cellular architecture of those T-cells is not super well understood, or again, as we had presentation before, people mainly use electron microscopy to look at those T-cells. Why cryo is interesting, or cryofixation is interesting for those T-cells, you could see that they are extremely dynamic, and there is a huge membrane and cytoskeleton reorganization. So first, when the T-cell interact with cancer cell, you have actin that goes to form a ring, so it goes outside the cell center. Then you have a polarization of the centrosome here that will bring some lytic granules, and these lytic granules are membranous organelles as well. And when you have this interaction, then you are going to form supramolecular activation cluster, where you have different domains with protein localized different parts. So we wanted to see if cryo expansion microscopy works with those cells. So we use T cells that are activated in surface. So here we don't have the cancer cell, but we can activate them on the surface. And as you can appreciate, we can stain for tubulin and actin, which also sometimes are difficult to have both very well. Here, we could recapitulate very well by expansion those cytoskeleton and the different stages of T-cell activation. But what was really of interest for us was these lytic granules. So these lytic granules are released, so you have this interaction between the T cell and the target cell, the cancer cell, and then you form this immune synapse. And inside this immune synapse that is very thin, only few nanometer or dozens of nanometer, you have, it's known that you have those lytic granules made of porphyrin and granzyme B that will go in this space and attack and kill the cancer cell. And these stages here has been surprisingly well studied by electron microscopy, but only surprisingly only recently, 3 years ago, people discovered there is two types of lytic granules. And why? It's because here they use cryofixation, cryosubstitution, and then they found that there is, they got higher quality, and they find those two types of granule, the multicore granule that you have here on the right, and the single lytic granules. And again, this EM, we don't have the molecular composition. Are they made of perforin and granzyme B? This was not clear. So we addressed that by cryo-EXM, and you can appreciate here, so this is M-cling, so this is the membrane you see in gray. And then when we look at granzyme B and perforin, it works also very well. The staining was very, very strong, and we could detect those vesicles. And what we find using this approach is that we can now also reveal, using expansion microscopy, those two different types of lytic granules. You have the single core where granzyme B and perforin co-localize inside, so they are a bit smaller. And you have larger multi-core structure where here there is no real co-localization. They are both part, granzyme B and perforin, of the same endosome here. And you see that, so it stands for LAMP1, sorry. You see that there are several granules inside a larger domain. And we have even some cytoplasmic granzyme B and perforin. This we don't really understand. It should not be there, otherwise the T cell itself will suffer from it, so probably maybe also with other membranous organization around. So in that model that was made from the EM, they were proposing this release inside the synapse. This has never been also observed very clearly by super resolution. Do we have the LAMP1 going there? Do we have a release of the entire structure? Or do we have the granules going in the immune synapse? So to test that by cryofixation, we now not use only the T cell activation on surface, but we did a co-culture with a cancer cell. So you see the T cell on the right and the cancer cell on the left. And then now what we see using NHS-Esther, so you see the two cells, the T cells here and the target, and here with the contrast is not super high, but we have the synaptic cleft, so where the T cell will interact and kill the cancer cell. And if we zoom in a bit, what we see are those granules. So here's a multicore granule that basically goes very close to the cell surface, and now we can see the event of degranulation, so the moment where we have fusion of LAMP1 and we see granzyme B and perforin going in this synaptic cleft and entering into the cancer cells. You can see it in 3D, where, again, thanks to cryofixation, all structures were preserved, so we could really catch very fragile also events. Here, for instance, you see real degranulation where we see LAMP1 very close to the cell surface with the release. If I still have 1 or 2 minutes? Oof. Okay, so maybe I will try to go fast here. Something that is not linked to cryofixation, but I just wanted to highlight that because I think it's a good place to discuss this new protocol that we just developed. So as you know, we can do iterative, and we do even ourselves this iterative UXM protocol where we have this 16X. Virginie will talk about it also tomorrow. And this was developed by Vincent Louvel that did his PhD in our lab. And before I left, he wanted to push even more the expansion and see can we go higher. And we are a big fan of UXM. When we look at centrioles, so far this is the one we really prefer. So we decided to stick to the UXM as the third gel, but the idea is that can we expand more in only one iterative step, but then take a larger gel, a second gel very big to do in once a 40 to 50x expansion. And to do that, so Vincent decided to use the T-Rex because the T-Rex is supposed to be a 10x, right? But we wanted to push a bit the T-Rex. Especially the T-Rex in our hands was too soft to really push the expansion. So he made like a new recipe where he took the T-Rex recipe and then play with the bis concentration by replacing bis by MPS. So we got now, maybe you don't see here, but a 13x expansion gel that it's quite big and quite rigid. So we did the expansion, was working, but then, as you know also, and something we did not really discuss this here, is the volumetric dilution of the signal. So even we do post-expansion labeling, at some point, if you push, we are going to lose the signal, and it's quite challenging. So Vincent also try what we call the boost. It's quite a simple approach, but it works very well. It's just doing a triple labeling, adding a primary, secondary, and ternary antibody, and with that, we can boost the signal. So when we do iterative, we do intermediate staining, so after the first gel, and then we expand that immunofluorescence. And just to show you that now with a 50x or 40 to 50x expansion, now with only a 10x objective, we can reveal the eightfold symmetry of the nuclear pore. So these are quite obvious. You see here the top view and the side view here. And we also use the nuclear pores rulers. And in that type of experiment, we obtain a 45x. And this is, again, just with a 10x objective. And the beauty with that is that we can do super resolution with this air objective. So it's very fast imaging. And we can cover in a few minutes the entire nucleus and get this high resolution image. We also saw yesterday that phage are the new very good ruler for expansion, so we use them as well. So we did that already some years ago on the UXM, so this is how the phage looks like in UXM. And now when we combine that with the big protocol, we could image phages that were 11 microns long. So we have now, again, this was a 40x expansion experiment. Does it work with NHS? Yes, so we can also stain very well with NHS ester. So again, the signal is very low. So here, Vincent used NHS ester IFTC and then an anti-IFTC. So we stain with an antibody the NHS ester, otherwise the signal will be too low. But now, directly with a 5x objective, we can see the cells very nicely. And so you can appreciate that now. So this, we are early days, so there is still variability in the expansion factor, depending how the first one shrinks. But in that experiment, we could even reach a 50x. So this is where we can go. Why I wanted to show that is also something we wanted to discuss, because the challenge say, okay, Vincent, push as much as possible the expansion factor. At some point, we are going to see every individual protein inside the microtubule. At least in our hands, with a 63x and 50x expansion, where microtubule should be more than 1 microns large, we don't see that. So there's something happening at the molecular level. Everything is nice. We can see every individual microtubule. We can do a lot of super nice super resolution. But still, there is some stuff happening at the molecular level. So now, basically, we are going to continue to work on that, but try to do now super resolution with light sheet microscope and basically get the same resolution we have in a single molecule light microscopy with just a 10x. And with that, I think I already highlighted the people that did most of the work. So Marine Laporte was our cryo-EXM developer. Vincent developed IUXM and the big protocol that I showed today. And Florent Lemaitre is our T-cell expert and our funder, and thank you for your attention.
Thank you. So, very nice talk, and I'm especially enchanted, the last part, of course, is beautiful, and I have not seen it before, but I'm especially enchanted by your M-Cling pictures, because they always show so much detail, and they're so cool. And it leads me to one little story for the young guys here. If you invent something, even if you think it's like, yeah, whatever, it's not very important, patent this thing. Because 10 years ago, M-Cling felt like, yeah, nobody's going to make ever any profit of that, so who cares? And it's being sold now by the bucket for expansion microscopy because you need lots of it. And all of that profit doesn't go to me, So, unfortunately. So, there's a lesson somewhere for all of us. Invent something, patent it. Ed's doing it right. Questions? Who has a question? No urgent question. Oh, there we are. Please.
Hi. Very nice talk. I guess in the field of synaptic physiology, there have been some important experiments done where they have this flash freeze of, so you study these lytic granules, but I study synaptic vesicles, and I'm wondering if anybody has tried to couple this optogenetic stimulation and then immediately immediately do the high pressure freezing too.
Yeah.
So that has been done for with just regular EM, but I think that'd be very powerful if it's coupled together with for the molecular picture with the expansion.
Yeah, exactly. So again, I come from the EM field, and I think that we have a lot of things now we can take from them. And indeed, there is many things that has been done correlative, where you do correlative live imaging or flash, live cells, then freeze, and then now you can just go to a molecular detail by using electron microscopy. There's a lot of work doing that. I think we can go now with expansion, and that's a possibility.
Okay, then thank you everybody again. Let's thank Paul again. And now we have our coffee break until 10:55. And at 10:55 we start again. So Nadia, please organize the chair. Where's Nadia? Ah, there we go.
And while our next speaker is getting ready, let me briefly introduce him. We have as next person Kiryl Piatkevich from Westlake University in China, and he's going to talk to us about Beyond M and Expansion Microscopy.
All right, thank you very much for introduction, and I would like to start with thanking Silvio and Ali for invitation. I think it's a great opportunity for me to present my research, and this is the best EXM conference ever. I like it so far a lot. So, and it's been also very exciting to see how much has been done with expansion microscopy in the past 10 years. So, anywhere from single molecule shape visualization to the very dense tracing of mammalian brain with a single synapse resolution to even whole body imaging of a whole body. And I should confess, and what's even more exciting is that this all been predicted by Ed 10 years ago, and I here should confess that I didn't believe him 10 years ago, and it's very great to see this visionary from Ed. And what was particularly exciting and encouraging for us is that there are two more technologies that allow us to perform untargeted genomics and transcriptomics that also been invented in Ed's lab. And this also demonstrates how EXM chemistry is very versatile and allows many different chemical reactions to happen in the gel. And we were thinking about if there is already untargeted transcriptomics and targeted genomics, can be also extended to untargeted in situ proteomics. And when one of the most established method for performing proteomics and targeted proteomics right now is mass spectrometry. And one of the modification of mass spectrometry is bottom-up proteomics, where the proteins are digested using usually trypsin or lysine enzyme, and then sent to a LC column for separation and subjected into the mass spectrometer for identification of the MS1 and MS2 spectra. So, and this is a very powerful method for untargeted deep proteomic sequencing. And when we start thinking, can we combine it with expansion microscopy, we decided to see how we can extract actually peptides from the gel. And interestingly, in mass spectrometry field, it has been done routinely for more than 30 years when protein gels, 2D gels been run, and then you can cut piece of a gel and again digest it in a tube and send it to mass spectrometer. So since proteins are retained in the expanded gel, we were thinking, can we just add trypsin to the hydrogel, expanded hydrogel, extract the peptides, again digest them, send it to mass spectrometer. And actually when we've done it, in comparison to the regular golden standard methods in mass spectrometry, such as in solution digestion and pressure cycling technology, we obtain pretty much identical mass spectrum. It means that proteome that we extracted from the gel was pretty much unaffected. And what was even more exciting, that peptide yield that we obtained was even higher compared to the PFA fixed samples, compared to in solution and PCT digestion. Another exciting news was that miscleavage rate with trypsin was also lower compared to the golden standard methods, and we think it's happening because of a better access of trypsin to the proteins inside of the expanded gels. We did lose a little bit on the identification of a protein, so we were able to identify about 4,000 proteins in the 5 nanoliter volume of a tissue, and it's actually happening due to slight modification, chemical modification of peptides. But nevertheless, it has very good overlap with, again, golden standards, and there is no bias on extraction of the peptides from different compartments of the cells. And everything was working well, and then we decided to optimize it further, and we tried to see what other gel chemistries and methods can work with extraction of the peptides from mass spectrometry. Here we compare proteomics, MAP, and Magnify. And what's happened, so MAP protocol didn't work really well. It clogged the LC column. If you've done mass spectrometry, you know it's terrible because your machine is out of the line for 2 weeks. And in case of Magnify, we also got low molecular weight contamination, which actually prevented good identification of peptides. ProXM, excitingly, was working very well. However, one of the problems with Acrylac-X in China was less accessible, and we have to pay $1,200 USD for 25 milligrams. And this is encourage us to try to see if there is any other cheaper and efficient anchors. We screened another 3 more anchors, and we found out that actually NSA works as good as Acrylac-X. However, it's almost 1,000 times cheaper. Another problem with ProXM was that it has quite limited expansion factor, only 4, but we wanted to go beyond. And when we started with work 5 years ago, there were no other gels with a higher expansion factor. So what we had to do, we had to slightly reinvent the chemistry of hydrogels. We screened more than 200 different monomer solutions for optimization, both expansion factor and mechanical stability of a gel. We also had to give up on sodium acrylate. It was, again, very hard to get a very consistent purity of a sodium acrylate with a good quality that doesn't contaminate our mass spec. So we switched to sodium metacrylate, which is way more, it's also cheaper, cleaner, and way easier to use. And as the anchor, or as a crosslinker, we use this kind of new PAA molecule that actually provided us a little bit better in terms of mechanical stability of a gel. We had to optimize a little bit of a chemistry more, and again, expanded samples supported all sorts of chemistry. We also perform alkylation and reduction inside of a gel. This is in order to improve the proteomic profiling of cysteine-containing peptides. So this is how the final workflow works. So we take tissue, we expand it, and homogenization should be SDS, as you can guess. Pro-K will not work because it will chop the proteins into small pieces. So this is a expanded brain slice in scale that's been stained with Kumasi blue. So I started this work with mass spectrometers, and we really hated to see transparent samples in the lab. So we start using same dyes that we use for paint gels. And taking the pictures with a phone and then feeding it to a deep slice can very nicely annotate it. And since the brain sample is so big, you can just take a biopsy punch and selectively select area of a brain, get this piece of gel out. So we usually use 1 millimeter punch, which gives us 140 micron of lateral resolution, get the piece of gel, extract peptides, and just send it to mass spectrometry facility. This is a black box for me, because we at Westlake University, we have one of the best mass spec facility in China. All we do, we just send our peptides. A few days later, we get the analyzed protein tables. So in these protein tables, we have protein and intensity. And I don't have any bioinformatics background or coding experience. So I started this project in the right way, because when we started this project, there were a few AI agents. Once I received these files, I just feed it to AI agents, and I interacted with them as I would interact with a regular bioinformaticians. Here's the prompt. I ask Biomni plot protein abundance curves and mark the most prominent neuronal markers. This is what it get for me, and it's actually validated. And as you can imagine, all this process now can be easily automated. So after gel expansion, it can be all dissected into small voxels of your choice of a volume. Then there is a robotic pipeline to extract peptides, send it to mass spectrometry, and analyze and interpret data using AI scientists. So here's an example of the cancer tissue that's been processed, about 700 spots. Each spots contain up to 4,000 proteins, and as you can guess, there are 4,000 more pictures of this slice for different proteins. I don't think you want to see it here. However, these experiments are quite laborious, and you don't need often to get entire sample dissected and analyzed. In some cases, you may care just about small structures. And since this conference second theme is kidney, I decided to put this slide where we expanded kidney. And here we use CyPro fluorescent staining. So NHS also work, but they do reduce MS identification due to modification of lysines. So after imaging in a super resolution, we punch single glomeruli, send it to mass spectrometry, and now we can look at the proteome of single glomeruli and compare it and get the also functional categories for each protein and see what are the proteome composition. So probably perhaps it's the first time when we can correlate super resolution imaging of some tissue functional units with a complete proteome. In my lab, we use AD model a lot, and I've been very curious about one question: why sometimes humans can have amyloid plaque in the brain, however, at the same time, we may not have any symptoms. And using proteomics, we decided to see the proteome of two types of a plaque, which usually called as cored plaque because we have this densely compact core compared to diffuse plaque. So after dissection and mass spectrometry, we see that they have actually quite different proteome. So as you can see again, the mass spectrometry is a quantitative method that gives you relative concentration of a protein in your sample with a dynamic range more than 6 orders of magnitude, which is way higher than microscopy. Usually our cameras working in 16-bit. And in order to make it statistically significant, we had to analyze a little bit more plaques. So in this case, for this study, we use about 30 plaques per slice from 2 different ages of animal in early stage and late stage of AD. And we were able to identify the differentially expressed proteins responsible for different types of plaque. And again, in cored plaque, we identify this kind of protein interactome that defines its cytotoxicity, as well as perform pathway enrichment analysis so we can tell what kind of cellular and signaling pathways actually dysregulated. And so we also feed these results, mass spectrometry results, into the AI scientists, and it has come with some of the findings. Very interestingly, some of them been actually already confirmed that it's been known that cored plaques are more cytotoxic than diffuse plaque, and this cytotoxicity coming due to the calcium hyperexcitability of the neurons. There are more findings that we have here, and we're trying to validate it further. However, in mass spectrometry field, in spatial proteomics right now, the bar is single cell proteomics. And in the past 2 years, Thermo released an Astra machine that can actually perform single cell proteomics. You can think about Astra as a mean flux in a super resolution field. It's also as expensive and as difficult to use. And we decided to see if we can use expansion microscopy for single cell proteomics. And actually we decided to go beyond. We decided to try to do even subcellular proteomics. Here we, in order to dissect this nucleus that's stained with DAPI, we use laser capture microdissection. We cut single nuclei, perform mass spectrometry, and compare the proteome in comparison to a single cell. And while it's interesting to figure out that nucleoplasm proteins occur to be in nucleus, we decided to think about more biologically relevant applications, as well as another problem here. You can imagine this experiment is quite laborious, and if you want to sample entire brain slice, you may not have enough resources. So what we decided to do next, we decided to switch to mass spectrometry imaging, since we learned a lot about from LCM SMS methods. And in MALDI mass spectrometry, the principle is a little bit different. The sample is placed on a slide, and a high power laser shines on it and let the molecule evaporate, fragment, and then we send it to mass spectrometry. You can think of MALDI as a point scanner confocal microscope, but instead of exciting fluorescence, you actually evaporate the molecules out of it that you send to mass spectrometry. The problem with a MALDI, though, but it's not a method for proteomics. It's usually used for lipidomics or metabolomics analysis, because it's very hard to get proteins fly away. And we decided, what if we just expand the sample and put it process with a regular MALDI preparation method and put it in a MALDI machine. And to our great surprise, when we've done so, we increased the number of identified proteins per pixel to 640, which was 300 times more compared to a regular MALDI. And with this powerful technology, I decided to ask a very simple question that bothered me for a long time. I wanted to understand what is the first protein that can be that is dysregulated in a cascade of molecular events that's happening before AD starts. For this, we scan mouse brain tissue at different ages, and we found out that we can start to see dysregulated proteins as early as 2 months old. And one of the most prominent dysregulated protein that occur both in hippocampus, corpus callosum, and the cortex was acetyl-CoA, acetyltransferase A. And this protein, this enzyme is responsible for DHA synthesis. And to our big surprise, when we start searching its relevance to AD, it figured out that 3 years ago, it was a family in China that actually had this mutation in this gene that leads to early onset AD. So I'm taking DHA every day now. So and I believe that at the moment, expansion microscopy can provide a very valuable platform for true spatial multi-omics technologies combined with nanoscopic structural imaging, where we can perform spatial untargeted transcriptomics, genomics, and proteomics, and even lipidomics. I didn't have time to talk about it, but lipidomics is also possible on a single sample. Just one more minute, 30 seconds. Although I'm shifting away from protein engineering, fluorescent proteins still have a special place in my heart. We found out that epoxy anchors actually preserve the fluorescence of proteins very well in expanded samples. Here are examples of this M-Baugene, Msrseille imaging in different samples without any antibody staining. I know it's not the kind of major dyes for expansion microscopy, but I would invite you to give it a try. If you want to get the plasmids for free, you can scan this QR code and order it from Gene. And with this, I would like to thank all my collaborators.
Thank you very much, Kiryl, for a nice talk. We would have time for one short question, if there is any. Okay, I don't see any urgent questions, so let's thank our speaker one more time. And the next speaker is from the Göttingen University, Alexander Egner. He is the leader, the director of the laser laboratory at the MPI, and he's going to talk about pushing the limits, innovation in supra-resolution fluorescence microscopy. Wonderful.
Okay,
Stage is
so
yours.
it works. So thanks a lot. And I also want to thank Silvio and Ali for inviting me and having me here. Also, I will not show any expansion data to date, but because in my department we are working on the instrumental side and in the preparation side. But I really enjoyed the last few days and really saw very interesting and excellent results. But just to show you, so we are concentrating on generating images. And as we all know, there's more than resolution. There are many things we have to consider when we record an image, right? So there's also stuff like the light dose, the speed, signal-to-noise ratio, automatization, as we saw during the time. And then, of course, we can also split it up a bit, that we, of course, have diffraction-limited microscopy, which many of you are now using because you have this fantastic expansion shields, but there's also on top of it the super resolution microscopy. And I just want to share two findings we were working on. And the first one is also going away from fluorescence, but going to Raman microscopy. And we also had this excellent talk by Wei Min on the first day, where he already explained a bit how Raman microscopy works. So actually it's a two-photon process working with virtual states, and what you actually do, you shine in two lasers into your sample, which have different wavelength, and if the frequency difference between the two lasers matches the vibrational of a molecular bond, you get a resonance, and by doing this you can transfer photon from the pump laser to the Stokes laser. And this is very specific to molecular bonds. So this is the fingerprint regions where you can see that you can identify many different molecules in the sample. And the nice thing is that it's label-free. So you can do SRS imaging on unlabeled samples. That's, for example, CH2 band, which is mainly showing lipids in HeLa cells. But of course, you can also combine it with fluorescence, and you can do correlative imaging. So you have chemical imaging and fluorescent imaging on top of it. One drawback of SRS microscopy is, as we also learned 2 days ago, that actually it's a very weak process. So cross-section is the order of 10 to the power of minus 50 Goebbert-Meyer, which we know is a typical 2-photon cross-section. And this means that it's difficult to measure in this fingerprint region, and what you usually do if you want to boost your signal, it's a straightforward way, right? That you just increase your pixel dwell time. So record longer on a single pixel and you collect more data. But actually there's more what you can do, and that's just some result from a project we had together with laser manufacturer APE, because the way how you record SRS data is that you have your pump beam, you have your Stokes beam, you transfer photons. So actually you have to measure again the very high background of the laser. So what you do, you modulate one of those lasers, and then you just look for this modulation in the other laser beam, which hasn't been modulated. And because you are measuring against the background, what you usually do, you are doing lock-in detection so that you move away the signal from the low frequencies where you have substantially reduced the electronic noise in your system. But now comes a trick. So if you switch to subharmonic modulation, which means that your modulation frequency is half of the repetition rate, you move together in the lock-in amplifier 2 sidebands you have into your signal, which boosts your signal at the end by a factor of 2. Then, because in the laser, we also halve the repetition rate, which means at the end, if we calculate it with respect to the power of the electronic signal, we should get a signal increase by a factor of 8. And that's actually a measurement of the signal to noise ratio as a function of the pixel dwell time. And you see the linear behavior as you would expect. And now, if you do the subharmonic oscillation, you really get an increase. And what we saw in the experiment, because the pulses in the new laser are also a bit shorter, that overall for the same average power, we get an increase in the signal to noise ratio by a factor of 10. So this allows us to measure 10 times faster. And just to show you, that's again image of onion epidermis. And then if we just switch to the other laser, then for 20 microseconds factor same pixel dwell time, we get substantially better signal to noise ratio, but this means we can also switch over now to 10 times slower pixel dwell time and get comparable images, which allows us now to do faster imaging. Okay, and now just to share some results. So of course, so you can, we just recorded few samples. So this is onion cells again, where we see the CH2 band, cell walls, proteins, lipids. You can also go to oyster shells, where you can look at the distribution of calcite, or, for example, in yeast, where we tuned the laser to the fingerprint region and we just looked at the M81 band. And last slide about this topic, then as I showed you, you can also combine it with fluorescence imaging. And then actually, so that's first preliminary results where we just tried out to do combined SRS and min-flux imaging. So that's on the same cell. First, we recorded SRS image on a larger scale, and we just tuned it that we just see the nuclei, for for example, and then we concentrated to this ROI, and on the same ROI, we then just performed wind flux imaging, where we also saw the nuclear power complexes. But this is work in progress. Another topic, which might not so important if you look to expanded shales, because they are quite homogeneous in terms of refractive index, as I learned. But if you are looking in a more natural environment, or you want to record within living specimens, of course you always fight with aberrations. And aberrations are caused by non-uniform distribution in refractive indices, and this means that your image gets distorted. And that's how it would look in a normal microscope, so undistorted image. When we focus deeper within the sample, we get less resolution, dimmer images, and then usually what you can use adaptive optics to correct for the aberrations which are introduced by the sample, and that at the point where you image, you get an unperturbed wavefront. So of course, there are techniques around, and people try to measure aberrations in fluorescence microscopy, which actually is not so easy, because in astronomy, you're, for example, using guided stars. You have point emitting sources. In microscopy, you have to do tricks, and what people usually do, they take metrics like image brightness or image sharpness, and what they do, they change their wavefront and then scan images and just look, does the image get brighter, does the image get sharper, and so on and so on. But of course, that's not a nice thing you want to do, because we are fighting for fluorescent photons, right? And if you have to record many images in a row before you can get the best aberration correction, your sample already might have bleached. And that's where we sneak in, because we thought that we can also use photophysics in order to check for aberrations. And that's just recorded in a STAT microscope. And of course, if we apply the STAT laser beam, we deplete the photons, and what we actually see, we change the measured apparent fluorescent lifetime of the molecules. And this we just can detect in the photon stream we get from the microscope. And by doing this, we can alter through the aberrations already while scanning. And just to show you that this works, so this is now just early divided by late photon. So the metric has a maximum. And at this maximum, we also have maximum brightest and the best resolution. And that's not only works for primary spherical aberrations, but actually it works for the whole set of spherical aberrations where you can test through. And this is now just an experiment on single beads layer, where you see we just recorded the image. Then here, during the first few lines, we scanned, we went through the aberrations, corrected the aberrations, and then we get perfect imaging while the rest of the stack is recorded. And just last slide now, this is in Drosophila gut sample, where we also are in uncorrected way. And here we corrected for 5 different types of aberrations. And here you see the line. So here the microscope did aberration correction. And then for the rest of the frame, we get unperturbed image quality. Okay, so I'm already at the end of the time. So this just brings me to the last slide. So just want to thank a few of the people from my lab, especially Lenny, Mark, and René, who were contributing to the SIS measurements, and Deborah Tritter, who was working on the aberration corrections, and also with the cooperation partners and the funding agency. And thank you for your attention.
Thank you, Alexander. Is there an urgent question from the audience? Right, then please find Alexander in a lunch break. Thank you. Our next speaker is already getting ready, and while he's doing so, I'm going to introduce him. We have Christophe Leterrier from the CNRS in Marseille University, France, with us. He's going to talk about the axonal cytoskeleton down the nanoscale.
Okay. Is that okay? Yeah, you can just launch it.
Wonderful. Welcome, Christophe.
Yeah. All right. I am Christophe Leterrier from Marseille. I'll try to be short and sweet, like Sabrina Carpenter. And I will tell you about the axonal cytoskeleton down to the nanoscale. And we are using a range of super resolution and advanced microscopy techniques. And maybe there will be a bit of AXM at the end, if I have enough time. So, neurons. Here's a neuron. And the defining feature of a neuron is the functional asymmetry. So it gets a signal from upstream neurons, inputs on the cell body and dendrites, and then you have the axon that propagates the action potential to downstream cells. So this functional asymmetry is reflected and supported by a cellular compartmentation, polarization between two distinct compartments, and there are also more compartments inside the axon, like the axon initial segment, the axon shaft, the presynapses, and the growth cones in developing neurons. What we do in my lab is to try to understand how the neuron organizes these compartments and how it builds them, maintains them, and transforms them. And of course, the cytoskeleton and unique structures that are unique to neurons are what builds this architecture. And to see the details of the cytoskeleton, we use super resolution microscopy. So now you know why we do what we do. And what are we looking at more specifically? So for the first almost 10 years of my lab, we've been looking a lot at the axonal actin-spectrin periodic scaffold, so membrane-associated periodic scaffold, I would call it MPS for the rest of the talk, is a very peculiar structure that has been discovered by Xiaowei Zhuang at Harvard in 2013, and it's made of actin rings that are connected every 190 nanometers by single tetramers of spectrins. So you have this kind of repetitive structure where you have actin rings and one layer of single tetramer, because each tetramer is 190 nanometers long, is connecting it to the next one. And I mean, for me, this is the most important structure that has been discovered by super resolution microscopy, because this is maybe the only purely, absolutely new structure. It's just not adding details to something we already knew. Nobody had any idea that this structure existed before super resolution discovered it. And for me, it's like discovering the Golgi apparatus or the mitochondria. So I don't know a lot of other examples of completely new structures that have been discovered by super resolution microscopy. Anyway, we have been working on this, and here you see something that I want just to stretch. That is that in the initial segment of the axon, which is the initial part here, you have the tetramer of spectrin that contain alpha-2 spectrin and beta-4 spectrin, and they are here in yellow. And in the distal axon, the beta-4 spectrin is replaced by beta-2 spectrin. So if you want to see the beginning of the axon, you mostly stay with beta-4. If you want to see the distal axon, you will stay with beta-2 spectrin. And what you see here are two color images by combined storm and paint. So the actin in gray is made by storm, and the spectrin is made by paint, and you see the alternation of the actin rings and the center of the spectrin tetramers. We can do better than that. We can do 3D, we can do the deep learning based algorithm to process, and so we can do this kind of nice things where we are riding along axons on an image in 3D, and you see that each of these rings here and the lumen of each of these axons is clearly resolvable, even if none of them is larger than 500 nanometers. So this is very nice, and we have learned a lot about the organization of this compartment. But still, even with this kind of imaging, we cannot see the ultrastructural details, like what are the actin filaments involved, how are the spectrin tetramer organized, and things like that. And to do that, we turned to electron microscopy, and I started to work more than 10 years ago with this guy, Stefan Vassilopoulos in Paris, who is one of the few specialists of this technique that is called platinum from replica electron microscopy. So you've seen unroofing from Helge Ewer's talk 2 days ago, and here we are also unroofing cells with a different technique. We use a sonicator, ultrasound, and so we remove the top part of the cell, and then this is fixed and transferred to an EM grid after replication. What we image is actually the metal replica of this unroofed cell on an EM grid, and you can see the submembrane cytoskeleton with a lot of details like these clathrin plaques or these actin filaments. And we said, okay, this scaffold is called membrane-associated periodic scaffold, so it has to be associated with membrane. So PREM is the best technique to see it if someone wants to see it with electron microscopy, because nobody had at the time. So we tried to unroof neurons. We were lucky that it was possible. We can unroof hippocampal neurons in culture, and then looking at them with electron microscopy, you see the cell body here, and the axon here is in yellow. And what you see here is this part, the very beginning of the axon. You see the microtubules that stay attached to the membrane, which have this very bundled appearance, and the mesh that you see here is the submembrane scaffold. And if you look here, you will see that you have these very peculiar vertical filaments that are regularly spaced, and the distance between each of these filaments is about 200 nanometers. And these are actually actin filaments, and there are even two actin filaments kind of braided together, like you can see them splitting in a Y shape here. So we said, okay, maybe these are the actin rings because they seem to be made of actin and they seem to be distant from 200 nanometers and connected by this mesh that could be the spectrin. So we worked a lot to actually show that this was the case and that this is the first visualization of the MPS by electron microscopy. And I would say the most important or demonstrative experiment was to do correlative STORM and PREM. So we were able to image the actin rings at the beginning of this axon here using STORM of phalloidine, of fluorescent phalloidine, and ship the sample to Paris to Stéphane, who will do the platinum replica EM of the same exact axon. The beginning of the axon here, you see it's split open with the microtubules in the middle, and within the dense mesh here, you see some of the filaments vertical. You don't see them all along because they go in and out of the spectrin mesh, but when you have the superposition, it's very clear that what you see here are the bands, the rings of actin that go across the axon. So there's no doubt that the braids that we see in EM are the rings that are seen by STORM. So that was all good, and we are very happy to see these actin rings and the spectrin connecting them. But Stéphane is working historically on endocytosis and clathrin-mediated endocytosis, and he kept bugging me like, but look, there is something here, there is a clathrin-coated pit. We should look at that because nobody has described endocytosis along the axon shaft and not at pre-synapses. And so after we published the actin ring stuff, we said, okay, let's look at the clathrin and the endocytosis. And this was the work of Florian Werner, a PhD in my lab, and Satish, who was a postdoc in Stéphane's lab. So we started labeling the clathrin along the AIS, and you see here now we have SIM, and SIM can resolve the periodicity of the spectrin and the clathrin-coated pit, and each of these clathrin-coated pit falls inside a little hole of the spectrin mesh. And this is clear from intact neurons, and if you unroof them, it's even clearer. You never have colocalization. The clathrin pits are inside holes of the spectrin. And of course, if you do PREM, you see it with much more details. You see here you have a region where you have the actin rings, and here you have clathrin-coated pits that are on bare membrane areas. And if you look at a lot of these, you see that these bare membrane areas are very stereotypic. They are like circle of about 300 nanometers in diameter, where you can find all steps of clathrin-mediated endocytosis, from a few clathrin trimers, yes, to triscales, to fully assembled clathrin-coated pits, or even several of them. I would like to show you that here you see there's always, or very often, a little actin filament connecting the clathrin-coated pit to the mesh around it. This is just to show the average size, 300 nanometer, compared to the clathrin-coated pits, which are about 80 nanometers. And of course, if we do our correlative technique, we can see what is this clathrin area made of, and this is mostly actin that makes this area. And so this is a new structure that we call clearings, because we say, okay, it's like in the forest, and you go and you have the house of the witch in the middle of a clearing. So this made me thought about this, and we call them spectrin clearings. So new structure that would explain how you can make a clathrin-coated pit on a membrane that is covered with a very dense mesh of spectrin and actin, and you can get access to it. So it's okay, that must be the place where endocytosis occurs. But is there really endocytosis from this structure? Because when we did dextran feeding, so we use dextran and see how it goes inside the neuron, we were very surprised because we could see a lot of dextran spots, and we say, okay, there is endocytosis at the AIS, but all these dextran spots, I mean, most of them were colocalized with clathrin pits after half an hour, and on unroofed neuron, it was all of them. All of them were colocalized with clathrin-coated pits. And you can see here the averaging of the spectrin hole, the clathrin pit, and the dextran cluster. And that means that the dextran is not getting inside, because half an hour it would be far away from the from where the pit was. That means that it's stuck on the membrane, accumulating in the pit, and that means that the pits are super stable. Half an hour is very stable. A clathrin-coated pit lasts for about 5 minutes or even less. And if we do correlative SIM and PREM, we see that we can see this accumulation of dextran inside structure that are unambiguously clathrin-coated pits. So that means they are super stable and they don't do endocytosis, and the last way to check it was to develop live cell imaging with SIM, so we could see the spectrin and the clathrin together, and we see the periodicity of the spectrin very well, and we see every clathrin-coated pit with a good resolution. And what I want you to see in this image is that here along the initial segment, those clathrin clusters are very stable, whereas along the dendrite, and you have the zoom here, or the cell body, they are blinking a lot more, so they are far less stable than in the AIS. So this spectrin and the fact that they are in clearing seems to really make them a lot more stable. And you see here the chemographs, like the vertical bars are basically stable, stable clathrin-coated pits for the 20 minutes of the movie. So I won't go into all the rest of this study, but basically what we found was that this is triggered regulated endocytosis. The clathrin pits are there, they are waiting for something, and they don't endocytose unless they have the right signal. And the end of the endocytosis, for example, if you treat with NMDA, is actin polymerization inside the clearings. So if you have stimulation, you have a triggered endocytosis that makes actin polymerizing and finish the job for the scission of the clathrin-coated pit, and it's only happened with specific signaling, which is of course very important in neurons. So you are not endocytosing things at this very important compartment that is the initial segment all the time, but what you need to remove some sodium channels to, for example, regulate the excitability. The clathrin pits are there, you don't need to push everything, and it would be very difficult. It's there, it's ready, and it can endocytose with the right signal. And this was published last year, so you can read all about it. And that leads me to the next thing I want to show you, because basically the conclusion is that the MPS here is an insulating layer for the axon shaft. So you can only have access to the membrane and do endocytosis if you make a hole in the MPS. And we said, okay, if this is the case for endocytosis, it should be the case for exocytosis. So what happens for exocytosis? Is there exocytosis along the axon shaft? And is it regulated? And is it happening at specific places, or like holes that would be for exocytosis, like we found holes for endocytosis? And one of the things that tells us that we're right in this hypothesis is that the synapse, where you have a lot of exo and endocytosis, of course, is the place where you don't have an MPS along the axon. The MPS stops right before, and then if it's an en-passant synapse, continues after, but there is no MPS so that the membrane is accessible. And this was the work, this is the work of Teresa Wissner, a postdoc in the lab, and she used live cell imaging because, of course, it's easy to see endocytosis with the clathrin-coated pit and the feeding. You can see it in fixed cell. Exocytosis, you need living cells, and you need to do live cell imaging to see it. So she was using VAMP2 fluorine to look at exocytosis of vesicles along the axon. We use VAMP2 because it's the most general marker of exocytosis we could find in neurons. And she had labels where we see where is the presynapse and where you don't have synapse. And she could see exocytic events, both at presynapse, of course, and along the axon shaft. This is spontaneous, non-stimulated. We don't want to bias the system toward exocytosis at synapse. We want to see, are there non-synaptic exocytosis events? And the answer is there are. And I think in most papers, people show you these kind of very small figures where they center on the exocytic event, and you never know exactly are the neurons healthy, what they look. So I wanted to have a full neuron image. So here you see what these experiments look like, and you see all these different compartments. And in the same movie, you see exocytosis on the dendrites, on the cell body, at the AIS, in the proximal axon, and in the distal axon. And this is really a striking realization from Teresa. And what I want to say is that when we started counting the spontaneous exocytic event, we were very surprised to see that the AIS, among all compartments, has 2 times more exocytosis than the other compartments. So that's a very interesting thing. We don't know why and what is exocytosed, but if we apart from synapses, presynapses, of course, the place where you have the most spontaneous exocytosis is the AIS. And now that we can do this, and we also have all these nanoscopy experiments, how can we try to zoom on the nanoscale environment of these exocytic events? And here we have correlative live cell imaging and 2-color storm. And my movie has just played once, so I'll try to launch it again. Yes. So what you see here is a proximal axon, and the movie of exocytosis with 2 events up here and here have been superimposed with the organization of the spectrin in green and the clathrin-coated pits in orange. And the yellow you see is the detection of the periodicity with an algorithm called SRED that we help Ricardo Enriquez develop. And now you can see that the exocytic events also happen in holes of the spectrin mesh. That is very clear. And the second thing we know now is that these are holes that don't have any clathrin-coated pits in them. So indeed, the MPS seems to really be shaping the axon with different areas, some for exocytosis and some for endocytosis. And now the end, I saw you just got up. I will show some AXM for the people who like it. So different projects about microtubules, because for all of this, we don't really need the XM. Storm is really good, but for microtubules, we need the XM. So what we do in the lab, so yeah, check that, bioarchive last week, and now microtubules. And the EXM we do is to look at microtubule repair, microtubule in lattice turnover in cells, which was discovered by Manuel Leterrier and Laurent Blanchoin, or at least rediscovered, because there's always someone who has seen it in the 1950s. So this phenomenon is like everyone knows that the microtubule grow and shrink from the plus tips, but they have evidence that you have a turnover in the lattice of the microtubule. And we helped them look at it in cells. So what they were doing at the time was microinject fluorescent tubulin and see incorporations away from the plus tips. And we helped them with STORM, really looking at it and showing that they are really inside the lattice and not some tubulin that would go on the side of the microtubules. And once we are doing that, we say, okay, but there is one type of cell where microtubule repair would make a lot of sense. It's neurons, because you wouldn't need to completely depolymerize an architecture and repolymerize it to turn it over. So we started to say, okay, maybe we could try to microinject neurons and see where is the turnover in neurons from this technique. And this was the work of Karen Butler-Hallisey in the team. So he was able to microinject hippocampal neurons in culture, which is incredibly difficult because basically these cells are just a big nucleus with membrane around it, and that's why nobody had done it so far. So he was able to do it, and we could see after that we could fix and label the total tubulin in green and the injected tubulin in red, and starting to look at where is the microtubule integration in the lattice after, and this is 3 minutes after injection. So very fast after you just fix and see where your fluorescent tubulin has been integrated. But still, we are not resolving single microtubule on these spinning disk images. So we said, okay, we have to have a better resolution to really know, is it a plus tip, is it an integration in the lattice? So of course we said, let's do STORM. The problem with STORM of neurons, and this, I like this image that was done by Karen, is that you have a neuron here with these 2 dendrites and this axon and a glial cell. And you see the individual microtubule in the glial cell, and you don't see anything in the neuron because the microtubules are so close to one another that you cannot really resolve them, and you label them as a big block. So you have a zoom here. This is from the glia, this is from the axon. You immediately see the problem that we can see individual microtubule in the glial cell, but not in the the neurites. And then Kiryl said, I think maybe expansion microscopy might be better to do that. So there's this nice course at EMBL. So he went there and learned UEXM with Paul and Virginie. And he started doing EXM on these neurons, and it works better. This is an EXM image of a very young neuron, one day in culture, with UEXM and post-expansion labeling. And you can see that now we can really resolve a lot of these microtubules, not always single ones, but at least there are a lot of places where we are pretty sure they are isolated. And this is a rendering of this cell. So this is very dense, but still there are really a lot of places where we can be sure. And so the next step, of course, is to do that after the injection. So it's a very complicated experiment. You inject one cell, and then you have to expand the gel, and then you have to find that one cell and image it. And fortunately, we can do it. You see here an expanding neuron, and I want to see the expanding neurite here. And this is this region of the neurite. And now you see with the expansion, we can really see every microtubule and every event of integration of microtubule in the lattice and also in XZ. And what you see here is that most of the integration happens at the periphery, the ones that are just under the plasma membrane. So, of course, there is this Lucas Capitine idea that the ones that are at near to the membrane are dynamic and the one in the middle are stable. So we are, but now we know exactly, because this is really stability. This is not PTM. This is really stable versus renewing. And this is the end. I'm sorry, I'm done. This is the team. Everyone, Florian, who has worked on the cytosis project, Kiran on the microtubule, Teresa on the exocytosis. These are our collaborators, partners, and founders. And I want to have a word for my poor NIH grant that disappeared, and for my colleagues in USA that have a very hard time right now. And I thank you all for your attention.
Thank you, Christophe, for this firework. I see there's a question coming up. Yes, please.
So I'm curious, do you know if those exotosis events are releasing neurotransmitter or other materials? And secondly, you see that very sort of active exotosis in the AIS. Do you think that you see also the AIS plasticity in expansion or shrinkage?
So the answer to the exocytosis cargo, the answer is we don't know, and we don't know by design, because exactly like for endocytosis, we choose dextran because it's the less selective thing, because we wanted to see all possible endocytosis. We chose VAM2 because it's the one that is labeling the most exocytic structures. So we don't even know if they could be synaptic vesicle precursor that are traveling along the axon. That would be interesting, because that would mean that this insulating layer ensures that they are not spuriously fusing before reaching the synapse. There could be dense core vesicles that have been described to exocytose along the shaft. There could be extracellular vesicles, and in the IS it would make sense to have some extracellular vesicle, so we are quite excited also by that. But my answer is basically that will be the next step to really try to understand what it is. And for your AIS plasticity question, these experiments are about, so they are 5 minutes or 10 minutes movies, so there is the AIS plasticity is much lower than that, and also we are in completely unstimulated spontaneous situation, so we don't stimulate or have any treatment that would cause the AIS to shift. It might be involved in the shifting, in the plasticity, in those contexts, but right now this is really a basal mechanism.
Hi, very nice talk and very beautiful movies. The structure of the actin spectrum kind of is reminiscent of the muscle sarcomere organization, if anybody recognizes. But my question is if you've investigated with the live cell imaging the axon migration dynamics, and how the structure of it plays out during that.
This is a very good question. Actually, we are doing that right now. And so this is not a XM, of course, because we are looking at live cells, but we are really interested because there is so much work on the MPS that has been done in fixed cells and trying to do it in living cells and looking at the long term, how the MPS is formed, how it's transformed, what happens when you make a branch, what happens when you make a synapse, and what happens when the axon has to grow. And this is exactly what we're doing now with SIM. So we have developed ways to label endogenous proteins and to make long-term movies with SIM, and so stay tuned because we will have answers for that, but not now.
Okay, one last question there, and then I'll have a comment, and then we have to move on.
Okay,
please.
thank you. About the indolent exocytosis events, have you looked at the surrounding architecture, whether or not you have synaptic architecture? That is one. The other, do you see the same mechanisms also in tissue context? I assume these are all cultured cells, right?
What was your first question? Sorry?
The first question was whether or not you have seen synaptic architecture nearby where these events take place.
We are, so far at the IS, we know there is no pre-synapse nearby because the IS only has post-synapse. It's actually a post-synaptic compartment in the axon. So we have synapses that are other neurons that connect to the IS. And in the distal axon, we have done most of our experiment by co-transfecting a presynaptic marker and making sure to separate to be able to tell this is not a synaptic event, this is a synaptic event. Now, whereas we have synapses from other neurons nearby, I don't know because we don't see them in the transfection. And for your other question, we don't know what happens in culture, and of course all this is done in cultured cell, in cultured hippocampal neurons, and of course most of these techniques are impossible in tissue culture, but that's also why I'm interested in XM, because this is also a technique that is more amenable to 3D samples than what we do, like the storm and these kind of things.
Do you know the identity of the cell, like what type of cells these are?
Yeah, these are pyramidal excitatory neurons from the hippocampus. I mean, based on the morphology, we have two types in our culture, either this or GABAergic neurons, but it's only 10% of cells and they have a very different morphology, so we usually don't image them.
Thanks.
Thank you. Thank you, everybody. One point that I love how you mentioned, somebody discovered something now because, well, probably somebody in the 50s did something similar for the tubulin. For the guys interested in this process, definitely it's synaptic vesicle precursors. They have been well characterized in the 90s and 2000 years in the nerve muscle synapse. I mean, you have this beautiful release along the nerve when it grows towards the muscle. The immature nerve muscle synapse has a lot of axonal release, and then it gives it up as it matures, and you can put it back by mutating things like adhesion molecules, like NCAM. And you have this, like 25 years ago, you have these NCAM papers with lateral release next to the synapse. So I'm sure if you were to look there in the muscle, these pictures will become so much more fascinating because it's more frequent. I mean, okay. It's a very, very, very well organized structure in the muscle. So yeah, this is it was mind blowing to see it last year, and I was like, shit, this was things that I was seeing in my PhD, 25 years ago.
That's very
beautiful stuff.
Thank you.
Beautiful stuff. But let's thank all the speakers once more, and Nadia. And now, please don't run away. We are not yet ready for lunch. We will have 3 short talks from PhD students of our group. So basically, first we'll have Sushovan. He's a PhD student.
Okay, it's 2. I think we should start. It is my great pleasure to introduce Dr. Stella Hurtley, the Deputy Editor of Science. She joined the journal in 1994 when they first opened the office in Cambridge, United Kingdom. Over the years, she has worked across a wide range of fields, including cell biology, developmental biology, prions research. She's also responsible for This Week in Science section, which summarizes the recent scientific advances. Stella studied at the University of Cambridge and earned her PhD at EMBL in Heidelberg. Alongside her editorial work, she has mentored many colleagues and authors over the years. She showed over the years great love for neuroscience and advancement, and I'm fairly confident that many groups in this very hall are very grateful for the input and help that you did for their work before being published. So Stella is a long-serving and highly respected editor at Science and within the scientific community with broad impact on life sciences publication. The talk today is Science Publishing and Publishing in Science. Well, for me, it feels a bit of palindrome. However you read it, you will end up with reviewer's comment. And I hope reviewer 2 is not here in the lecture hall. So without further ado, we are very excited and looking forward for your talk. And thank you for being here with us. And let's welcome our speaker.
Well, thank you, Ali. That was more than kind. Anyway, yes, so I am one of the editors of Science magazine. I've been there since the Dark Ages, more than 30 years. So don't ask me about anywhere else, because it's only Science that I remember. So whilst I've been at Science an awfully long time, I haven't actually been there since 1880 when it was started. But just to show you the sorts of changes that have happened over the years, that's the, original instructions to authors. Now our instructions to authors are a lot more detailed. Science, I think one of the things I want to make sure you understand is a not-for-profit journal published by a not-for-profit organisation, the AAAS. So we do a bunch of good stuff, and one of the things you'll be most interested in, though, is we publish the Science family of journals. So I only work for Science, but we do have a bunch of sibling journals, 4 also run by professional editors, so Signalling, Translational Medicine, Immunology and Robotics, and we do have a gold open access title, Science Advances. But those are the only journals from which you can transfer directly or from Science to those siblings. What I also want you to remember is that Science is run by a bunch of professional editors, so we're real people. So whilst the process as you experience it might seem very impersonal, in fact it is real people reading your papers, and when we say each paper is handled on its own merits, we really mean that. There is no, this is the way and this is the only way we will do things. So this slide was made before, during the pandemic, to be quite honest, and you can see that all of a sudden we had an enormous spike in submissions in 2020. And that was across all fields, but also particularly in the fields that looked after things that relevant to COVID and so on. But anyway, overall, we publish or aim to publish papers from across the whole realm of science. So that has a few percentages of the sorts of things we do. So going back to the editors, there are about 30 editors at Science who handle papers across all fields. So each of us is a discipline-specific editor, but over the 30 of us, we handle everything from astrophysics to ecology and evolution. So our publishing model, we run, our basic job is to run a peer review process. So at Science, we have a two-tier process where in the initial round of review, we have people out there in the wide world who are called our board of reviewing editors. So they help us, the editors, triage the submissions that come in and decide which ones to send out to in-depth review. And about 80% of all submissions are rejected after that first round, which takes 10 days up to 2 weeks. So those papers, we will not have in-depth peer reviews for you to help you understand our decisions. That said, the editors can often help you to understand the decision if you send them an email. But of the 20 or so % that we select for in-depth review, it's the editor's job to find the peer reviewers. So we will usually find at least 3 for each paper. We ask them to look at papers within 2 weeks. And then when we have the referee comments, we'll make a decision based on the referee comments, based on our own judgment, based on circulation amongst the editors. So whilst only one editor is responsible for looking after your paper, each paper will be seen by at least one other in-house editor, as well as the board of reviewing editors, as well as the referees. And again, it's a very selective process, so that the net number of papers we publish, it depends a little bit on the field, but overall it's 6 or 7% we end up publishing from the original papers that come in. And with respect to numbers, we sort of by definition publish 15 papers a week, and that real papers, so research papers a week. That number hasn't changed in the 30 years I've been at Science. So while Science has expanded, with a small s, Science magazine has not expanded. So and whilst the number of submissions has gone up, the number of papers we can publish hasn't gone up. So the process has, whilst it seems more difficult, it really has become more difficult. Okay. I've already mentioned the board of reviewing editors. One little extra thing that we do at Science is once we have all the referee comments on board, we do cross-review. And cross-review at Science, what we mean is all the blinded referee comments are shared with each of the referees who saw the paper, and they have an opportunity to make extra comments. It's not conversation between the referees, as is done by some journals. It's you as a referee have a chance to have some input about what the other referees have said, or to say something extra about what you said. So, for example, given we often have interdisciplinary papers that we're judging, so I have a neuroscientist, I have a cell biologist, and I have a microscope tech person. Quite often, the criticisms of one will sort of contradict the praise of the others. Actually, not often, but sometimes. Anyway, cross-review gives us an opportunity for the referees to either downgrade or upgrade or just clarify their thoughts on the particular paper. And we've found that mostly it's a for information thing, but when it's useful, it's really useful. Okay, so Science, we're basically, we consider ourselves a top-tier title, and so what we want to see in papers is a significant advance in scientific understanding. So we'll expect you to present novel and important data, concepts, and to be interesting beyond just your immediate field, but also we expect excellent technical quality. So for you as authors, how do decide whether to submit to Science versus whichever is your favorite society journal. So, you know, the first most important thing you do is your research. Obviously, that's what takes most of your time. That's what you spend most of your thought process on. That is the most important thing that you do as research workers. But, you know, we all know the publish or perish thing. You need to get your work published in order to share it more widely. So, you've done your work, the basic work that you need to get to the stage where you're writing it up. You write it up, and then you should it's advisable to stand back and assess the work both yourself and to have other people whose judgment you trust to also help you assess that work. So, I mean, hopefully it's your best work. Hopefully it will have some potential to have major impact. Hopefully it will also be interesting to people beyond the people that work in your own lab. The sorts of things that are often deemed interesting in that way will be things that overturn conventional wisdom, so things that people see and think, oh, I wouldn't have thought it works that way. So the paper obviously has to survive peer review, so it has to have convincing data with appropriate controls, be presented carefully and in a balanced way. And papers often fail. I mean, realistically, they fail because either fail with us is because they're not deemed either a significant enough advance, or they can fail because of they're not deemed technically excellent enough. So realistically, the submissions that we get are all good papers. They'll get published somewhere, but it might not be science. Things that help are a good cover letter. So the cover letter is your opportunity to communicate directly with the editor. The referees don't see that. The board of reviewing editors don't see that. So concisely say, make us understand why you submitted the paper to us. All right, I had loads more slides that I don't have time to show you, which is absolutely fine because it's all, to a certain extent, common sense. Real editors handle your real papers carefully and as rapidly as they can by running a peer review process that we aim to be, whilst it's critical, we want it to be fair. We want you to understand our decisions when we communicate them to you. And the other thing I wanted to say is that, so I already mentioned that science is published by a not-for-profit organization. The papers that you publish with us, we have a green open access policy, so you don't pay us to make your paper freely accessible. You are allowed to put your own papers at places like PubMed Central to be freely available, so the accepted version of your paper can go onto those public repositories as soon as they're published, and that's the way we get open access at all the professional editor run titles, and you have a free electronic referrer link, so on your own website you can put that link and people can get to the sort of nicely formatted version of your paper at the Science website without paying us or anyone else anything. So I'll let that visuals play because they're very proud of their visuals-y stuff. If you want careers advice, we have a careers advice website. And thank you for your attention.
We have time for 1 or 2 questions. If not the case, I mean, you're also staying for dinner with us. Would you have time?
Yeah,
Then, yeah, I think then you can ask her directly. Okay, then we move with our next speaker. Now we move with our next speaker, Dr. Doa Mugahid. So Doa, if you allow me, I will start with introducing you while we set the stage. So Doa is a researcher and scientific editor with a background in data science and public health. She also served as an associate editor at Nature Communications before joining Harvard's T.H. Chan School for Public Health, where she is a data officer for high impact TB. Did I say it correctly, Doa? So how? Yes, okay. High impact TB, so it's written in a weird way. Consortium. This NIH-funded international collaboration brings together more than 100 scientists to advance tuberculosis vaccine research. At Harvard, she coordinates data management, curation and sharing, and helps link academic, clinical, and industrial partners working with large immunology and diagnostic data sets. Obviously, she has much bigger profile that I cannot really fit here, but this is all cool, but the most inspiring part of your work, at least for me as a person from the Middle East, from Lebanon, is that the Science Arab Forum, who was established by John Daniel. Do I see John in the audience? Oh, he's not here. Okay, he is. Maybe stand up so people can see you. So John established this and is being weightlifted by Doa. She's the chief editor of this forum. Who I believe personally, I'm obviously biased. It has a very noble cause where it tries to empower Arab scientists, and especially from the MENA Middle East region. And with that, actually, we have two students that won the SAF competition that was led by Doa and John. And we have them in the audience, Ala. And yeah, maybe you stand up, both of you, and let's clap for them. So thank you for being here. And thank you, John. Thank you, Doa, for this noble mission. And without further ado, I'll leave the stage for you.
Thanks very much, Ali. I've done so many things that I find people try to put me in boxes and it never works out very well. So I'm here, but I'm here representing the Scientific Arab Forum, and it's a real pleasure to be here. Congratulations to Ali, to Silvio, and the GEF25 team on bringing such a great group together. Okay, without further ado, I will be talking about the need to democratize access to critical research infrastructure. But first, before I get into that, I did want to say a few words about SAF. So our vision is to make science a household topic in the MENA region. And our mission is to promote scientific excellence, communicate scientific knowledge, and influence science policy in the Arabic-speaking world. And we do that by creating a visible community of outstanding researchers, of
of whom Ari is one member, and John is also here as founder. We try to promote our members to regional and global leadership in science and provide thought leadership in the community, as well as shaping the intellect of the general public by making science accessible to people in Arabic. If this speaks to you as a member of the audience and you would like to learn more about the forum, please scan the QR code. If you have trainees in your lab who you think would be a good fit, please also put them in touch with us. We'd be very happy to have them. Having said that, that is not the premise of my talk today, but my talk really stems from my belief that the modern world is increasingly connected. We have increasing data to prove that. And some of the really nice things that have come out of this increasingly connected world is we can collaborate with people across very long distances through virtual interactions, the globalization of supply chains, and the flow of global talent, which I think are very strong stabilizers for the world. But the other hand of this, obviously, is that pandemics spread much faster, where climate change is a shared problem that requires very innovative, very large-scale thinking. Aging is a problem that almost every continent except for Africa is grappling with because of an increase in longevity and a decrease in birth rate. And obviously, the scale and implications of geopolitical changes around the world are felt much more broadly. And so I'm an idealist and I'm also a scientist, and I spend a lot of my time thinking how we could do better as scientists to make the world a safer, more stable place for everyone. Yet, unfortunately, when you look at the stats of where science is being done and who's publishing science, it is striking to me that 6 countries in the world produce over 50% of scientific literature. And it's the reason that is very striking is that means there are giant gaps in global knowledge and data. So how are we going to fix and fill those gaps? I think there are many things that need to be done, but what I'm going to talk about today is really the need to democratize access to experimental infrastructure. I don't need to tell anyone in this audience that this is kind of the premise of what we do in science. We take money from taxpayers to train and retain scientists and buy and make infrastructure that allows us to then create samples, collect data, write code, synthesize all this knowledge and put it out there for the next generation of scientists or another group of scientists to build on. And the thought is this cycle then can lead to improved health outcomes and also lead to future innovation that brings in more money that is then pumped into this cycle. I think we've done a pretty good job with building infrastructure or working towards building infrastructure for biobanking and sample collection and sharing, bio-bricking and standardization. We've also been getting towards better data sharing, also models and code. We have infrastructure to do that sharing better. And no offense to the commercial publishers, but preprint servers and peer review platforms are also like really making headway. And I think this is very good for science. But it's still, our main problem is at the fundamental level of how science is done in the lab. So we are generating more and more amounts of data everywhere in the world. Yet, and that means we need more compute and we need more data storage, but we also need new experimental methods to probe new things in new ways all the time. And while I think, and we can discuss and debate about this, that cloud computing and cloud storage has solved largely the compute and the storage problem, what is an unsolved problem is the inaccessibility of experimental facilities. But since this is a life science audience, I will focus my talk on the life sciences. But I think we've been for centuries trying to understand how RNA, how DNA, how protein, sort of the essential building blocks of the dogma, central dogma in biology are what they are, how much there is, what the spatial organization is, and what their structure looks like. And these tools exist. And we're making lots of progress on that front, but they're so inexpensive in ways that people cannot imagine for other parts of the world, that it's really led to whole regions being excluded from engaging in this scientific endeavor. But we do have a poster child that makes me always think that we can do better. We can push the limits of technology in ways that make biology accessible to everyone. And I draw a lot of inspiration from transcriptomics and genomics, where we started off at one point, and within a few years, with the advent of more accessible technology, cheaper technology that was driven by commercial providers, we have been able to push the price of next generation sequencing technology exponentially. Who are the players on this field? Everyone knows who Illumina are. They really popularized short-read sequencing. But we also have other players in the field, like Ultima and Oxford Nanopore Technologies. And I like to think a lot about how to make infrastructure accessible and what are the success stories. And I noticed that one thing that Illumina did really well, and I think has come out in this many of these talks, is multiplexing. So they've done a really good job at multiplexing, and when you multiplex, you bring the price per unit down significantly. Yet they have a very large physical footprint, and despite years and years of development, the cost of their infrastructure, of their machines, has not gone down. We have Ultima, who are trying to push the boundaries there. They make cheaper chemistry, very importantly. They have a lot of optics-free parts in their machines, which has also brought the price down, but they're still large and bulky, and they have while they brought the cost down a third to a half, these are estimates, their numbers are not public, we can still do better. And that's where Oxford Nanopore has really set themselves apart. They have cheaper core technology, which is also optics-free. It goes from tabletop to handheld almost, and the price of their low-throughput technology is 10% of an Illumina setup. So doing great there. And for contrast, this is the size of Menayan from Oxford Nanopore, really tiny compared to what Illumina and Ultima have to offer. And the really nice thing about having accessible technology is that you're not just engaging more people in science. This has a compounding effect. And we've seen this in genomics and transcriptomics, with the advent of next generation sequencing being so widely popularized. We've been able to do things like single cell genomics. We've been able to do spatial transcriptomics, which has been opening our eyes, and we're learning new biology every day. And I really hope we can do this for other biomolecules. But it is no secret, I think, that despite mass spec being around even before next generation sequencing, the technology has been very, very slow to move forward. And this is a sort of an indirect metric of that. Next generation sequencing is mentioned so much more in 2023 than a mass spec in most papers. And the same, I will say, is for protein structure determination as well as DNA and RNA. The technology is just requires a lot of expertise, is very expensive, and that has really held the field backwards. And obviously DNA and RNA also hard to structure and put in crystals and all of that. But there's a need for fundamental new ways of doing this kinds of measurements, and I think expansion microscopy really allows us to do that, and why I've been very happy being in this crowd today. So a few things to say to wrap. What I think could really help with accessible technology development, especially in academic labs where innovation is really being pushed to the boundaries and forefront, is to attract, train, and retain talent from low resource settings or limited resource settings, because their idea of what is expensive is so different from what your idea would be if all you've grown up with is, yes, I can just buy this off-the-shelf thing that costs $100,000 because I'm at this and that university. They have a frugal first mentality, and that is a valuable thing, I think, in the scientific community. I also think we should train and build multidisciplinary research teams, and we've seen some elements of that today, because that's where out-of-the-box ideas are bred. Company creation whenever possible, because in academia there's a lot of stigma around doing that, but I think this is maybe one of the most effective ways to unlock economies of scale. But if your technology is inherently not scalable, right, then, or you have things that you can't patent, right, then please engage in open development so that others can rapidly build and improve on ideas. And I think this is very true for this field. And finally, something that a lot of people don't like to think about, standardization. In fields where methods and technology move very fast, I feel like spending time thinking about standardization and interoperability is really important, especially today in the world of AI. And with that, I would like to conclude, and I thank you very much for your attention.
Okay, time for questions. Okay, I have one. So I'll share this little bit of weird, boring story with you guys. Please bear with me for a second. We wanted to apply for the AI team in my group for this computing service that's partially funded by the German government. And I realized as a person from the Middle East, I had to prepare two additional documents. The first document that I will not use this infrastructure for generating weapons of mass destruction, nuclear weapons. And the second document that Silvio had to certify that I'm a real person. So the question now to you, for the data management services and the AI part, is there geographical restrictions and also nationality restrictions for these services?
For services, these are like, it's Amazon, it's Google, it's whatever, you know.
Which
like
most likely there is then.
So they inherently serve all continents. The problem is data sovereignty. So where is the data? And I don't have that stat right now, but I can pull it up if anyone is interested. Data centers are very localized in certain parts of the world. So Africa, for example, has a giant gap in data centers. So if you care about data sovereignty and you want your database to be on your soil, then you obviously, it would be very difficult for you to
thankfully,
engage.
we don't have to worry about it in Europe because we don't even allow it in the States to be stored. So we have to store it locally. So yeah, but this, I understand your point, yeah?
Yeah. That's okay.
So do we have more pressing questions? If not the case, let's thank our speaker and Denise. Yeah. So in the meantime, I will start introducing my friend Denise. Can I call you my friend? I mean, Denise and I were stuck for a 12-hour flight by accident from Rio de Janeiro back to Frankfurt. And she realized, oh, you gave the talk, the concluding plenary talk in Rio. I said, like, yes. And then she introduced herself, and I kept blabbering for the next 12 hours. I hope you didn't hate me for that. So Dr. Denise Soltukoglu is a cell biologist with a PhD from the University of Freiburg. During her postdoctoral research, she worked at the interface of biology and imaging technology, helping researchers apply advanced microscopy to address complex biological questions. In March 2024, she joined EMBL to lead the Imaging for All funding program, which provides researchers and imaging specialists from low and middle income countries, or like Denise would like to call it, the global south, with access to the state-of-art imaging equipments and training in the Western world. From my perspective, I want to embarrass you a little bit. I think of Denise as a force of good in science, and I truly like what you do here. And actually, my group benefited from your initiative, the initiative that you are leading, by having these interns joining us. And one of them, I'm trying to convince to stay for a PhD position. They are very talented people, very smart. And then when you join, you see this motivation and being appreciative of this initiative to get access to this high-end imaging equipment. And this experience that they gain. So Denise will tell us more about this initiative. Let's welcome our speaker. The stage is yours.
Okay, thank you for this super kind introduction and the invitation, of course, for me to speak here and being a big supporter of this program. So I think Doha really set the stage for this talk, and here I would like to take the opportunity to introduce you to this funding initiative called Imaging for All, in short I4A, which is a pilot mobility grant that was created in partnership between the UK-funded Wellcome Trust and Global Bioimaging. So Global Bioimaging office is situated at EMBL Heidelberg, and Global Bioimaging is a network of bioimaging communities across the globe. And the grant's purpose is to support scientists from low resource regions, and the metric here is the low and middle income country list published by the OECD. And it is to access, for low resource regions, to access bioimaging technologies and expertise that they need for their scientific work. So Wellcome sees bioimaging as a vital part of the future of health research. They fund progress in bioimaging, and they want to understand the barriers that affect progress in this area. So not so long ago, they commissioned a report on identifying these barriers, and these can be grouped. So the result of the report is they can be categorized in two. So we have infrastructural barriers and we have technical barriers. And technical barriers are what this conference is also dealing with, like spatial resolution, spatio-temporal resolution, how to manage bioimaging across scales, and what to do with this huge amount of data that is being generated. But not surprisingly, low and middle income country researchers emphasize infrastructural barriers much more significantly than the high income country counterparts. So these are high instrument costs, lack of availability of equipment and technical expertise, also inadequate communication and technology transfer to the global south, and competition for funding. So to address some of these points, Wellcome decided to experiment with this low-hanging fruit of establishing a mobility grant, and they started Imaging for All as a pilot project. The aim here is to provide opportunity to travel to resources that are otherwise seemingly out of reach. So this is about reducing barriers in science and democratizing access to instruments, training, and expertise. So why join forces with global bioimaging? So this is where I work. So it is because of this map that you're seeing here. So we are a global network connecting imaging communities across continents. We have ties with the global north and the global south. And in some cases, our partner communities are research infrastructures, like official research infrastructures like Eurobioimaging, and perhaps many of you in the audience are familiar with this. Or we have community-driven efforts like Latin America Bioimaging, LABI. And in fact, the LABI Super Resolution meeting in Rio in 2024 is the reason why I'm here. This is where I met, I had the opportunity to meet Ali. He had a talk, plenary talk about one microscopy, and he was also speaking about expansion microscopy from the perspective of democratizing super resolution. Yeah, as he said, we had a coincidence. We came back with the same flight, and we had a long time to chat, and he became a big supporter and one of our hosting labs, actually. So how does the grant look like? So it is for researchers and imaging facility staff alike. It supports visits from a week to a few months, depending on which track the applicant is applying. And it is to labs with imaging equipment, or to imaging facilities, or to imaging courses that is anywhere in the world that the applicant wants to go to. So the applicants can have connections with the host at the time of application. This is of course nice. Or they can ask for support for this matchmaking with the hosts, and we try to do our best to match the applicant to a host. The budget covers almost everything from equipment usage fees, service fees, travel accommodation, visa, and so on. And overall, in this funding period, we have a budget to award about 120 awards in 3 structured open calls. So the money can be used for conducting experiments, or it can be used for skills development. So some of the items here are accessing instruments not available locally, learning specialized sample prep methods to take home, developing imaging assays, learning from experts, setting up optical systems, participating in imaging-related training courses, strengthening image analysis skills, data management skills, and etc. So as I said, we've had 2 open calls already. So we received about 200 applications from 33 different ELMIC countries, which is to me a clear sign of demand, and it does address an important gap, and it also shows good outreach work and the extent of the global bioimaging network and its partners. So applicants chose overwhelmingly European hosts. I think this is reflecting this strong open infrastructure culture that we have in Europe in the form of open access imaging facilities, but we also had applications to individual labs. So we are looking here at a 40 to 60% distribution in the case of Europe. So among the courses and projects that we already funded, there are some nice examples. So I'm going to give you some of them. So we've had people going already to fundamental courses in light and confocal microscopy, image analysis, but also to courses that teach advanced methods like imaging-based spatial omics course or advanced imaging techniques course at EMBL. For this audience, I will emphasize this tissue clearing and expansion microscopy workshop at the Harvard Micron facility that was attended by a PI with a new lab in Ecuador. He had obtained his PhD in the US, and this is so in the feedback, he put it in a nice way. Expressing the goals of the program. So he said, we don't have confocal microscopes, and detection of proteins is complicated in large frog embryos. So they work with this poison frog embryos, and he says tissue clearing and expansion microscopy are 2 techniques that can help us get the images we need. And I actually went and checked the content of the course and saw that Ed Boyden was also one of the instructors there. Now to the projects that we've been able to, so these are now the projects that we've been able to support so far. So we supported cryostructural studies on a mycobacterial adhesion protein that is important for the bacterium's infectivity, supported imaging mass spectrometry, so a researcher going from Brazil to the M4I facility in Maastricht. So we have lattice light sheet of cardiac organoids. Also open source 3D printed microscope applied to microbial research. So one of our grantees took one of these microscopes back home to Argentina. Here I also want to emphasize the expansion microscopy project. So we also funded 2 expansion microscopy projects for Ali's lab as a host. So Luciana, who is a PI from the University of Buenos Aires. So in her case, we did the matchmaking with the host. So she came to Ali's lab to study protein arrangements in single vesicles in the secretory pathway. And I think she's now applying for additional funding to come back and continue this work, and she wants to establish the methodology in her lab in Argentina. And we have funded Malak, who's right here in the audience. She's working on the characterization of the toxic amyloid beta 42 in serum of Alzheimer's patients with 1 microscopy, and as I hear, her project is going pretty well. So as a note, I do think expansion microscopy is particularly interesting and special for this funding initiative because, as it's been said before, it does open the door to advanced imaging in resource-limited settings where people may not be able to afford fancy equipment. And I would really love to see more of these projects supported through us. So with that, I want to finish off with the announcement that our third call is going to open around December, with visits starting from March next year, about. So applicants hear about the program from the hosting labs and facilities quite a lot. So my message would be, so if you have LMIC contacts who are in search of funding for a scientific visit, so you can point them to this program, and if I were to reach out to you for hosting an applicant, so now you know who I am and what the initiative is about. So yes, so we have social media accounts and we have a newsletter, Global Bioimaging. You can learn more about the other activities that we have other than Global Imaging for All. And yeah, my email address and our website down here. Yep.
Thank you. Do we have a question? Nope, he's going out. I have a question for you. What happens after the third call finishes?
Yeah, we recently had a meeting with Wellcome, and we are going to see, so there needs to be another funding period. So if there is, which is, yeah, we will see. So the call, it will continue. Yeah, we will continue supporting.
So then I will continue, if you allow me, with some shameless advertising. So if you believe in the message that Denise is trying to send, especially the colleagues with big social media accounts like Rita, Christophe, Paul, Virginie, Tiago, if you believe in this noble message, I'm biased, I like the message, maybe give this initiative a shout out on social media platform. We will be very grateful.
And be some competition to you.
Yeah. Thank you again.
Okay, yeah, thanks.
With that, we move with the last editor speaker in this session, Anja Wecker, Dr. Anja Wecker. I will, if you allow me, I will start introducing you.
Part of a barrier. His talk is entitled Expansion 3D Stat Multiplying Resolution. We are looking forward to your talk.
Okay. Hi, I'm Gero, and I would like to talk today about how we can combine optical and expansion super resolution microscopy to get the very best out of our samples. We're from a company here in Göttingen, but we have offices in all parts of the world, and we are dealing in anything from confocal to super resolution microscopy. We not only provide microscopes, but also dyes. And amongst the microscopes, we have entry-level machines with a 2D super resolution, the Steadicon we call it, and going down to the Mirava in the middle here. This is our newest model and probably the one which is most useful for anybody working with EXM as well in this combination, because we can do 2D and 3D super resolution with this machine, 2D down to 20 nanometers and 3D down to 75. We also have MinFlux integrated in here, where you can, without any expansion, go down to about 3.2 nanometers resolution. And we have a larger MinFlux machine, which is specialized to have even 3D and tracking functions in MinFlux. So why do we want to combine optical super resolution and expansion microscopy? Well, if you look at the principle, which everybody here is familiar with, we have our original sample, we expand it, and well, simply speaking, we see the difference straight away. If we use an epi or confocal microscope, we can see structures which were previously just a blob, basically. With STAT, we can basically go, each time we are doing expansion, one step further, or even in non-expanded. So if you expand 4 times, you can go another factor up using STAT. So you're always one step ahead in optical super resolution, even when looking at expansion samples. We've done this back in 2021 in a workshop at Mifobio in France with Virginie Georgette, and we looked and compared confocal images, non-expanded, expanded here of nuclear pores, and you start seeing the ring structure, and with STAT you see similar results, maybe actually take further in this case, and then combining both, you multiply the resolution enhancement of both methods. Adding deconvolution on top, which is built into our software, we can see actually here now subunits, you see the dots of the single parts. For the combination of STAT and EXM, I think we need to zoom in a bit more. And now we see really the detail you can get on the nuclear pores. And this is actually going somewhere towards the resolution you would see with the MinFlux, which has just pure optical resolution beyond that. But I think the combination of STAT and EXM can really give you really nice results. And we were working on this from the start. So we were collaborating also, or providing some images for the early paper of Paul Guichard and David Gambarotto, which came out the same year as the Evers paper, which we've heard earlier about. And here we can see the confocal is already really nice. We're looking here at, procentrioles, which is the much weaker structure than the actual centrioles. And we were able to get a much improved resolution of the subunits here. Why is it important that they are the procentrioles? Because they are much smaller than the actual full kind of barrel-style structures. And to see these, we are working here with the highest efficiency detectors to actually be able to image these structures with full signal here in super resolution. Can we look at the whole structure? Yeah, if you're looking here at the both proper centrioles and the procentrioles here, we can image this in 3D. And this is actually the raw data. So this is right out of the detector and rendered. So you can take this even further with deconvolution approaches, for example. And here the trick was not only the sensitivity not to bleach the sample, but also something that we call adaptive illumination. I'll show you this in the next sample here. So we took this not only to specialists who published these methods papers, but this was just customers coming to us and having a demo on the system. And you can see a similar structure, similar image, 2 channels here. And this is the trick. Basically, we don't illuminate the whole sample. We can actually switch the lasers off in the pixels which are not used. And in a volume, that makes a big difference. So you can image your volume at a high resolution without losing all your fluorescence. We call that full exposure, and it's built into the Mirava microscope. Obviously, I don't want to only show centrioles. We also looked at other samples here, trypanosomes. These were provided by Melanie Bonhiver. And here we were actually working with green dyes. And when we apply STAT, we see already a bit more structure. Plastic convolution, we can now see here the 4 microtubules, which we would find in this flagellar attachment part. Similar, another parasite, Toxoplasma. This was early on with Isabelle Tardieu, who we also worked with at the workshops. And here we did another 3D imaging, and you can see the enhancement from the confocal image to the top. The whole method is done scanning, so we don't have to do large post-processing or anything. It's really acquired like a confocal image, just with STAT technology. We make the scanning spot much smaller. And with full exposure, we can make sure that in a 3D volume, we don't bleach our structures doing so. Last image I would like to show is root EXM, which was this year published by Magali Crison, who also spoke or speaks here. And here we see plant cells, which are traditionally hard to label. But here with EXM, it's a nice combination because you have to digest the sample anyways. And you can, here in this case, do a NHS stain, which gives you a pan staining. And looking with the STAT, we can see again more detail in the sample as well. What I look forward would be trying also our matrix detector, which can do much faster imaging, higher detection rates if you want to. And it could work in these denser samples where you have pan staining. So in this case, you could probably get an even better sectioning and detail without any background in these samples. So this will be very interesting. And I'm also wondering what will happen if we combine MinFlux and expansion microscopy, because we there have a much higher optical resolution of 3 nanometers already. But in total, yeah, we have definitely worked a lot with expansion microscopy before. It works really nicely. The adaptive illumination or exposure helps you not to bleach the sample, and the sensitivity works. For the dyes, yes, we have tested a lot of them with expansion as well, and we have here representatives outside who you can ask. And if you're really interested, come and visit us. We are here in the same city. Our headquarters are here. Contact us, and you can actually look at the microscopes. If you have a sample or not, we can give you a little tour. Thank you.
Thank you very much, Gero. In the interest of time, we are continuing with our next speaker, Sven. Please reach Gero at his booth or at dinner. He will be happy to have questions. So Sven is part of E11 Bio. I don't know if it's still part or now he's starting his lab at Cambridge. He can tell us more probably. And his talk is entitled Multimodal Multiplexing with Expansion Microscopy as a Path to Molecular Connectomics. Sven, the stage is yours.
Thank you very much for the kind introduction. So I would also like to start out by thanking the organizers, in particular Silvio and Ali, because my own journey in expansion microscopy actually started here in Göttingen 10 years ago, when in the late stages of my PhD, Silvio approached me with the first paper printed in his hand and said, "This is super cool, you should try that." And I did, and basically I never stopped. And today I want to show you some progress that we have made at E11 Bio, which is a non-profit organization. So I feel like a little bit of an imposter in the company section here, but still not a lot. PRISM is a technology that basically helps us with enabling molecular connectomics using optical techniques. And some of you have already seen and approached me about this. The manuscript that shows everything that I'm talking about here is now on bioRxiv. But I would also encourage you to scan this QR code and sign up to E11's mailing list, because there will be a launch announcement on Wednesday, and you will also be able to keep up to date with new technologies and developments coming out from E11. Okay, so what do we understand molecular brain mapping to be? In our case, we all base it on expansion microscopy. This is the technology platform that enables everything that I'm showing you in this talk today. And then you can, of course, get the the morphological readouts, as we have so nicely seen in the talk by Hans Danzel showing Lycon, developed by Mojtaba Tavakoli, who is now leading a group in Janelia. And on top of that, we can get molecular readouts for endogenous proteins, for example, synaptic markers, and we get access to all the molecular tools that exist in molecular biology. And I'm going to focus now on molecular readouts and molecular tools. So what are we trying to address? There's a huge scalability challenge in connectomics. This here is to scale the first connectome of a fly brain. Here it is magnified. It took a lot of time and it cost a lot of money. And you can scale up to microns, which cost even more money, which is 1 cubic millimeter. And obviously, we all would like to ultimately get a whole mouse brain, which will be, with current technology, $10 to $20 billion. And that money is mostly in the proofreading. This is a report from the Wellcome Trust, led by Greg Jeffries from the LMB. And the major challenge is really getting down the proofreading cost if we want to scale up connectomics. And this is what we are trying to do at E11. So here, let's take a look at the PRISM pipeline. How does this technology work? First, we deliver viral barcodes to neurons. So this is a mixture of viruses, each carrying a protein tag, and they randomly combine as they are injected into a neuron. And the point of this is, with morphological readouts, as we do in EM or Lycon, you get the very nice morphological structures at very high detail, but they all are one channel of information. Adding barcodes, as you can see here on the right, will help you to distinguish these neurons from one another. So after we deliver the barcodes, we perform expansion microscopy. This is the basis of everything. This is also at E11 to a large part led by Hugo Damstra, who is in the audience today. Then we perform multiplexing to read out these barcode bits that we delivered. So first round, extinct the signal, second round. This was majorly led by Rosa Park, and we will see some of her data also later on. Then you can also put in morphological labeling, which again, Hugo will talk about more in his talk just later in the session. And finally, you have to read out the data and reconstruct it using computational pipelines. And here I would like to highlight Arlo and also Julia, who has been also instrumental to getting Lycon up and running. Okay, let's look at our milestone data set that we acquired using this technology. This is about 13 million cubic micrometers of mouse hippocampus CA3. We are multiplexing 25 different protein targets, 18 of which are barcodes, 6 are synaptic markers. I would like to highlight this is a project very much held together by the vision and the project lead of Andrew Payne, who is the CEO of E11. And Arlo developed the segmentation strategy. He is one of the first authors. Rosa did the data acquisition. She's the second first author. And that was done at Sam Rodriguez's lab at the Crick Institute in collaboration with us. Okay, let's take a look at the barcoding. This is part of this volume of hippocampus, and now we focus on one neuron, and you can see bit by bit we are reading out the barcode. It's 0 or 1, barcode is present, or barcode bit is present, or it's absent. And that allows us to assign each neuron its individual barcode. And now you can see all these individual bits being put together here. So that's great. You can see some static images of this here. It works on the level of the cell bodies. It works on the level of the neurites, axons, dendrites. It traffics very well. And now we take all this information and we put it together. This movie shows you the final reconstruction of this volume. And each neuron has a different color assigned based on its barcode identity. Here we also see now the neurites shown in black, and highlighted are pre- and post-synaptic labels, which allows us to assign synapses to these meshes. So how does the barcoding help us? Well, one challenge in connectomics is you often get disconnected volumes. You can see here two examples based on affinity segmentations, and you can see these segments have different colors. The different colors indicate that there are splits between them, and they're actually not connected in the automated readout. But if you take into account the barcode information, they all have the same color, so you just assign them back, and you can correct using the barcode information your segmentation. And this should hopefully really help us drive down costs. So this also helps us to bridge actually across distances. So if you have an experimental problem, a bad slice or a bad plane in your image, the barcodes can help you to actually cross those gaps too. And yeah, more about this you will find in the manuscript. Speaking a little bit about molecular data, we have here an annotation of excitatory and inhibitory synapses. And you can see it all makes sense. The inhibitory synapses are clustered on the cell body, as we would expect them to be, and the excitatory synapses are more distributed across the dendritic tree. And of course, we can also not just multiplex barcodes, we can multiplex endogenous proteins. So here we read out GFAP as a marker for glia, for astrocytes, synapsin as a marker for synapses, and then we have Bassoon and PSD95 to identify excitatory synapses, and GABA and VGAT to identify inhibitory synapses. And of course, you can imagine that you can extend this multiplexing technology to do even more in the future. We can also do nice statistics on this just to convince ourselves that the labeling is correct. When there's an excitatory synapse that has PSD95 and Bassoon, obviously would expect to not find GABA and VGAT, and that is indeed what happens. And I've only really shown you a small slice now of what is possible with molecular connectomics. There's way more that we can do. We can, of course, do the neuron barcoding. We can do assignment of synapses on top of the barcoding. We can look at organelle characterization using the morphological data. We can look at the synaptic sites and characterize those. You can actually count the nanocolumns present there. You can even go and look at the nuclear pore complexes, and you can see that you kind of make out the shape that we know them to have from more structural data. And yeah, you can do the neuronal mapping, obviously. Again, this is only a small slice of what will be possible in this field in the future. And yeah, as mentioned in the introduction, I'm currently also sharing a small announcement for myself. I'm starting my own group at the Laboratory of Molecular Biology in Cambridge. I have 3 open positions for a PhD student, for a postdoc, and for a data engineer. You can find those online, or you can approach me directly. So there will be some nice opportunities for people interested in working on this. And likewise, E11 is currently looking for a multi-omics scientist to help with the molecular brain mapping. So there's a lot of very exciting opportunities in the field. And Mojtaba Tavakoli, who is here in the audience, he is also looking for somebody to work with him at Janelia. And if you scan this barcode, it will lead you to that particular call. But please also approach him directly. Okay, these are all the people who made this possible. It was a massive collaborative effort. The people at E11, also we have a board of amazing advisors, and I would like to highlight our collaborators, some of whom are in the audience at Boyden, Sam Rodriguez at the Crick Institute with Rosa, and then Jürgen, who is also in the audience here. And finally, if you want to learn about PRISM, please contact Andrew Payne, co-founder, CEO of E11. And if you want to talk about molecular brain mapping, maybe in relation to my group, then please contact me. And again, if you would like to take another opportunity to scan the barcode, it is here. Thank you very much.
Thank you, Sven. I have questions, but really we need to move on. Sorry, please guys, approach Sven afterwards. Our next speaker is Stefan Frey, who is representative of another local company, Nanotech. He's going to tell us about smart secondaries, and his talk is entitled Rethink Immunofluorescence with Smart Secondaries. Stefan, the stage is yours.
Hello, so today I'd like to introduce you to our smart secondaries and their use in multicolor immunofluorescence applications. So I'll start with some really trivial facts first. So for a comprehensive understanding of a complex biological system, for example, a eukaryotic cell, we need to know the relative localization of the important key players. This is classically done by indirect immunofluorescence, where the target of interest is first recognized by a primary antibody, which then in turn is recognized by a fluorescent secondary antibody. If we want to image more than one target at a time, we need to have primary antibodies from different species, along with multiple species-specific secondary antibodies. So obviously, this multi-target imaging is quite limited by the number of host species that we generally use for raising primary antibodies. So for spatial proteomics, for example, we require imaging of many more than only 1, 2, or 3 targets at a time. So for this purpose, for indirect immunofluorescence is not an option. So what we can do to circumvent such species limitations is either using directly labeled primary antibodies, which is quite expensive and may not be tolerated by all primary antibodies, or we actually try to preform primary-secondary complexes before we do the staining. Well, unfortunately, such premixing does not work with conventional secondary reagents, as we see here on the right side. So why is that? The first is that conventional secondary reagents are bivalent. That means that each secondary antibody will bring in another free binding site for an additional primary antibody. At the same time, these conventional secondary reagents are polyclonal, which means that multiple binding sites there are on one primary antibodies, and together these two properties lead to massive aggregate formation when we mix primary antibodies and conventional secondaries in solution. And these aggregates are exactly the bright dots that we see here on the right side. So our solution at Nanotech is smart secondaries. Smart secondaries are secondary single domain antibodies that bind primary antibodies with an extremely high affinity and in a species-specific manner. As they are monovalent, they do not form these clusters shown before. So we can form complexes of primary and secondary antibodies, in this case smart secondaries, in a test tube. And because of the really high affinity, they will then be stable for hours or even for days. Using such preformed smart secondary complexes, we can now indeed perform one-step immunofluorescence, get results very similar to the sequential two-step setup. Of course, this saves a lot of time, but there are definitely many more important implications than that. So if we preform several of such complexes in separate tubes, then mix these complexes when they're preformed and apply them to this sample, then the primary antibodies which are present in each of the complexes will target the bound secondaries together with the fluorophores to the correct target structures. Importantly, now there are no limitations regarding the species or the isotype of the primary antibody anymore, which means that multiple primary antibodies, even from the same species, can be used on the very same sample. At Nanotech, we actually provide such smart secondaries in various formats for different applications. I would like to give a few examples. The first example is a one-step immunofluorescence using simple fluorophore-labeled smart secondaries. So in this experiment performed by Felipe Opaz's group, 3 mouse monoclonal IgG1 primary antibodies were used on the same sample. For that, as shown before, the primary-secondary complexes were first preformed in separate tubes, then mixed and used for a one-step immunofluorescence followed by conventional confocal imaging. So as you can see, although using 3 mouse monoclonal IgG1 antibodies, the signals from all the 3 markers remain nicely separated. The second example I would like to show uses smart secondaries featuring cleavable linkers. Such tools are used for a technology called Nanoplex, which Felipe already shortly mentioned on Saturday. So Nanoplex is a 3-component system. We need first a primary antibody, secondly a Nanoplex reagent, which is basically a smart secondary which is fused to an alpha tag via a cleavable linker. And we need a fluorescent NB-alpha, which is a high-affinity nanobody binding the alpha tag. If we mix these 3 components in solution, then we get a ternary complex connecting the primary antibody with the fluorophores via a cleavable linker, basically. Due to the high binding affinities, this is kinetically stable, this ternary complex, and can be used for immunofluorescence applications. The beauty of this system is now that it is possible to release the fluorescent NB-alpha by using highly specific and very mild stimuli. Depending on the type of stimulus, we can discriminate between optoplex, enziplex, and chemiplex. As the name already proposes, the main application of nanoplex is multiplexing. For this, multiple ternary complexes are now preformed in separate tubes, then combined and used for a one-step immunofluorescence. Now, however, the signal can be removed after imaging, and the sample is ready for additional cycles with more ternary complexes. Because the signal is erased in each of the imaging cycles, the same channels can be used again and again in the following cycles. So what's the potential of this method? Here, the authors used a single channel in STAT mode in 8 consecutive cycles to get an 8-plex STAT image. Such a single channel setup might, for example, be advantageous if specific channel is preferred for a given imaging modality. This preferred channel can then be used again and again and again in the following cycles. If the imaging modality is less demanding, as confocal imaging here, for example, one can even image several channels in parallel. Here, for example, a 21-plex image was obtained by imaging 3 markers each in 7 consecutive cycles. As a third example, I would like to show smart secondaries conjugated to DNA oligos. Based on such tools, the groups of Ralf Jungmann and Jörg Beversdorf independently developed similar technologies termed SumPaint and FlashPaint, respectively, which are both highly sophisticated multi-cycle DNA paint setups. I'd like to summarize the Jungmann paper in a nutshell. So using smart secondaries conjugated to DNA docking strands, the authors managed to image 30 targets on a single sample with 5 nanometer resolutions in 3 dimensions. This means that you can basically zoom in on an interesting image area and even focus on a single synapse to see the relative localization of various synaptic marker proteins at a really amazing resolution. We are really happy to say that our smart secondaries contributed to these breakthrough works. I would like to end with a short summary. So, smart secondaries are nanotech's species and isotype-specific secondary single-domain antibodies. They are small, monovalent, and high-affinity IgG binders that allow for one-step immunofluorescence and species-independent stainings with multiple primary antibodies. With that, they offer virtually unlimited multiplexing potential in various applications, including nanoplex, DNA-plex, DNA paint, and other imaging modalities. Of course, I'd like to mention our collaborators as well as external funding and support. Thank you.
Thank you, Stefan. I should have said at the beginning our vendors are going to be here also tomorrow, so please reach out to them, talk to them, and get informed on their wonderful technologies. Our next speaker is Josiah Passmore, who will be presenting Visualize Bio. And his talk is entitled Automated Deformation-Free Expansion Microscopy. Josiah, the stage is yours.
Thank you. Thanks. Yeah, I'm Josiah. I'm one of the co-founders of Visualize Bio. And it's amazing to be here because us as a company, we are focused on expansion microscopy. So it's been great to be able to engage with all of the experts of the field. And the feedback so far has been super positive. So feel free to ask more complicated questions as well. So a bit about us. So we are a brand new startup. So we're actually less than a year old. We started end of last year. And we're based in Amsterdam in the Netherlands, and we're a spin-off company of Lucas Kapitein's lab at Utrecht University. And in the Kapitein lab, we've had a lot of experience in expansion microscopy over the last 10 or so years, really on terms of recipe development and method development with T-Rex, GelMap, but also experience with a lot of different sample types, but also teaching expansion to a lot of new users, interacting with other kind of diagnosticians and pathologists. And it's through all of that experience then we realized that there's kind of a bit of a need for a bit of simplification and democratization of expansion microscopy. Because as we're talking to these new users, especially in kind of pathology applications, we find that some of these questions about anisotropy, ease of use of the technique, sample variation, labeling, are really at the forefront of these people's minds. And if we're really talking about kind of this standardization, then really as expansion moves a bit towards more of these clinical applications, then we find that this is a real need, especially if you're trying to kind of convince a pathologist that the best way to perform expansion is to scoop out the gel from a well plate using a paintbrush, then all of this standardization that they're used to is not necessarily appealing to them. So we want to provide solutions for expansion microscopy users, both new users but also experienced ones. And these experienced users such as you, it's really important for us to work together with you lot in the community in order to collaborate and really move the expansion field forward. So what do we offer? Well, we kind of focus on three main different streams. So the first one is expansion specific consumables, just to make day-to-day life easier for people who do expansion. The second one is on expansion quality control, so increasing a bit of this reliability and quality control of the technique. And finally, we're really focusing on the automation of expansion, so really going from end-to-end automation of the whole process. So firstly, this one is a bit more for the PhD students and the postdocs that actually do the work of expansion. And this is a bit of just a quite simple thing that we thought of that we think is actually quite valuable. And it's basically just a well plate that has two separate compartments in each well to make your life easier, because the separation between these wells is a partition that lets your liquid through, but not your gel. So if you're used to washing your gels in well plates and trying to stick an aspirator tip in the well, you probably know the sound of a gel being sucked up into the aspirator. I know I do. But this makes your life a little bit easier because you can just put the aspirator tip in this external well thing, suck out your liquid without touching your gel. So just making the day-to-day life a bit easier. They're manufactured from clear polystyrene, same as a standard well plate, and we have 6 and 12 well formats as well. And the second thing, you kind of heard a bit about it from Lucas yesterday, is GelMap. And GelMap is a fluorescent reference grid assembly that gets introduced into your hydrogel. And as Lucas really kind of highlighted yesterday, it provides a way to intrinsically calibrate your gel expansion. So having this intrinsic ruler all the way throughout your gel, but also map different deformations in samples, especially at the longer length scales, where you really kind of occur from mounting the sample, for example, where stretching and shearing. But it also is quite handy because it has this coordinate system embedded in it to allow you to do a bit easier correlative experiments. So all of those very nice pre and post correlation images, I know how annoying it is to try and find back the same cell in your expanded gel, so this makes it a little bit easier. As I mentioned for the deformation, so we can show using the GelMap grid that you really get a lot of differences across your sample plane. As one example here, you can see regions that are a little bit more deformed than others. And we can also do this even in tissue slices. So here you see a GelMap grid imprinted onto a tissue slice. And I hope you can appreciate if we overlay the ground truth that you can see how warped it is at the macroscopic level. And we are able to then use this ground truth to correct for it in order to show then be a bit more quality controlled in your experiments. And this kind of the sample navigation aspect of GelMap is quite cool because it allowed us to also do correlative live expansion microscopy, CLEX-M, using GelMap. So what I did here is we imaged a cell going through mitosis, and we fixed the cell on the stage. We located the cell back using the GelMap grid very easily, expanded this piece of gel, located the cell back very easily, and then, we can now see everything with the very high resolution provided by expansion. So finally, kind of the main thing that we are here for. So we're looking at automation of expansion. So when I talk about automated sample processing, then the automation comes with all of those benefits, from maximizing your efficiency and your reliability of your workflows, which saves ultimately time and resources by not having to physically be at the lab all the time, and just increasing throughput by parallelizing your experiments. So what is it? How does it work? So we call it gel cast. It casts a gel. And you can see that around the back you can load in some of the more common your buffers. And at the side we have a well plate entry area. So we can, how it works is you would present your sample in this well plate format, the same well plates that I previously showed. And we want it to be able to be compatible with many different sample types. So each of these well plates can have a little sample reservoir at the bottom of each well to make sure that we can accommodate many different sample types. And we also want it to actually be recipe agnostic. So as we can load buffers in through the back, but we can also load gelation solution in through the side. And because we can control the temperature really from 4 degrees up to 100, we can cover a lot of different expansion recipes, both from proteolytic digestion, but also denaturation. And also we are looking at how we can do 4x, 10x, and even higher expansion factors as well. Of course, it comes with GelMap, so you can include GelMap in the sample workflow because we just process the samples exactly the same as normal expansion. And we really talk about fully automated in terms of also gelation. So we really can cover the sample entry to the anchoring, the gelation, the homogenization, and the full expansion, and even also the sample mounting. So we really are this end-to-end machine that can go from your sample to the microscope. And just a bit of a cartoon to show we really mean full automation in the gelation. So you would present your cells on a GelMap grid, expand it up, and then also use this grid to then do the correction and the intrinsic calibration to really get this molecular level detail. So ultimately, we want to be XM made easy. So we have easier manual workflows for those of you that are used to the technique already. We have standardized automated workflows, especially useful if you want to make the technique a bit more automated, but really useful for newer users and as expansion moves towards clinical applications, and all with the quality control that comes with GelMap. So if you're interested, then definitely come and talk to us. We have a booth over there, and let us know any feedback because we'd really love to hear it. Thanks.
Thank you, Josiah. So our next speaker is Hugo Damstra, who is actually also a co-founder of Visualize Bio, and he's part of E11 Bio, and his talk is entitled Enabling Scalable Optical Connectomics Using Expansion Microscopy. Hugo, the stage is yours.
Oh. What's the pointer? Oh, perfect. Thank you. I want to thank also the organizers for this fantastic meeting so far. It has been great to also see some of the faces behind the names you know so well from the papers that we read. And I'm going to be talking about the work that we're doing at E11 Bio. And of course, Sven already gave a great introduction about some of the details. And I'm going to try to add a bit of the high-level context to that, which is about enabling scalable optical connectomics using expansion microscopy. So a little bit about E11 Bio. We are a non-profit research organization, and we are on a mission. And that mission is to enable scalable connectomics in mammals, first by starting with the mouse, and ultimately to scale this up to humans. And we believe this is important because having the wiring diagram of the brain, and especially if we can get this routinely, can inform us about how the brain computes information, but it can also inform us and guide drug development for therapeutics, for BCI, and could inform other fields as well, such as neuro AI and brain simulation. So how do we attempt to get there? What we try to do is to develop and crucially also integrate 3 enabling technologies into one single technology platform, and that platform we call PRISM. It consists of 3 pillars: protein barcoding, expansion microscopy readout, and then AI neuron tracing. And when we say we want to reach, how we plan to reach this, we have some targets for ourselves. So one is that we want to target a 100-fold cost reduction to get the mouse connectome. We want to be able to add molecular annotation to this wiring diagram. And that's, of course, one of the key advantages of using expansion microscopy, where we can also read out specific proteins. And then finally, we want to broadly disseminate our methods, which is in line with our non-profit organizational structure, and really share our protocols and have, hopefully, the community work with this technique. So when preparing for this talk, I thought it would be nice to also take a step back and think about what has changed. Why now? Why do we think we can do this with expansion microscopy? And when I think about the past 10 years, which has made this possible, it comes down to 3 factors. One is, of course, increasing the expansion factor that we needed to get the resolution required to resolve individual synapses. And there has been great work done on new chemistries, for example, X10, X20, but also iterative methods that have been also adopted by other groups to make this a reality. The second is improved anchoring and homogenization, which we need to, of course, be able to expand brains relatively uniformly. Work comes to mind from the Chung lab that introduced also the chemical denaturation and the PFAA anchoring. And then finally, the readout of ultrastructure. And this is something that I'm personally also extremely excited about every time I talk about expansion microscopy. And also when I was back in Utrecht and we worked on T-Rex, and first seeing the ultrastructure through the scope and like this enormous wealth of information that's in these stains is really fascinating. And when we think about these three things, in the context of connectomics, they sort of came together, of course, last year, and we have seen this also from the talk from the Denzel group and the work from Mota Vakoli with Lycon, which I think is a great example of which conclusively shows basically that we can get dense connectomic segmentations from expansion microscopy data. And we at E11 were very excited, of course, when we saw this, and we quickly worked to integrate this also into our pipeline by integrating an iterative recipe that goes also up to over 16-fold expansion. And here you can see an example of the hippocampus of a mouse brain. And then we can segment this volume, and you can see that we can get both larger structures such as dendrites, but also even the tiny spines and spine necks from the dendrites reconstructed. So that's fantastic. But now when we think about scaling up connectomics, we face two challenges. One, and that is something that Sven explained before me, is that tracing neurons is very expensive. And the reason for this is mainly to do with the proofreading, which is a laborious and mostly manual process where you have to go through and see what these segmentation algorithms spit out and confirm that it's true. But the second one is a bit more practical, and it has to do with the fact that both for EM, which has been the gold standard for connectomics, but also XM, we cannot image an arbitrarily large volume at once. So we have to subsection our volume. And we can illustrate that the reconstruction is sensitive to discontinuity and section loss by looking at this map of the San Francisco Bay Area rapid transport. Now, if we start to subsection this map and suddenly along the process we accidentally miss a slice or we damage a sample, you see that when we rely only on grayscale morphology, it becomes quite tricky to see which line you have to take to get from San Francisco to Berkeley. And our concept is, what if we can literally inject more information into the brain and color code each neuron? That would help you to correct tracing errors, but also to bridge spatial gaps that may occur throughout the process. So Sven already showed some details about this, so a quick recap of what we did. Inspired by previous approaches such as Brainbow, we devised a combinatorial viral delivery strategy to expand the color space to over 100,000 unique cell-filling protein barcodes. Next, we read it out with expansion microscopy to assign individual barcodes to each of these neurons that you can see here. And we also included molecular markers to assign pre- and post-synapses for molecular context. And then the final part of this platform is that we developed machine learning algorithms that use this information to improve the segmentation accuracy almost tenfold if we look at compared to grayscale alone if we add the color information, and also enables reconstruction across the spatial gaps. And so preprint is out. Please check it out. It's a very exciting work. And what we have done here is really look at basically prove this concept of the protein barcodes. And we relied also on the protein barcodes to segment out our volumes and to reconstruct the cells. And in this volume, about 50% of the cells is labeled. And that brings us back to the beginning of the talk where we talked about the entire morphology, which we can, of course, also read out using expansion microscopy. And so the next step for us is to integrate the morphological readout into this PRISM pipeline. And I will show an example of what that looks like. Here you can see a zoom in of a barcoded cell, and we're moving through the volume, and you can see that the barcode is very nicely self-filling, so it also fills in the spines, and you can see where this neuron is trafficking. And that information is now, there's basically more information to do your segmentation on and to correct errors afterwards. And I wanted to to end with a bit of a teaser to see where we're going with this approach, and that is to scale in basically two respects. One is, of course, the actual volume that we're trying to map, and the other one is to add now more barcodes to the mix, and we're actively developing on this front also on the expansion part to really get to like a good recipe and procedures and protocols that we can get high expansion factors, good morphological readout, and good post-expansion labeling, which is what we're using here. And then I want to mention we are also hiring as an organization. So for this crowd, I've highlighted the spatial omics scientist position. So if you're interested about expansion microscopy, signal amplification, particularly in gels, please reach out or come find me after the talk. And we're also hiring for some other roles. So if you know people that could be suitable for that, then please let me know. You can also scan the QR code for that. And then finally, I would like to acknowledge the E11 Bio core team, which you can see here, the collaborators that were involved with the PRISM CA3 pilot study that I talked about. And then finally, also here, please follow our work, join our mailing list, and then you'll be the first to know of any new things that we are working on and whenever we have exciting stuff to share. And with that, thank you all.
Thank you very much, Hugo. So our next speaker is Daniel von Wangenheim from 3i Intelligent Imaging Innovations. His talk is entitled Turnkey Light Sheet and Super Resolution Microscope Systems for Expansion Microscopy. Daniel, the stage is yours.
Thank you. Can you hear me? Okay. Thank you very much. So I'm excited to represent 3i here and show you some cutting edge technology from our company, which is around already over 30 years. We're celebrating this year, and we have customers around the globe. We have a broad portfolio on systems. So we have spinning disk confocal, multiphoton, TIRF. And what we like to do is we like to implement new technology very quickly in a system that is then a turnkey system. So I think one of our most famous system is the lattice light sheet that was sub-licensed to 3i, and we immediately made it available and a turnkey system that you can buy. But we have other technologies like adaptive optics in spinning disk confocal. We are expert on computer-generated holography and ST4 and a lot of other cutting-edge technology. And I would like to highlight today on light sheet microscopy and spinning disk super resolution confocal. And for this, I show this one example. That's a spinning disk confocal, and it's a plan scope. And what makes it a plan scope is that we turned the microscope on the side. And then what you can then do is you let the plant grow upright, and then so I'm personally proud of this project because it all started at IST Austria when I was a postdoc in Jiří Frimel's lab, and we published a vertical confocal system. Now at 3i we took it to the next level. We implemented tracking in our software, even montage trackings. So this is a tile scan. And we've added a lighting and some other gimmicks. So it's a planned scope package now. One example of what we do at 3i. And it's a spinning disk confocal. We are selling mostly the Yokogawa spinning disk. And this also comes with a super resolution unit. And this boosts optically the resolution up to 120 nanometer. And with this, and in combination with expanded tissues, you can then reach resolutions that are near electron microscopy, so molecular structures in a light microscope. But I think So now I'd like to switch to light sheet microscopy. And what you see here is our 3 light sheets. Lattice light sheet for very small samples, Mariana's light sheet for small embryos, and then we have the cleared tissue light sheet. So different scales. But I think the nice thing about expansion microscopy is it could be used on any of those microscopes. It all depends on your sample. What you expand? Is it a cell or is it an entire organ? So I would like to focus on cleared tissue light sheet and present what this technology is in our microscope. We're using axially swept light sheet microscopy. We licensed it from Reto Fjolka and Kevin Dean. So this remote focuses a very thin beam across the sample, and we synchronize this with the readout of the camera. And by this, you can create very thin optical sections. So let me show you this in this animation here. Imagine you have a mouse brain in the microscope. We have excitation objectives that we designed. So they are optically corrected to have its best focus in high refractive index media. So to create a very nice optical section, 3 microns thin. And this is as large as maximum 10 millimeter. So you get an optical section 3 microns thin in 10 millimeter. And this then allows you to capture a whole mouse brain in one shot at isotropic resolution. Because here the pixel size is even larger than 3 microns. So you have true isotropic resolution in this image. And the nice thing about this microscope is it's very flexible in terms of magnification. So this is the largest we can get, but we can zoom in to a certain extent. And also it combines with different objectives, so there's a lot of flexibility. And flexibility also with the sample chambers. We have a range of sample chambers that you just exchange smaller volumes for small samples and up to a liter if you have a very big sample, like a whole mouse in this example. This is a whole mouse imaged at a lower magnification, and then I mentioned the zoom range. So here I'm now zooming in up to 6.5x, still not the highest zoom we can do, but here already you can appreciate axons and see dendrites and all. So this is what the raw data looks like. So each square here is a field of view, a 10 millimeter field of view. And then what I did is I imaged the brain at a higher magnification. And now I'd like to highlight one thing. If you want to image these large samples, yes, so imagine you've imaged this whole mouse at the resolution we imaged the mouse brain at 6.5x, you would end up with hundreds of terabytes. And one thing is on our side also to provide solutions for handling this amount of data. So in the future directions we go, that we try to help on data processing, with AI-driven pipelines for, let's say, you image the whole at a lower magnification and then find spots that you then automatically detect and zoom in and image at higher magnification. We also work on more efficient file formats, low level like, and then we also have Allen Brain Atlas. So this is not a future direction, this is implemented. So you can map your brain in our software, map the brain on any atlas, and then overlay it with your data to see where is the region in the brain. And then I have some new exciting technology to announce. So we now licensed ExaSpim from Adam Glaser. So ExaSpim is a bit like our microscope, but with a massive objective and a massive 150 megapixel camera. So this just scales up on the field of view and resolution you can get. So that's exciting. No? And my final slide, by the way. Here's my summary. I hope I could show you some exciting things ongoing at 3i. So thank you.
Thank you, Daniel. Our last speaker of this session is Tomasz Gereschej from CSR Biotech, a company that joined our event just on time to support our prizes, actually, awards, the Poster Award, the Media Award, and the Stage Award. So, Tomasz's talk is entitled Cell Expands Multimodal Super Resolution. Resolve your sample in any dimension.
All right, so I know I'm the last one before the main act, so just hang in there. And thank you for the organizers for accommodating us. So CSR Biotech, unlike many of the speakers at this conference, I feel like does need an introduction, so that's going to be my first priority. So our company originates in the group of Professor Liang-Yi Chen, who is a long-time expert in structured illumination microscopy, not just the hardware part, but also when it comes to deconvolution algorithms and the computational part and all that. And he and some students founded our company in 2019 and put the headquarters in Guangzhou, China. If you're not familiar, it's a small village of 50 million people, and it's also known as the Silicon Valley of China. Now, the reason why you haven't heard about this company probably is because we only opened the international branch in Heidelberg very recently, so that's where we are also located. Back in China, we have almost 150 people developing the technology, doing R&D, so pretty intense over there. And we build microscopes, so that's the profile of the company. And our main microscope is this one in the middle, the Machine Intelligent SIM, so Structured Illumination Microscope, that I will introduce, but also I will mention that we can now combine it with the Yokogawa spinning disk system on the same body, and also our own developed ODT, so label-free microscopy. And on top of that, we also make our own software, so for image acquisition, but also for image processing, that includes all kinds of deconvolution algorithms, denoising, a lot of that coming from the original inventor group. So SIM, or Structured Illumination Microscopy, in case you're not familiar, just a quick recap of what it does. So instead of illuminating a sample with point source or homogeneous light field, which is normally in microscopy, we actually illuminate it with a structured illumination. What that means is that we have a sinusoidal intensity pattern. And the reason, so why we would do that, is that when we image the sample like that, then the acquired image will contain higher frequency information from the sample. So in Fourier space, you basically get a bit more information from those areas that are not resolvable because of the diffraction limit. That's in one direction. We switch this illumination pattern in 3 different directions and then shift it 3 times to cover the entire picture. And at the end, we generate 9 pictures. And from that, we construct 1 super resolved image. So you can see here that the resolution in theory can double compared to the wide field imaging. And it's important to mention that this has nothing to do with AI or no kind of deep learning. This is math from 200 years ago, and it works very nicely. So on top of that, so this is the normal SIM principle, and what makes our platform special and why it's called MI-SIM, or Machine Intelligent SIM, is because there are also a bunch of deconvolution algorithms that are optionally in the software. So you can already see here on the right side that with our sparse deconvolution, you can find all those, all the details about it in the paper, but if you look for the technology, it's really easy to find, that you can bring down the resolution to 60 nanometers, still with a really high frame rate. So that is really the power of SIM, is to have high resolution, well, low number, and still high imaging rate, which makes it really adapted for live cell microscopy. So the main point is that we do live cell super resolution. The photo bleaching and also the phototoxicity is incredibly low, so you can do these measurements for several hours or even days on live cells. And on top of that, we have multiple modalities, so also 2D and 3D sim. We can talk about it at the booth, but also TURF for looking at membrane-bound proteins. So there's a theory, and let's look at some pictures. So just to reiterate, long-term super-resolution imaging live cells. So here what you can see is a multi-channel acquisition. So we have the ER and the mitochondria labeled in different channels. And this kind of interaction, this dynamics is very fast. So you can see on the time here, this is a video of, I think, like 15 minutes with really high frame rates. So you can see how the fast dynamic interactions happen. And at the same time, we have a really nice field of view. So this is 100 by 100 microns super resolved picture that is, of course, very useful for axons, that you can see the entire neuron, but still the mitochondria being trafficked in there is also very clearly visible with high resolution. So this is just a few examples of the actual SIM microscopy. And then when we combine it with spinning disk, then something amazing happens. So there has already been some talk of kidney slices at this conference. So this is one example of that. With spinning disk, of course, you can take a really nice field of view with still good resolution and get an overview of the sample. But sometimes you zoom on certain structures and you wish, if only I could resolve this a bit better. And that's exactly what you can do if on the other side of the scope you have the MiSim, because then you can just acquire a picture there and immediately get a higher resolution image that can resolve relevant structures, such as these podocyte food processes here, that would not be resolvable just simply with a spinning disk. And on top of that, our spinning disk also includes the SACD modality, which is a kind of image processing algorithm where we take 20 frames of one image to increase the resolution to 120 nanometers even, just based on fluorescent fluctuations and how they can be processed. So another nice application, of course, is when you have really big sizes of, for example, this brain slice, which is 10 millimeters in diameter, and with the SACD, you can see that the resolution is quite a lot improved. And if you still cannot resolve it, then on top of that comes the SIM imaging. So the last modality that I still want to mention, because I found it really interesting that the context of where things happen has come up a lot at this conference as well. And of course, we know that when we do a fluorescent labeling, then that is great for imaging, but it limits us. So the moment we do a labeling, we only see what the label shows. However, we also know that a lot of the times there are other important things happening around those labels. And this is exactly when combining super resolution live cell imaging with the ODT, so this is a phase contrast-based tomography, optical density tomography, we can also see non-labeled parts. And in that sense, you don't only get what you labeled for, but also all the other information, which is very important because, well, in science, of course, the exciting things happen when we look at something that we didn't expect. And if we didn't label it, then we didn't expect it. And then there is one really nice example of this, for example, where you can see protein aggregates that were labeled in the SIM image, but also you can see all the context around it thanks to the ODT. And the good thing is also that we can have an event-based triggering in the software, so you can look at the ODT picture, you can do a long-term imaging, and when some kind of structure emerges, then we can do the SIM imaging just specifically there to even avoid more photobleaching and phototoxicity. So it's a very modular system. We have a lot of exciting developments coming up, and we have a booth right out there. So we're a new company, but we have microscopes going over Europe and the US. So if your lab or your core facility would like to see a demo of these microscopes, then we are really happy to ship it. If you're located in a nice beach town, even better. If not, we can still do it. So just come to the booth and talk to us. Thank you very much.
Thank you very much, Thomas. So guys, please do visit the vendors, convince them to sponsor future expansion events. And now we are entering the last presidential plenary talk. And I will give the word to Silvio. Let's applaud once again our sponsors.
Thank you very much. So we are coming to the last event of our what is it, third day of the symposium. I mean, I will give the word then to the chair of this session at Boyden.
Okay. Let's see. And he bring his laptop. All right. So it's a great pleasure to introduce and welcome our keynote and presidentially plenary lecturer, Stefan Hell. Stefan Hell was born in Arad, a medium-sized, ethnically diverse city in the western part of Romania. He moved with his family to Germany at age 15, settling in the region of the university city of Heidelberg, where he obtained his diploma in physics and then continued toward his PhD degree, already working on the problem of how to improve the resolution of microscopy in a fundamental way. He joined Ernst Stelzer at EMBL in 1991, and within a few years, conceived of and implemented the 4-pi microscope, which improves resolution dramatically along z-axis. Due to issues with finding funding in Germany for his ideas, which were far too advanced for their time, moved to Turku, Finland, where he conceived within a few months the STED principle. The full implementations made in Göttingen, where he was recruited in 1996, of course rewrote the story of optical microscopy and revolutionized the thinking about resolution, which of course had been thought of as dogma since the 1800s. And of course, STED microscopy, as you've heard, has been introduced and has been very powerful in biology, with higher and higher resolution and entire fields of optics and photophysics that result. So of course, a plethora of prizes and acknowledgements followed, including the Kavli Prize and of course, ultimately, the Nobel Prize in 2014. Stefan has, of course, been renowned not just in academia and science, but also in business, for example, winning the Ernst & Young Entrepreneur of the Year prize for work with his company, Leterrier, who you've heard about with the work on commercializing and bringing forth STED and other tools. And of course, the story has not stopped. Stefan continues to accelerate in research towards ever-increasing resolution. In recent years, he's introduced MinFlux and MinSTED, which bring the resolution in live cells down to angstrom level. And so, yeah, we look greatly to your lecture today, and thank you so much for being here.
Thank you very much. I need a mouse pointer or something, so. Okay. Thank you very much, Ed. It's a great honor to be introduced by you. Thank you. It's a pleasure to be here, Silvio, Ali. It's an invitation that I followed, of course, with very much enthusiasm. Also because, you know, I mean, the field of expansion microscopy has developed so tremendously. And in my view, it's clearly a very, very, very important field, and definitely something that wasn't expected at the time, and now is so powerful and will have a huge impact in biology, I'm sure. So I'm going to speak about a very simple problem, as a matter of fact. I'm going to speak about how to find the position of a fluorescent molecule with maximum accuracy, precision, and speed in the focal plane of a microscope. Now, when people think about localization, as it is called, then they usually think of having a wide field illumination. So if you want to find out the position of the molecule here in the focal plane of the microscope, usually what people take is a picture of the fluorescence. That is, of course, a diffraction blob, and this diffraction blob that we get on a camera scales with the wavelengths. Now, the narrative goes like this. It's no problem. We can still find out where the molecule is, although this blob is quite blurred. The only thing we need to do is we need to calculate the centroid of this diffraction blob. And once we have the centroid of this diffraction blob, we know where the molecule is, because that tightly correlates in space with the position of the molecule on the camera, so by simple geometrical optics. Now, the precision with which we can find out that position scales with the wavelength. So the longer the wavelength, the less precise it is, but fortunately, inversely with the square root of the number of photons detected by the camera. Now, it's quite obvious that this way of localizing individual molecules fundamentally depends on the maximization of the number of detected fluorescent photons. And this comes, of course, with in my view, major drawbacks, because, you know, if you want to get a very high precision, so a small sigma, very small uncertainty, we need to get a high number n. So that takes a lot of time because a fluorophore emits only a single photon at a given point in time, and so you need to wait. Then there is a problem with bleaching and blinking. In fact, blinking, in my view, is even worse than bleaching because if it's gone, you know it's gone. But if it blinks, you know, the fluorescence rate is low. It goes somewhere you cannot follow it. And that's an obvious problem. But there is a problem which, in my view, is totally underestimated, perhaps for good reasons. If the molecule emits or absorbs, it behaves like a dipole because it has a transition dipole, meaning that if the molecule is rendered like this, it will never ever be able to emit in this direction or in that direction. It will always emit in this direction. And as a result, the cone of detection of the objective lens will not be illuminated uniformly. So there is a problem, so to speak, a phase problem, as a matter of fact, that leads to a shift of the position of the molecule on the camera. So while you might get a very high precision, so for a nanometer or even less, after having collected, I don't know, 10,000 of photons, you're still in the wrong place. And you're in the wrong place off easily by 10 nanometers, 50 nanometers, even 100 nanometers, especially if the molecule is slightly out of the focal plane, dislocated in the Z position. It's very hard to find that out because if you dislocate the molecule by 200 nanometers in the Z direction, it hardly changes its brightness. And so there is a big problem with that. Now, usually people discuss that away. They say, oh, molecules rotate freely. But believe me, there's no such thing as free lunch. And there's no such thing as free rotation because the fluorescent molecule is attached to something and cannot occupy, the whole orientational space uniformly. There's no way. So, as you know, most of you will know, I've come up with a different way of localizing individual molecules at the expense of not being able to eliminate the whole field of view at the same time. And it's based on an idea that I called coordinate targeting, and in fact, it's another word of saying that I'm making a coordinate with the zillions of photons that come out of a laser. So a laser produces many photons. There is a very, very high photon rate. And especially if you convert the laser beam with a phase plate that induces a phase shift of pi on the opposing parts of the aperture, then you get destructive interference in the center. So in other words, you get a donut which has a perfect zero here, intensity zero here in the center. And this position is perfectly defined and can be easily measured because there's zillions of photons out there. There's no shortage of photons. You always need many photons to make a position optically, because of Heisenberg's uncertainty principle. But here, we don't require the fluorescence emission. We don't require the fluorophore to provide those photons. Those photons are provided by the laser. There are zillions of photons coming out from the laser. So we have defined this position with ultimate precision. And not only that, we can also shift it around with perfect precision because there's a beam deflector over here. Could be a galvanometer deflector, could be an electro-optic deflector. And because of the magnification factor, you can have angstrom precision of positioning this donut in a sample plane. Now, the whole localization process boils down to finding the unknown position of the fluorophore with respect to the perfectly known position of the donut zero, perfectly defined position of the donut zero. So how can we find that out? It's actually very easy, because if the molecule is here, it will produce fluorescent photons because it's excited. So this is green light for exciting the molecule. And as a result, the detector here will count fluorescent photons. Please note, there is no camera. It's a confocal detector. So in essence, this is a confocal microscope whose beam has been converted into a donut. That's it. Now, if the two coincide in space for some reason, then there won't be any excitation, hence there won't be any detection. And as a result, we know exactly where the molecule is, because we know where the donut is. And if there is no signal emitted by the fluorophore, then we know the fluorophore must coincide with the position of the donut, then we know exactly where it is. So in other words, if it's slightly off and the rate is low, the emission rate is low, it cannot be far away from the donut zero. So the brightness of the die or the fluorophore will tell us actually how far we are from the donut zero. Now, this concept is very powerful at finding the position of the molecule with a minimal number of fluorescent photons. And in order to make this vividly clear, I've decided to introduce a little demon. Now, this little demon has the capability, humans don't have that, but this little demon has the capability of instantly placing the donut zero on top of the molecule once the molecule starts moving, within an attosecond, okay? Fluorescent lifetime is a nanosecond. So within an attosecond, once the molecule goes away, the demon puts a donut zero on top of the molecule. Now, in that case, there won't be any fluorescence detection here. The molecule will not emit. Yet, the demon can trace the position of the molecule with arbitrary precision, picometer if you want, yeah? Because it knows where the molecule is. Based on the absence of fluorescence emission, the molecule's position must coincide with the known position of the donut zero. Okay, now this concept, at least in the thought experiment, tells us that in principle, at least in the limiting case of a demon, you can come up with a localization methodology that is just the opposite of what you've heard before. It does not maximize the number of photons, but in the limiting case, doesn't require any fluorescence emission. So zero fluorescence emission. Not only that, as a matter of fact, the dependence on the wavelengths has gone. If you do the normal localization, you've seen it. I mean, it scales with the wavelengths of emission, with lambda. But here, the wavelengths dependence is gone. And if you ask me for a hand-waving argument, why is that? Because if you're trained as a physicist, as I am, then you're being told that if you have freely propagating waves, light waves, everything must scale with the wavelengths. But here, the wavelengths dependence is gone. So why is that? Because, you know, in essence, you can say the molecule is a point, yeah, and the donut zero is a point. The zero, as such, is a point. So if you match 2 points, it doesn't really matter whether the donut is made of green light, yellow light, X-rays, microwaves, whatever, because it's just about that central point, which is a mathematical singularity. And so that's also a fundamental conceptual difference. And if you look at it, also the dipole orientation dependence has gone. So there is a strong reason to develop this concept. But unfortunately, I cannot hire a demon because there's no such thing. But what you can do is you can kind of try to mimic a demon with some electronics. For example, a controller. Now, if you have that controller will measure the number of counts that you get from the molecule and react accordingly. So if the molecule starts moving like this, yeah, we will get counts because it's impossible to place the donut zero just on top of the molecule within an attosecond or something of that order. There will be counts, and those counts can be used to find out how far the molecule is away from the known position of the donut zero. And then, of course, the position of the donut zero can be changed, accordingly adapted, and then the molecule tracked in this way. So in this way, of course, we can save fluorescent photons, not so much because the donut zero doesn't excite the molecule, it's because there is a residual distance actually between the donut zero and the fluorescent molecule, the measurement of which requires much fewer detected photons. So if you look at the equation that governs actually the precision of the localization in this min-flux concept, it scales now with this distance L, which you can loosely say it's, if you will, the average distance between the donut zero and the unknown position of the molecule. So the closer this match is, you can easily figure out if L is 0 because we have a demon, then here you have 0 and sigma is 0, meaning precision is infinite. Now this is quite different from what we had before, because before we had the scaling with the wavelengths. But now we've replaced the wavelengths, and you can do nothing about that, yeah? With L, with the distance L, that can be changed, can be made smaller, and if it's made smaller, the precision sigma goes up, because sigma goes down accordingly. So the better you implement this concept, the closer you come to the demon situation, the higher the precision will be. Now, in reality, at this stage of development, we can save about 100 times the number of detected photons as compared to a camera. And of course, we also reduce this problem of the orientation. And so these are very, very important factors, especially if you want to conquer dimensions that are below 10 nanometers, because in that case, it really matters. As I said, you can be very precise with the camera localization, but you might be easily off by 10, 20 nanometers without even knowing. Okay, so this concept can now be used to either have a fast localization. Fast means faster than with a camera. So you get, as a ballpark, a localization precision of about 1 nanometer within 5 milliseconds, or you can be more precise, meaning that if you do a type of localization, fluorescent, super resolution microscopy, you get for the same number of photons a 10 times higher precision, so of the order of 1 nanometer relatively easily, and less bias. Now, this is certainly one of the most popular cartoons in science, the walking of kinesin on microtubules. And how has this been found out? I think there is pioneering work, truly pioneering work by Ahmed Yildiz and Paul Selvin, Urbana-Champagne. And they published a seminal paper, actually, in 2004, which was based on attaching a fluorophore to this stalk, the kinesin, the dimer, or to one of the motor proteins here at the bottom. And they followed basically how that fluorescent molecule changed its position while kinesin was processing on the microtubule. Now, the spatial temporal resolution they got was 1 nanometer per 350 milliseconds. We are faster because we need fewer photons, so we got something like 1 nanometer per 5 milliseconds routinely, and can redo basically those experiments with the fluorophore attached, for example, to the stalk or to the motor domain, depending on what we want to do. I'm going to show an example with the fluorophore attached to the stalk, and then you would expect, of course, steps of 8 nanometers in size, because that's textbook knowledge. In fact, due to the high spatial temporal resolution, we could make out sub-steps that happen, of course, if this stepping takes place, so 4 nanometer sub-steps. And those sub-steps, they were measured actually with beads attached to the kinesin, which is kind of invasive. But here, with this minimally invasive method, where you just attach a fluorophore, you can detect them well and you can trust your results. So here are these 4 nanometer sub-steps, very vividly well depicted and measured, and this is the distribution of steps. So I don't want to bore you with kinesin biology, and I'm not a kinesin person either. But the point that I'm making here is that Paul Selvin's experiments and Ahmed Yildiz's experiments required dilution of ATP. They couldn't work with physiological ATP because it would have moved too quickly. Yeah, it requires a single ATP per step. And so they diluted ATP 100-fold. But we can go up to physiological ATP concentration of the order 1 millimolar ATP concentration because we are fast enough. We can follow the fast movement of kinesin. So that's a big advantage. Now, encouraged by this finding that the method is fast enough to measure, say, physiologically relevant processes, and of course knowing that just looking at structure is now strongly complemented by expansion microscopy, we said, okay, we have to do something that others cannot do, namely to look into another protein, in this case, dynein. There's a lot of speculation about how dynein works, but this is actually the first work that looked into the stepping of dynein in a living cell. So this is a living neuron, and done with endogenous dynein. So dynein was tagged with a halo tag using a CRISPR-Cas methodology, and then attached, then linked with a fluorophore. And this has allowed us to observe individual steps of dynein directly in the axon of a living neuron. So for the first time, actually, with this minimally invasive method, people speculated that there is a so-called tug-of-war, like a stochastic process between dynein pulling towards to the nucleus and kinesin pulling towards the periphery of the cell. We have seen that very rarely, surprisingly rarely, but what we've seen, and this was not known before, because the method was simply not there, spatial temporal resolution was not there, is a quick change in direction. So on the time span of a single step. So obviously, there must be a regulated mechanism that leads to a change of direction. This is something that needs to be taken further. So I'm aware of the fact that several people now I can say it purchased a MinFlux microscope built here in Göttingen, and they apply now this MinFlux technology to the study of dynein and, of course, kinesin. And I think this MinFlux is going to have a big impact. It's probably the way to do motor protein studies with minimal invasion and with a very high spatial resolution. But what about the localization? At risk of boring you, yeah, this is just historical data that I'm showing you, but it's going to lead to the point that I will make towards the end. So that's why I'm showing you this. So published back in Nature Methods, we published these nuclear pores. This is essentially a 2D recording, which at the time was quite impressive because you can associate individual fluorophores with located here with sub-nanometer precision in median, so 0.9 nanometers, and you can associate those fluorophores with individual proteins. It's clearly much better than STAT, and definitely by far better than confocal. So there is of course a factor of about two orders of magnitude or so 100 between the two, and it's going down. I'm also very proud, and I'm pleased that Ed mentioned that. I felt that you cannot have an impact unless you commercialize this, and this is why I'm really proud that, especially with major contributions from former people in my lab, there is now a system out there that looks like a confocal system. In fact, it is a confocal system with the option of having the excitation beam converted into a donut. And this system gives you confocal overview, of course, it gives you STAT, and also gives you the, say, molecular scale resolution that you can have with MinFlux. But there's something else it does. Because it's confocal, and because it's a pinhole and not a camera, you don't need turf. Yeah, you can, of course, venture deeper into a tissue. Yeah, and then go into a tissue and then detect individual fluorophores, locate them, and take a picture. So this is now done with fixed cells, and I know expansion microscopy makes beautiful images with fixed cells, but this can be taken further into living cells. We have first indications, in fact we've published it, that we can see spines also in living cells, and basically down to about 80 micrometer. I think we just scratched the surface here because we haven't used any active adaptive optics corrections of the donut or so, which of course makes a lot of sense. In this particular experiment, which makes a lot of sense if you look deeper down, because there will be spherical aberrations, there will be all kinds of things. So I believe, as I said, we're just scratching the surface, and this will, in a few years, or maybe even earlier, be much, much better. So I'm shifting gears here, and I'm coming back to one of the pet problems of my life, if you wish, how to separate actually two fluorophores that are close. Now, let's be very close, closer than 10 nanometers. You might know there was a paper in Nature Methods basically saying that if you do destorm, any distance closer than 10 nanometers will be challenging to measure. And usually what biologists think of when they look into such small distances, I mean true small distances between two fluorophores, think of FRET. So let's do FRET. And FRET is a great technique. I don't want to play it down. It has huge contributions to cell biology and the life sciences in general. You know how FRET works. Well, there's a donor and there's an acceptor. You excite the donor, energy is transferred to the acceptor, and based on the efficiency of the transfer, which scales inversely with the distance, you can infer the distance. Okay, but there's a catch. The transfer heavily depends on the orientation of the molecules. If the molecules are oriented like this, the energy transfer doesn't really take place, and you think that they are far apart because the energy transfer is absent or heavily damped, but in fact they're just badly oriented. So it's inaccurate to determine distances using FRET. And if you look at the FRET literature, there's a lot of stuff out there, many papers that just tell you how to correct for this inaccuracy. So kappa square corrections and so on. So it tells a lot about the method, although it's of course very useful. I don't want to play it down. And of course it's limited in range. Below 2 nanometers, Doesn't really work because the 6 power scaling breaks down. And then beyond 8 nanometers, the energy transfer is too weak because of the 6 power scaling, of course, but there's hardly any transfer. So what about just resolving them? Yeah, just by measuring the coordinate. That is the most natural way of finding out the distance. You just locate this and you locate that, and that's it, and then you subtract the two. And that would give you added information because Fred just gives you distance, just a scalar. But if you have the position, you have vectorial information. You know, this is here, this is further up, not just the distance, or this is further down, not just the distance. You really know where they are. So that's the most natural way of resolving two molecules. Now, at the risk of sounding like a broken record, I've said basically in every talk I gave since, I don't know, 2005 or so, the key to separating fluorophores at subdiffraction scales is a state transition, an on-off state transition. Yeah, you make sure that you see this one when the other one is not seen, yeah, when the other one is not signaling, and vice versa. So in order to discern, not to locate, but to discern molecules, to make them visible as separate entities, at subdiffraction distance, you have to use a state transition, on-off state transition, that makes sure that they are not emitting at the same time. That's the basic physical ingredient in STAT. The role of the STAT beam, the red STAT beam, is to shut off molecules to make them invisible when there is some visibility at the center. It's the same in Palm Storm. You have to have something that switches on and off or activated, like in Palm. Or in Paint, you have something that you attach or it gets detached, yeah? So this is SOFI, the same thing. So in essence, without on-off, none of the so-called super-resolution methods would be able to separate two molecules at subdiffraction distances. Now, with decent min-flux, and that is the high-end min-flux also developed here in Göttingen, Steffen Saal actually took the initiative and said, let's prove that we can separate these distances that are typically in the Fred range, yeah, just by changing the distances between two fluorophores. And then if we change the distance between the two fluorophores, we can check out whether these distances are measurable, yeah, and we have some say variation of distances by using polyprolines. In fact, this polyproline experiment was done in the 60s by the people who kind of introduced Fred or developed Fred as a method, and this was a very, very heavily cited paper because it really put Fred on the map as a practical tool. And now we use activatable fluorophores. So at the time this one was emitting, the neighbor was not, like in PON, so no difference. So a heavily active, a fluorophore that is active when the other one is totally inert, yeah? And so you can keep them, there's no threat between them, yeah? Because the other one is inert and only one is emitting. Okay, and in this case, you can see distances ranging here in this experiment, 2.8 up to 11.4 nanometers. This is the Fred range, so it goes beyond the Fred range. And obviously, you can get here the right distance. In this case, it's 2.8 nanometers, as you would predict here on the basis of this polyproline. So it shows that the method actually works. Now, you can have them even closer. Yeah, so this is a protein, it's a homodimer actually, where there are two binding sites where you can attach two fluorophores, activatable fluorophores. The predicted distance is 11 angstrom, so 1.1 nanometer. It's a collaboration with an NMR person, actually quite famous NMR scientist here at the Max Planck in Göttingen. And we confirmed, yes, we can measure that distance. So 10.9 angstrom, 0.6 angstrom precision. Does it get smaller than that? Of course not, yeah, because the fluorophore itself has about the size of a nanometer. So if you do a method like DNA paint, where you replace the fluorophore and look at one and the other one is out there and goes away and makes space for the other one, of course you can have, to some extent, smaller distance, but it has its own limitation. Having this DNA hybridization has its problems. Here we just attach a fluorophore to the binding sites, and get the distance. Of course, you can measure smaller distances if they are in projection. Yeah, if they are in projection, and this is the case here, you have, say, 3.1 angstrom, 1 angstrom. Now, keep in mind, this 1 angstrom distance measured all optically, but in projection, the Euclidean distance, the 3D distance, never changes. So in all these measurements, it was confirmed that the distance, 3D distance, stays the same. Yeah, and as I said, we just don't look just at the distance, we see also the orientation. All done optically, all done with focused visible light. And having looked into this for 3 decades, of course, this is almost like a dream that has come true, getting actually to basically distance nothing. Yeah. So the highest possible resolution that you can imagine. I want to relate it here now with a different way of representing the structure. So this is a structure in a different way. This is the structure of the floor force, so to speak. And actually when we localize something like the centroid of emission, if you will, yeah, of the floor force. So because it's very small, yeah, it's much smaller than the extent of the floor force. But it's still useful, even though it's smaller than the molecule, because you can use, of course, minflux to detect conformational changes of proteins. So it's a method of looking directly into conformational changes of protein. In this case, parallel and antiparallel. And here the distance is 9.4 angstrom, and here it is, in this case, is 43.1 angstrom. Another example, not properly published, but why not? Why not showing it? So here, the steroid, you have the 2 floor force attached to different places, 10 angstrom, so 1 nanometer, 19 angstrom, so 2 nanometers. And I think this is useful. It will be useful for many things, because you can look into conformational changes of proteins. So basically, I could say now, work done, I retire. I'm not the youngest anymore. I'm not feeling old, but still, I mean, I could say I'm just going out and give talks about what I've done or what I haven't done and what I missed out and going to high schools. I don't want to do that, actually, because there was one thing that really bothered me. W. Myrna, actually, and Eric Bettig, with whom I shared the prize, they deferred to me giving the banquet speech at Stockholm. So on the 10th of December 2014, I went up and spoke to the people in the banquet and said, you know, the reason why we got the prize, if you boil it down to the essence, is that we incorporated an on-off transition. And that was the missing piece. And once you have that, yeah, then you can separate and the diffraction barrier is gone. Then I also alluded to the fact that Erwin Schrödinger, a famous physicist, said very famously, we will never ever play or see single molecules. That's ridiculous. But that was done actually by W. Myrna. He was the first to detect individual molecules. It's a central part of Eric Bettig's PALM methodology. So I said that. And I said that this on-off point, I made it several times, and as you may probably know, in all speeches I said, well, what if I'm wrong? Maybe there's still a way of separating even without an on-off transition. And if that is the case, at least I want to find it out myself. And so I'm going to challenge this notion, this basic tenet of the 2014 Nobel Prize of Super-Resolution Fluorescence Microscopy. Is the on-off really needed? Okay? Now, in the case you were not interested in what I've been saying, say, okay, I have better pictures with expansion, which I would understand. There is something new now. You should listen to this. Now, let's assume we have 2 fluorescent molecules emitting all the time. So there's no on-off transition. You don't need to have a switchable molecule. You don't need to have an activatable molecule, no stat beam. Just take the best fluorophore or the one that you like most. Any fluorophore could be placed here. So let's keep them on, and let's revisit the Rayleigh criterion again. We take a normal confocal microscope. The confocal microscope has an excitation spot that looks like this. And what do we do with it? We scan it over the 2 molecules that are sitting very close, much closer than the diffraction barrier. What will we get? We'll get a picture that looks like the excitation spot. In principle, it will be slightly oblong, of course, yes, because the molecules are slightly apart, but it's hard to distinguish it from the situation where they sit in the same place. Sit in the same place, you get something very similar, and if you were to make a subtraction between the 2, yeah, just to see a difference would look like this. Yeah, so there's just basically noise. You cannot use this as information to find out that the molecules here are slightly further away than here. In fact, even if you took a profile, take a profile like this. So the orange one would be the one where they're slightly apart, and the red one is the one where they are not apart, sitting in the same place, with 1 million detected photons, which is quite a lot, yeah? Still within the noise band, yeah? You cannot tell them apart. So that's why people say it, and rightly so. There's no way of separating any molecules that are closer than the Fulvescent maximum of the excitation spot. Forget it, unless you play on-off, as instead, palm, storm, then you can separate. But without on-off, it doesn't work. Okay, but let's do the same thing now with the donut. Let's assume, for the moment, the two sit in the same place. Okay, and we scan with the donut across the molecules, the two molecules sitting in the same place. Well, we get an image of the two molecules sitting in the same place that looks like a donut, because that, fluorophore will kind of reproduce actually the shape of the excitation spot. In that case, the donut image here will have a central zero, because if the donut zero of the excitation beam coincides with the two molecules here, there won't be any signal. Zero, nada, nothing. But if the two molecules are slightly apart, no way of getting a zero here, because if the zero coincides with the first molecule, the second will emit. If it coincides with the second molecule, the first one will emit. If it's in between, both will emit, much less than before, but they will emit. So there's no way of getting a zero in here. In fact, if you subtract them, there's a measurable signal, because you measure against zero. In fact, if you take the profile, the orange profile here you see is clearly beyond the situation where they sit in the same place. Now, you can separate them. It's getting even better. If you look at it, you can separate the better, the closer they are. It's kind of, wow, what is this, It's easy to separate something that is just 10 nanometers apart as opposed to something that is 30 nanometers apart. It makes sense, because if it's closer together, then you have a better contrast. It's very clear, and so you are closer to the part of the noise band that is very weak, that is square root of n. So in other words, now, the diffraction barrier doesn't exist if you accept a field of view that is very small, like a single protein. It doesn't exist. It's a strong message, Is it true? Yes. So here, we actually took two fluorophores that have a certain distance, 35 nanometers down to about 9 nanometers, distance controlled by origamis attached to DNA, and here this is the measured distance, And it's confirmed, so you can measure it. So here in this case, it's 8.9, so say 9 nanometers apart, measured. Both were on. No switching, no buffer for D-storm or something, No stat, no photoactivation. Just one fluorophore. And why is this important? Well, if you need to switch to play on-off, you inherently cannot see all the molecules at the same time. You can see only one within the diffraction barrier. But you cannot see 2, 3, or 4, because the one that is off cannot be seen. But now, if you don't have to switch, you can have two, emitting at the same time. You can track them and still separate them. So here we move two fluorophores with the stage. So this is an artificial movement, in a Lissajous manner. But still, we could separate them 15 nanometers apart. In fact, easier than 32, 15 nanometers apart, and tracked at the same time. Now, I would say 2 years ago, 3 years ago, people said, oh, in order to track two molecules moving like this, I need a red one and a green one or a blue one. So I need two different colors, because otherwise I cannot separate them. There's no way if I want to see them constantly. No, you can do it with the same color. And red and green even has a problem. There will be fret. It will be a problem if they are getting close. Here, fret does not matter. It's totally irrelevant which of the molecule emits. Completely irrelevant. The only thing that matters is the excitability of the fluorophore. So this is a conceptual breakthrough. Not only works for 2, it also works for 3. So here we have arranged them in this manner. Here we have 4. I think it works up to maybe 7 or 8, because if you add the 9s or so, then the signal of the 9s will be perhaps confounded by the noise of the other 8 or so. So there will be a limit at some point. Another limit, of course, is the background. You need low background in order to make it work, but you've seen we've measured it here, and we were able to track them. And again, any fluorophore that emits. And it puts on the table a vision which I must say, totally excited about. I mean, there's plenty of data out there now and methods that give us the structure of a protein. There is X-ray, there is NMR, there is cryo-EM, fantastic alpha fold even. You can even predict it, yeah? Fantastic, yeah? But there's not many good methods out there. Perhaps Fred, but with which caveats, yeah? 2 points, but not more, that tell us how the protein works. So if you have a conformational change, yeah, it's very hard to detect. But now, we can envisage the following experiment. Just attach 4 or 5 or 6 fluorophores to a protein in its native environment, even in the cell, yeah, and watch it. What does it do? If it binds to calcium or whatever, yeah, to a drug, yeah, we could see what happens. Does it change its function? Does it not change its function? Look at perhaps at rates, yeah, changing rates, and so on. So that's something that we're totally excited about and keeps us up at night, so to speak. And then so in other words, MinFlux, using the zero, should allow us to find out how proteins actually do their job mechanically. There's something else. Why was the diffraction barrier broken for fluorescence? Some people might think because fluorescence is so important for the life science. No, I can tell you this is not the case. When I looked into the problem, there was late 80s. The reason why I was convinced at some point that it would be possible to break the diffraction barrier in fluorescence microscopy is because I realized fluorophores offer something that metals and glass or whatever, silicon doesn't offer, states that you can play with. In fact, the first paper where I claimed it will be possible to go beyond the diffraction barrier was the paper I did 3 months before STAT. I said the way to go is to look into the states. And a couple of weeks later, I realized, well, you just shut them off, yeah, with the stimulated emission, yeah. So the reason why the diffraction barrier was broken for fluorescence was not the usefulness of fluorescence. It's just the fact that you have states, that you can make a state transition, on-off state transition. But now, we don't need a state transition for separating those points. There is no need for a state transition. So it means it works for any point object that you can distinguish from background. Full stop. Could be reflection, could be Raman scattering, could be anything. So it's a new physical principle. And to make it clear how this separates actually from the standard localization that is being used in, I would say, a couple of thousand labs right now. I'd like to compare it. Now, this state, as you may know, is heavily, draws very much on car industry. There's Volkswagen around the corner, more or less, the headquarters of Volkswagen. And the nicest Volkswagen they make is this one. It's called Bugatti, but it's a Volkswagen, okay? And what's the idea behind this Bugatti? You add the number of valves, yeah? So you get a high torque. So 16 cylinders working at the same time. But it's very ineffective. So usually a car has 4, 6, 8, now 16. It doesn't really scale because it scales very badly. And that's the way it works with localization with a camera. You have to add many photons in order to get that precision. And then you're even in the wrong place in many cases. MinFlux is totally differently. It's like going to the electric engine, yeah? There is no valves, yeah? High torque right away. And in fact, if you look at it, the precision scales exponentially with the number of detected photons. You ask me now, why is it exponentially? Well, let's assume this is the diffraction barrier, 250 nanometers. It takes only one photon to find out if the molecule is in this half or in that half. Let's assume it's in this half. You have with one photon. Then you know, okay, doing the same game again. Which half? That one or that one? Maybe it's in that half. 2 photons. And then you find it out again with the next photon. With 20 photons, you're down at the Ångström level. Of course, in practice, it won't work like this, but it tells you how powerful the concept is. And if a concept is powerful, it can be engineered in such a way that the advantage it provides in practical situations is huge. With that, I'm coming actually to a very important slide. Stefan is actually here in the audience. I don't have big groups. But I have very clever people, very talented people, and I owe a lot to them. In this particular case, to Steffen and to Thomas. Jonas was absolutely instrumental to do this dine-in experiments. Otto, and of course also Lukas for the kinesin, and also in particular Jesse Matthias, who is now working, who works now for Iberian Instruments America. So with that, I'm coming to my very final slide as a message. I think, in my view, if you ask me, if you look back the last decades, yeah, after the prize was given to super resolution fluorescence, and if people ask me what are the biggest, say, conceptual breakthroughs that emerged since then, in my view, it's two. One is expansion, no doubt about that. That's something that was not really expected and works extremely well and has provided enormously important data and will provide in the future. Then, with some immodesty, I hope you apologize, MinFlux offers now an optical window, as we will see, to the function of proteins at the angstrom scale that should tell us how they work in their native environment. Thank you very much.
Thank you so much for that wonderful lecture. We have some time for some questions now from the audience. Okay, I see somebody coming. Yeah, please come to the microphones.
Thank you very much for the talk. I have actually listened to a lot of your talks on MinFlux, and I still don't get how it gets around the problem of the dipole orientation. Because if, let's say, you are not detecting it because it is emitting in a different way or it's not excited at all, then how do you differentiate that from the center of the donut?
Okay. I give you an explanation, hand-waving arguments that are confirmed by if you make simulations and so on. What MinFlux does, or should I put it that way, what normal localization microscopy on a camera does, it entirely relies on the emission. Entirely relies on the emission. So there is, you're completely reliant on where does the fluorescence goes. And this is why there is a fundamental dependence on the molecular orientation, especially if it is slightly out of the focal plane, because it's not so much the fact that you have this skewed amplitude, you have a phase change. And the phase change actually leads to heavy shift in position, easily up to 100 nanometer. This is not exaggerated. Jörg Enderlein has shown that, W. Myrna has shown that, we have shown that, and it can be confirmed experimentally. What MinFlux does is that I don't care about the emission. Yeah? I just count the number of photons that come out of the molecule. And if I'm doing it properly, I'm using circularly polarized light. I'm using circularly polarized light. That is kind of agnostic to the orientation of the molecule. And then I'm putting that circularly polarized light as close as possible to the molecule. And that dampens actually heavily the influence of the emission. Yeah? It doesn't go away completely, but it's reduced, this orientational factor is reduced by at least an order of magnitude. And that comes out both in simulation and also in the experiment.
Thank you. Thank you.
So this is why I think, yeah, I sound immodest. If you really want to go to a sub-10 nanometer level, careful with camera localization. Careful, because it's a serious systematic error. And in many cases, you won't have that free rotation, because I said there's no free lunch. It will be some bias. And if you really want to go to small dimensions, you cannot afford a bias. You're not, and it's misleading, because you think, oh, I have a very high precision on 1 angstrom, yeah? But it might be in the wrong place. In some cases, it might be okay, yeah? But in many cases, it won't. You cannot rely on it, cannot trust it. That's why it's better to use something that you can really trust.
Great. Somebody else have a question?
Thank you for the fascinating talk. I have one question to this no off states needed. How sensitive is it to like fluctuations between the two different dyes? So if you're scanning over it, does it change the distance you reach, or?
Yes, that's a very good question. Yes. So what we need, or what we prefer to have, is fluorophores that are kind of equally bright. Yeah, that are kind of equal. So of course we would still, you've seen the experiment was done with ADO 647N, or you could use Aberes star 635B. Why? Because this is fantastic dyes. Yeah, there is very little, say variation in the brightness. Of course, if there is variation in the brightness, you have to find it out and then compensate for that. You can compensate for that. But of course, you would still like to have fluorophores that are about equally bright. Okay. Yeah, that is definitely the case.
Could you basically average it by measuring it multiple times?
You could, yeah, sure, that's what we do. Okay. Yeah, of course, yes. Yeah. We average, and so over time. So if you ask me what is the time scale which we can separate, it's of the order of milliseconds. Ideally, I want to have it down to microseconds. Yeah, but we're not there yet. But conceptually, it will go there at some point. So at this point of development, we are at milliseconds, which is not bad. It's not bad. Yeah, but keep in mind, our field of view is terribly small, size of a protein. Yeah, we're talking about 29. The closer and the denser the fluorophores are, the physics tells us the denser the fluorophore, the better it is. Why? Because you have more signal, you have a stronger modulation, and the background level matters less. So this is the kind of change of thinking. This is why I think it's a conceptual breakthrough. Yeah, I'm not saying that easily. It's a conceptual breakthrough because who the heck thought 3 years back that it would be easy to separate something at a distance of 8 nanometers or 5 nanometers as opposed to 50? And everyone has said 50 is easier to separate than 5. No. It's the other way around. That's why it's a conceptual breakthrough.
Thank you.
Like expansion, by the way.
If you have more questions, please come to the microphone.
Hello. Am I audible, right? I have 2 fundamental questions. So one of that is, when we use min flux, because you're saying it depends on the number of photons, what I would understand, it's in the concept of energy, and you're using the live cell, like 37-degree heat transfer. So do you see any difference with the cold sample min flux, and like when the temperature is cold, and when there is a heat, 37-degree? So do you see any difference?
So all the experiments were done at room temperature, full stop.
Yeah. Okay.
Yes. So I haven't heated it up because there was no...
I mean, 37 means, what I'm trying to say, not heat up, just like because more energy transfer, so number of photons would be more. So do you see any difference with the...
I might not have understood your question, but I think it's very important to understand energy transfer in the case of 2 identical molecules, min flux, does not matter. Because the concept just draws on the position of excitability. And this is a fundamental difference to having a red and a green colocalization. That's why I say it's conceptual breakthrough. Because if you want to look at small distances, and you get FRET, and you look at the stuff that you have on a camera, the diffraction blobs, they're distorted. It is distorted because you excite this one, but you get signal in the other one. So you cannot really trust them. So it's okay to say with the standard methodologies below 10 nanometers, it's getting hard. Here, no. Yeah, you can have 2 of them. Excitability is the only thing that matters. There might be energy transfer from here to here, but we don't care about which one emits. Yeah, even if there is FRET, we don't care. We could place, in essence, we won't do that because it's silly, the detector right next to the 2 molecules. We would get conceptually the same results. This is the decisive thing. Yeah, so the detection doesn't really matter. So why we put the burden of localization, on the excitability and take it away from the emission. And that is a game changer.
Okay, thank you.
I know that people have cooled, they went down to, I don't know what, like there's this method which was published by the group in Erlangen, and they cooled it down to get high localization. I think, in my view, I wouldn't do that because the optics is bad if you cool something down. You have a lot of drift. And the whole purpose of cooling down is to get a high number of N. But why would I do that if I don't need it and can stay at room temperature and much faster?
Because my concern was when we have like the room temperature, what we assume that energy also degrades, like the number of photons. I'm not sure. Like
Sure. You will get more photons if you cool down, exactly. But why would I if I don't need them? Yeah, it just complicates matters. Yeah, and I think actually this dimer experiment was done actually by Weisenburger et al. Yeah, in and Vahid Sandokhtar. Yeah. Basically, we repeated their experiments, but why would I cool it down?
I mean, my idea was just to like because we need more number of photons, so I was just.
I don't need them. Okay. I don't need them, and it freezes everything. I want to see functionality. I don't want to freeze things down. There's no purpose. Yeah.
Yeah, I was just trying to understand the concept. Thank you.
No, but it's a very good question because it tells us where the advancement is. Yeah, we don't need the cool temperature.
Just one more follow-up. Like when we kind of intake the fluorophore or something, you said we are trying to control the we cannot control the orientation. They are like free rotation. Can we envision something that can stop the rotation of those molecules? Like
I may have not understood you, but of course, if you freeze something, that the orientation is fixed, and then you have systematic errors. Of course, here you reduce it with interest, but if you have a normal look, you have systematic errors that are substantial. They are really big.
Thank you.
Yeah, really big.
Okay, thank you. I'm being told we have time for one more question.
I have actually just a simple question from a biological point of view. I like the concept of memflux is really nice. Yeah, I'm sorry, but we were tracking only one object. In a biological system, in an experiment, I mean, we have multiple moving objects. And actually, if the concept is that the donut is always moving, and actually the devil is actually is detecting always actually the negative point, I mean, how many points could be detected at the same time?
Yes, it's a very good question. So what I said, I mean, we do this methodology at the expense of seeing a whole field of view in parallel. Of course, there have been people, including us, thinking about parallelizing this. There is even a commercial system, I think, built by Iberia, actually, which has 2 donuts, to my knowledge. Yeah, that to track 2. Yeah, of course. Or you can jump electronically around with a donut to track several at the same time, because you can quickly switch the position with electro-optic devices. Yeah, for me, that's engineering technology, and it'll come at some point. Yeah, but I think the big advantage to be gained right now is to look at molecular complexes and look at their dynamics, and in their native environment, because just looking into structure, I mean, the community here does a great job with expansion. There's no doubt about it.
Great. Let's thank Stefan for his amazing lecture again. Great, thank you. It's so wonderful.
Thank you. Thank you. Okay.
Okay, thank you very much. We are now at the final points of today's program, and these involve that which all of us like. I was going to say we like most, but no, we like second after microscopy, money. It's about money and prizes. We have 5 prizes given by our sponsor CNRS, and our guys were going to put they're going to put the slides with the prize announcements and nominations very soon, and we have. Yeah, so CSR Biotech, just come up. And our judges, please. I mean, so I see 2 judges. Where are, so, yes, please, Michael and Ute, yeah? Wonderful. We have a very international and highly respected committee that chose the prizes, and a sponsor that gave them, and we will have 3 prizes for posters, which I will deliver, and then 1 prize for social media, which I don't understand, so Ali will deliver that prize, and 1 more prize for the photography activity that has to do with beauty, and that also I don't understand, so that's also going to be presented by Ali. So, how are you guys doing with, oh, look at that. So, yeah, so maybe we just... Yes. And maybe you give me the, let's start with the poster prizes. Let's not keeping the people cooking for too long. No, that's, so that's the third prize. No, this is a third prize. This is a third prize. Okay. Okay, well, so you have to wait a little longer to get your money. I am going to introduce the people because it takes longer. I'm going to introduce the people. Oh, they ruined it. Okay, Matthew, Jana, and who was number 3? Just come. Matthew, like us, come here. Yeah. Prize number 3. There you go. You get your nice diploma and the money sometime later. Yes. So yes. No, but I mean, I think if we can also with the judges, and you, just I'm just holding the microphone, I'm not doing nothing here. So and you, the judges, go on the other side, and then, yeah, that's life, It's a little bit. It's in honest fun, That's all. So, goody. Svilen takes a picture because I'm getting tired. Huh? And we. Okay, and we sneaking into the picture, you guys. Okay, good. Okay, second award. Second award, I already said it. Jana from Würzburg, Jana Eils. So Jana, congratulations. Second prize, silver, silver color. Have a photograph with the committee. Wonderful. Everybody smile, sort of. Excellent. Okay, now the first prize. The first prize, gold. I don't know. Have you seen this already? Have you seen the name already? You probably seen the name already. Okay. It starts with Benjamin and ends with Liffner. So, who is Benjamin Liffner? Faster, man. We have a strong schedule, so, move it, keep it moving. Congratulations. That's your award, and let's take the picture. Money, as I said, later when we figure out how to actually do the money transfer, which will take a while with the German administration. So yeah, thanks a lot. Okay, now we have the 2 prizes, and Ali, you will now deliver this. Okay, well, you want me to deliver it? Okay, help me with it. Okay, just give me the paper thing, Yes. Yes, this is the correct one. So, stage award. The stage award. It's for looking pretty and taking lots of pictures at the stage and then making us proud that we actually put this Hollywood thing here and somebody actually used it. So one person, the name, it's kind of the letters are as in R&R, rest and relaxation. It fits. Ronia Rem. Ronia. And congratulations. There you go. And let's take some pictures. Let's take some more pictures. Okay. Goody. And I guess, the last prize of the day is a social media award, presumably for somebody that spammed the entire community with needless and useless things. I guess, do we see anything? Do we see any pictures that the people posted or something? Oh, no, we don't. Okay. We didn't have time. Okay. But then let's see. Oh my God, this cannot be. I am astounded to be basically, I'm Eastern European, Svilen is Eastern European, Ali is from Middle East, it feels like a family affair, and it is. The prize goes to Abed, Ali's brother. You come. So, Ali insists he was not in the committee. I cannot be in the committee because I have no idea about social media, but I know that Abed knows. So, yeah, it's a fair prize. Congratulations. Okay, let's clap for all of the guys tonight, and we're done. Yeah. And of course, we thank the sponsor CSR who made this possible. Otherwise, the prize would have been a hefty handshake from me and about it. You know, no money. Not a very attractive price. So, other than that, I think we are done with the program for tonight. There will be dinner again at 7:15, and tomorrow we start bright and early again at the hour of 9. And that's the last day, so let's make it count. Thanks a lot.